matters. So we say that anatomical sciences are vital in forensic science for education and investigations. So anatomists who are interested in forensics should collaborate. And I think this whole CME, the concept, is about to sensitize our anatomists to collaborate with forensics. So happy learning. And I'm happy to share with you that globally, there are integrated courses like anatomy and human identification courses, which are being conducted by the University of Dundee. And one of the speakers is from the same university with us today. So let us collaborate and create opportunity for exchange of ideas required for proper investigations in the forensics. Thank you. Thank you once again. Welcome to our all speakers, eminent speakers, our chairpersons, our moderators, and dear delegates. Happy learning. Thank you. Thank you, Dr. Kaveri. Please go ahead.
Thank you, Anita ma'am. So beginning with our first session, which is entitled DNA fingerprinting and its medico-legal applications. And this topic is going to be presented by Dr. K. Thangraj, our guest speaker. The moderator for this session is Dr. Farah Ghaus. An avid researcher, member of editorial board of many reputed journals, her biography continuously published for 5 years in Marquis Who's Who World, an American biographer. Your presence is an honor, ma'am, and I request you to kindly invite and introduce the chairpersons of this session. Over to you, ma'am.
Thank you, Dr. Kaveri. I feel privileged to be a part of this National Conference of Anatomical Society of India. I would like to introduce Professor Mohammad Shakil Siddiqui and Dr. Gayatri Rath, who are the chairpersons of this session. Professor Shakil Siddiqui was formerly the professor and head of King George Medical University and Eras Medical College, Department of Anatomy. He held various academic and administrative posts in national and international universities during his career of more than 45 years. He has a huge number of various society held very to his credit.
I didn't do the board.
Can you please change? He was the founder member of UP Anatomical Society, and he was also the fellow of Royal Microscopic Society, England, and Association of Clinical Anatomists, London. Next in line is the chairperson, Dr. Gayatri Rath. Dr. Gayatri has been the ex-director, professor, and HOD of Anatomy, VM, MC, and SJ Hospital, New Delhi, and North DMC Medical College and HRH, New Delhi. Her keen interest is in cell biology and molecular biology. She is an ardent researcher and has presented various papers at international conferences, including Canada, Australia, Singapore, Mexico, South Africa, UK, and many more. She is also the fellow of IMA and International Medical Sciences Academy and founder fellow of Anatomical Society of India. She was honored Dr. S. Bajaj Award for Best Proffered Paper at the Annual Conference of Medical Education of India, Chandigarh, and also awarded with the BC Royal National Award. She was given the award of Most Popular Teacher for the year 2001-2002, and that was given by IMA, New Delhi branch. Now I would like Dr. Gayatri Rath to introduce the speaker.
Thank you, madam. Good morning, Dr. Thangraj. Good morning, Dr. Thangraj.
Good morning. Good morning.
Just I like to tell your CV to audience. Dr. Thangraj was the former chief scientist and group leader at the Center for Cellular and Molecular Biology, Hyderabad. He was the former president of the Indian Society of Human Genetics from 2011 to 2015. He was the founder of Society for Mitochondrial Research and Medicine. He is the elected fellow of Indian Academy of Sciences, Indian National Science Academy, National Academy of Science. His main research interests are origin of modern human, cardiovascular diseases, and mitochondrial disorders and male infertility. He is a board editor of Mitochondrion, associate editor of BMC Medical Genetics, BMC Genetics, Tropical Medicine, and International Health. He is a member of the editorial board of the journals like Scientific Report, Human Genetics, and Clinical Genetics. He has published over 275 research articles, including Cell, Lancet, Science, Nature, Nature Genetics, etc. He has been honored with a number of fellowships and awards in the international journals with high impact factor. Out of them, this is the mainly in the Lancet, Nature and Nature Genetics. He has been honored with J.C. Bose Fellowship, Sun Pharma Research Award in Medical Science, Raman Research Fellowship, Lifetime Achievement Award, Distinguished Scientist Award, and plenty others. All the best, Dr. Thangraj. Now you may proceed for your presentation. Thank you, Dr. Thangraj.
Thank you, Dr. Gayatri, for the very kind introduction. Also thank Dr. Jyoti and her team for inviting me to make a presentation in this audience. So I request you to unshare your slide, therefore I can share my slide. Organizer, could you please?
Sir, you are permitted. You have been made the co-host.
I'll try to share and see if I could share the slides.
Sir, please do that.
Slide is visible?
Yes, sir, your slide is visible.
Yes.
I request all of you to mute your mic because there's an echo. Okay, I request all of you to mute the mic, and whenever you want to ask questions, please unmute and ask. And good morning to all of you. So what I'm going to tell you is a little bit about the history of DNA fingerprinting and how it is useful in various applications, particularly in the area of forensic and medical applications. So let me start with the word how the fingerprint came into existence. About a little more than 160 years back. So one of the justice by name Sir William James Herschel, he was a magistrate of Hooghly district in Jangipur in India. So he has used the actual fingerprint to identify the individual because he felt that people might cheat him. Therefore, he used fingerprint to make the person is the same as either he has given money or whatever it is. So he realized that the fingerprint is unique to every individual. And not only that, but there was a constant observation of that for the last several years. I think after 30 years, and this work has been published in a journal called Nature. That's what you are seeing. So that means not only the scientists and even administrators also can publish paper in Nature. But the DNA fingerprinting came into existence when Professor Sir Alec Jeffrey.
This meeting is being recorded.
Professor Alec Jeffrey, he was working on beta-globin gene, and one fine morning he found that when he hybridized a DNA fragment with the class of repeats on human DNA, and he found every individual's profile looks very unique. Okay, so that was the first time it was identified in 85 that one can able to identify every individual very uniquely using their own DNA. And subsequently, he used for many other things. And that was the time Dr. Lalji Singh, most of you are aware, he was the former director of CCMB and he was the founder of CDFB where I am at present. And he used to work on various snakes. His basic research was to identify the DNA which are responsible for sex determination. And he found there's a class of repeat GATA, what you see here, which are highlighted, that was clustering in a W chromosome. And while he was doing this, when Alex Jeffrey published the paper, there's a class of repeat can be used for human identification. Then he tried to use this repeat and he found that, yes, it also gives the uniqueness of every individual. And in fact, this is the very first case which was come to the light of or to the awareness of the judicial system, that there's a technique which can be used for human identification. Very first case came from a village called Telicheri in Kerala. And he was a contractor, and he promised her to marry, and she became pregnant. The child was born, but then he refused to marry, and she in fact came to know about the technique, and he wrote to director at the time, Professor Bhargava, that she wanted to have the test done to get the justice. So that was the first time it was established in the court that, and in the court also given verdict based on the report. And subsequently, there are many cases, and government, particularly Department of Biotechnology, was interested in setting up the separate laboratory exclusively for providing DNA fingerprinting service. And it was established about 25 years ago. And today is our 26th foundation day, and which I already informed Jyoti that it will be difficult for me to give a talk, but somehow I managed to be here. I just want to give you some background how because it's a CME, I believe that how the technique can be used and what is the basics to know about this technique. As a biologist, all of you are aware that in a cell, there's a nucleus. When the nucleus started dividing, and the nucleus, the cell started dividing, the nucleus will condense and become the thread-like structure called chromosomes. And human, we have 46 chromosomes, all the somatic cells. And in the germ cell, we have half of it, 23 chromosomes. In addition to that, so this is where there's a 3 billion base pair of DNA exist. In addition to that, there are small organelles called mitochondria. And in mitochondria, there are tiny circular DNA exist. While we have only 2 copies of nuclear genome, there are multiple copies, hundreds of copies of mitochondrial DNA genome in every individual cell. In the coming slide, I'll tell you a little more about this. Then, most important aspect is to learn the inheritance of the genome. I mentioned in the somatic cell, we have 46 chromosomes. In female and male, if you compare, the 44 chromosomes or 22 pairs are common. There's no difference. The only difference between male and female karyotype is the sex chromosomes. In case of female, there will be 2 X chromosomes. In case of male, 1 X and 1 Y chromosome. The Y chromosome is the one which carries a gene called SRY, sex determining region of the Y chromosome, which is essential to make the individual as a male. So when the cell in the germ cells start dividing, the meiosis, the 46 chromosome become 23, right? Both in egg as well as the sperm. Then at the time of fertilization, again, these two union and become 46 chromosomes. So we maintain the chromosomal number. But when the cell start dividing, there's no organized way of segregation of the chromosomes. We never know in what proportion every cell divides. For example, when one cell is dividing, chromosome number 1, this chromosome goes, this chromosome, whereas other chromosome. Whereas the next cell division, again, it can be different. That's why every individual is looks very different, because we share different proportion of the chromosomes. I mentioned about the mitochondrial DNA. In case of egg, the mitochondria remains there. Therefore, the mitochondrial DNA also remains there. Whereas in case of sperm, the mitochondria exist in the midpiece. But at the time of fertilization, it remains outside when the nucleus goes inside. Therefore, only the nuclear genome goes into the egg at the time of fertilization. So there is no contribution of paternal mitochondria. There are rare cases where always it's associated with the disease. But we all inherit the mitochondria or mitochondrial genome only from the mother. We will see that in detail. And I also want to tell some fact about the DNA. DNA is very stable, unlike other molecules like protein, carbohydrate, any other RNA, very stable. When you heat it, it will become single strand, your DNA is double strand, and when you cool down, become double strand. And can be isolated from any biological material, right from the blood, semen, saliva, urine, hair root, not the complete hair, teeth, bone, any kind of tissue. So that's another advantage. And doesn't change with the age. If you take blood of a just born baby and wait for 50 years or 60 years, isolate DNA and compare the profile, it remains the same. There could be some changes in a tissue where there's kind of cancer, or sometimes mitochondrial DNA variation, particularly in a tissue where the organ is affected. Only specific place, but otherwise the DNA remains the same. And pattern of an individual from every tissue of the body is same. So when you generate a profile, what I shown in all the tissue, 10 different tissue if we take of an same individual, if you generate the profile, DNA profile, it remains the same. Therefore, somebody make the crime, either the person leaves a drop of blood, a drop of semen, or 1 or 2 air sample, you can still able to identify the person. And the most important thing is that it shows a lot more variation compared to any other biological material, any other polymorphs. Like protein, you have very limited. Blood group, you have very limited. But the DNA, if you take, there are billions of places, millions of places where there's a variations between the individuals. But there are selected variation which shows across individuals are the ones which are being used. So any contact of an individual, for example, somebody is closing the envelope with the saliva, there could be few cells sticking onto that. Chewing gum can have few cells. Cigarette butts can have few cells. A glass or cup when you are drinking water or coffee or milk will have few epithelial cells sitting on the rim of the cup or glass that can be used. Now coming to the variations that are existing in the DNA. In a large chromosome, or the whole chromosome as such, if you look at the variations, you all must have studied, there could be a monosomy or trisomy. One chromosome is missing. That happens only with the X chromosome. If any other chromosomes, autosome missing, the individual will not survive. It might lead to very early pregnancy loss. But presence of one X, absence of either one more X or Y chromosome, person can survive with one X, called Turner syndrome, must have seen. And there are three well-known trisomies. The most known is trisomy 21, Down syndrome. Chromosome 13, trisomy, Patau syndrome. 18, Edwards syndrome. So these are the one chromosome extra that happened due to non-distinction at the time of cell division. That exists at a whole chromosome level. There could be large either deletion of a chromosome or translocation of chromosome or inversion of the chromosome. That always happens. For example, here, different chromosome segment you are finding here. Okay. Then if you go down a little bit, you might find variation existing at mega base level. For example, here, there's a tandem duplication. So BC is a one segment, for example, segment of DNA is A, another segment is B, then you have C. But B and C is duplicated again. It happens in the chromosome, particularly wherever there's a repetitive sequence, you expect that, or you often see that the entire chunk of DNA is duplicated or deleted. Same way is deletion here. Inversion, the orientation is changed. If you come down further, you have Alu element, you have mini satellites, you have L1, line 1 element, and so on. But you come down further, and this is what is very important for generating the profile because it shows more variations. It's called as short tandem repeats or microsatellite markers or STRs. So where you see there's a tandem repeat of a particular sequence. Here CT80, again you have CT80, CT80, it goes like that. Similarly here, in one chromosome, the next chromosome. Then if you go to single base level, it's called as SNP or single nucleotide polymorphism, where you see that the rest of the sequences are same, where you have there's a C here, and on the other chromosome you have T. Okay, it's called as single nucleotide polymorphism. So mostly this is what being used for DNA fingerprinting, and this is also being used. How the profile is generated? When the technique was invented, it was invented using a southern hybridization based method or RFLP method. It's called as restriction fragment length polymorphism. We used to use a specific restriction enzyme. So each enzyme has a defined sequence motive, only then it will cut. For example, if you take EcoR1 as one of the restriction enzyme, it cut wherever the sequence exists as GAATTC. Okay, so for example, this is one of the chromosomes, this is another chromosome. You have one EcoR1 site here, the complete GAATTC. Here also GAATTC, there is another GAATTC. Whereas in the other chromosome, the same sequence exists here. Here also the same sequence exists. But whereas here, instead of A, there's a G. There's a A to G polymorphism. Okay. So because of that, the restriction enzyme will not cut the DNA at this particular position. So it will cut here, 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 but here it will cut only this first position and the third position. So now itself in this very small example what I am giving, in this DNA you will get 1, 2, 3, and 4 fragments, whereas here you will get 1, 1, 2, this is a very big fragment, and this is another fragment. That means these two will show the same fragment of these, whereas this fragment will be in 2, which I'll show you in the coming slide, 2 small fragments, whereas this will be 1 long fragment. Similarly, the whole genome, if you are digesting with restriction enzyme, there's a lot of opportunity you get that some individual might have the restriction site there, some may not have. Okay, so that's how the technique is being developed. For example, here, so this individual has 2 bands here. This individual doesn't have one here. One is very light. That means these 2 joined together, it is appearing here. And this is a process of RFL, restriction fragment length polymorphism. It is a very long, lengthy process. One has to isolate DNA from either blood or any tissue, then use restriction enzyme, which I mentioned earlier. Then do the gel electrophoresis. Load the DNA here. The DNA is negative charge, then it will move towards positive. When you do that, because when you do the restriction enzyme, will generate large number of fragments, but you may not see any band like this. You will see like a smear. Like for example, if you take any book, any biochemistry book, you have the book is very thick, but there are many papers on that. Okay. Similarly, there are many fragments of DNA. That's why it's looking as a smear. Then transfer that onto nylon membrane because gel cannot be handled easily. So you transfer onto the nylon membrane. And the parallelly you prepare the probe, which I mentioned earlier. Probe is generally a repetitive sequence. In this case, for example, you take GATA or CTAT, then label that with radioactive material. Then hybridize the labeled probe onto the nylon membrane where there's a DNA. While doing this, make the DNA as a single strand and make the probe also single strand. And when you're doing hybridization, then wherever it finds its complementary sequence, for example, GATA. So complementary to that is CTAT. Okay, G always anneals with A, C, A always anneals with T. Okay, that makes a base pair. Then you remove all the unhybridized radioactive material, then exposed onto X-ray film, then you get bands like this. So this is the old way, a traditional way of doing it. But nobody uses that. That technique is almost has gone. I was mentioning that there's a short tandem repeats. What you see here, CT tandemly repeated. It's called as dinucleotide repeat. So 2 nucleotides are tandemly repeating. This is CAG, trinucleotide, tandemly repeating. This is a CTAT, again repeating tandemly, called as tetranucleotide. 4 nucleotides are tandemly repeating. And this technique is much simpler. You do design a primer flanking to the repeat. When you do the PCR, depending upon the number of repeat, you get the amplicon or amplified product. For example, here, CA repeat, it's shorter in this chromosome compared to this chromosome. So you get, in this case, shorter fragment compared to this. When you do gel electrophoresis, you will get band like this. This is shorter, so it's it moves faster, and this is a little longer, has additional 8 base pairs, so it moves slower. So it gives like. When you do in a population, then you always get very many alleles. Now I'm going to take you to even more simpler way to understand how one can read the DNA fingerprint. This is a pedigree, most of you are aware. When you put the square, that indicates male. When you put circle indicates the female. And you put the line here, that means they are husband and wife, and you put the line below, they are their offspring. And if you put square, that means son, there are 2 daughters. Okay. I also put different colors, so that indicates the 23 pairs I was saying. Okay, so one of the pairs is in this color, another one is in this color. Okay, similarly, one of this is here, another one is here. Then also there are other colors. This tiny circle here, you consider that's a mitochondrial DNA, which I mentioned earlier. Same way here, the mitochondrial DNA. I put different colors to show you the inheritance pattern. Then this is present only in the male, that is the Y chromosome. Okay. So now, if you look at the next generation, how the next generation is inheriting their parents' DNA, it will be much simpler to understand. So let's take the son. Okay, this color, that means half of the genome, has come from the father. Right? Second half has come from the mother. Whereas this tiny circle is a mitochondrial DNA. There is no contribution from the father. It comes only from the mother. Because he is a male, he inherits the Y chromosome from the father. Okay. The next child you see, you don't see the autosomal pattern exactly the same. It's a different combination. So she inherited this half from the father, this half from the mother. Again, this half from the father, this half from the mother. So if you look at the autosomal combination, everything is different. That's why I mentioned earlier that that's the reason in a family, except the monozygotic twin, everybody looks different because the genome is inherited differently with the different combination. Again, the next generation, you see that this goes, the Y chromosome goes as such without any change to all the males, right? Same color what you are seeing. The mitochondria stop here in case of the male. Male cannot transmit to the next generation. Whereas if you put similar extended pedigree, then you will see that the mitochondria goes into the next generation. Okay. So keeping in this mind, if you look at the actual DNA fingerprint profile using the STRs, there are 9 different loci. Okay. Take any chromosome, for example, chromosome number 1, as I mentioned earlier, one of the pair comes from the father, one from the mother. So we inherit the genome equally from both the parents. That's why we always check whether the person, we wanted to make an identity, whether this person belongs to this family, or he is the father of the child, or she is the mother of the child. Right? Take one particular locus, for example. So this is one particular locus. Okay, let's consider that this locus has come from chromosome number 1 or 5 or anything, although they are defined. So what you expect? This is the mother's profile. The second one is son's profile. The third one is father's profile. So we always put like that so that comparison will be easy. So what you expect, as I was mentioning, that any locus, if you take, locus is a particular place on the chromosome. So one of the allele, what you expect, should come from the mother. Okay, that's okay, because the number of the repeat is the same in both the chromosome. That's why it is in the same alignment. So this allele inherited from the mother. This allele inherited from the father. Okay. Similarly, if you take this allele inherited from the mother, and this allele inherited from the father. This allele inherited from the mother. This is from the father. Like that, you can actually, if you look at all the 9 loci, all are matching. Of course, now we use many more markers. Then you can conclude, yes, he is the father, or she is the mother, or this child belongs to this family. Okay. On the other hand, if you look at here, the same thing which I mentioned earlier. Let's take this locus. Okay. One of the allele is matching with the mother. This allele is not matching with the father. It should be either here or here. Okay, it's not matching. Similarly, you see here it's matching, but this is not matching. Same way here, this allele is matching with the mother. This either should be here or should be here. Okay, so wherever this arrow, it's not matching. So that tells that mother is the actual mother, but the father is not the actual father, biological father. So it's simple, straightforward. So now I'll tell you that how these techniques are used in forensic and medical application. In case of forensic, this is one example which I wanted to highlight, that this is a case of rape where the vaginal swab has been obtained and it was matched with the suspect and the victim. Okay, this is a swab. The profile generated from the swab, DNA isolated from the swab, and this is the DNA isolated from victim's blood, and this is the DNA isolated from the person who involved in the crime. If you remove the alleles which has been contributed by the victim, these two, and there's one allele existing here. There's one allele exactly the same, so exactly with the same, so it is added here. So leave this, and this allele is matching with the person who has been caught to be involved in this crime. Similarly here, and similarly here there are two alleles. So two allele, both alleles are matching here. Same way here. So when you have similar situation, for example, one allele here, so these two are from the victim. This allele, these two alleles. Okay, when you match very carefully, then you can be able to figure out that yes, he is a culprit who involved in this crime. Okay, I'll tell you example of how to use the Y chromosome, the same case, okay, later on. Then this is the example of application of DNA fingerprinting in medical legal application. So here, one lady has been diagnosed with breast cancer, and a sample has been sent to the pathology lab, and they confirm that yes, she has a cancer. But she was not believing on that, thinking that there's an exchange of sample, that sample is not belongs to her. And she wanted to confirm by DNA fingerprinting whether the sample which was processed and diagnosed with cancer is her own sample or not. So the approach, we try to isolate DNA from the tissue which was embedded in paraffin. It was difficult, but still able to get the DNA profile. Now you see, this is the DNA, the profile generated from DNA isolated from the paraffin embedded block. This is a blood sample of that lady. You can compare and see that both the DNA isolated from the paraffin embedded block and the fresh blood obtained from the lady are identical. There are two alleles here, one allele here, these two alleles. Similarly, all of these are identical, confirming that there's no doubt that this is in fact our own sample. You might ask question that why these alleles are smaller. Okay, it's gradually reducing because the DNA quality is not good because it undergone a lot of process. So DNA has been fragmented. The smaller fragment, because when you run the gel, this is the smallest fragment, and as you go, this is a largest fragment. So the large number of fragment are fragmented. Therefore, when it's amplifying, it is amplifying the smaller fragment nicely, but not in the bigger fragment. Whereas the fresh blood is amplifying very uniform. Okay, this is a technical thing. So now I want to tell you about the use of the Y chromosome. Now, what I was discussing is on about the autosome markers, which we inherit from both the parents. Now, the Y chromosome, which is present only in the male, right? And it has very unique properties. It's an haploid genome. There's only one Y chromosome. Sometimes, very rarely, you see in Klinefelter cases multiple Ys. And most of them are non-recombined. During the meiosis, the homologous chromosomes, as I was mentioning, that every chromosomes are existing in a pair. This is called as homologous chromosome. There will be exchange of the material between these two homologous chromosomes at the time of meiosis. That doesn't happen with the Y chromosome, majority of the Y chromosome, except in the pseudo-autosomal region. So therefore, it goes generation after generation without any change. Whereas the nuclear genome, there will be an exchange of genetic material. And it has a SRY, the sex determining region of the Y chromosome, essential to make the individual as a male. Then there are a large number of genes essential for spermatogenesis are present on the long arm of the Y chromosome. Azoospermic factor A, B, and C. There are many genes which are mentioned here. And half of the Y chromosome is full of repetitive sequence called as heterochromatic region. Okay. This is just some background. And most importantly, to see that haploid and non-recombined. And mostly it transmits generation after generation in the male lineage. This I showed you earlier that it goes generation after generation without any change. And this is the autosomal profile, which I showed earlier also. But if you do the son and the father's profile using the Y chromosome specific marker, this will be identical. You won't see any change. Okay. Because the chromosome, Y chromosome is identical in all the males of other particular family. Where it is useful when you have mixed sample, again, it's a rape case where there are multiple samples here. This is an autosome profile. There are many alleles or bands which are very difficult to interpret. But if you use only the Y chromosome, and that can tell you that these are different samples of the same incident. If you use Y chromosome marker, this is what you get. There are 4 different source of samples are identical. Okay, that means one single individual. And this is another individual where the pattern is different. This is a third individual. So that means there are 3 individuals are involved in this crime. But here, using only autosome, it's very difficult to predict. This I mentioned earlier, where you use the vaginal swab, victim sample, and the culprit sample. The same thing we used with the Y chromosome, where you see that the profile generated from the vaginal swab and the fresh blood sample of the individual who involved in the crime, and the pattern is identical. Wherever there's a peak here, also peak here. Okay. So again, this is confirmatory that this is a person who involved. So now I'll tell you that how the Y chromosome markers are useful to trace the paternity even after several generations, several hundreds of years. This is one best example that Jefferson was the 3rd president of United States of America, who lived between 1743 and 1826. And he had a habit of having slave. So one of his slave named Sally Hamming, and one of her son called Easton, he resembled phenotypically, I can say feature-wise, with Jefferson. There's a lot of news in 1873 in the Hoyt newspaper. But that has been now established recently, maybe about 10, 15 years back, using the Y chromosome markers. Jefferson is no more. Easton also no more, right? You see that when it was happened. So there are three groups. Chris Taylor Smith from Oxford, Mark Jobling was a colleague of Alec Jeffrey from Leicester, and Peter Denif from Leiden, Netherlands. So all of them, although they're all good friend of me, so they have started looking at whether Jefferson is the real father of Easton. Okay, they took all the necessary permission to conduct this experiment, but they cannot do with them, and they have started with the contemporary individual who are living now. So they are now trying to establish the paternity of Easton after several generations. So each number represents one generation. So we have 2 plus 4, 6, 7, 8 generations after they try to do this. Jefferson had one son. He died in accident. So they have used Jefferson paternal uncle lineage. Okay, each one is one, as I mentioned, one generation. So they use different markers. Biallelic marker like ARG, like SNPs. These are microsatellite markers. This is a repetitive sequence, and these are mini satellite markers. Again, large chunk of DNA which varies. This is Sally Hamming's son Easton and his lineage. Thomas Woodson and John Carr, these two are other suspects, could be father of Jefferson. They generated the profile and they had given the code, color code. Okay. Now leave away these biallelic and mini satellite markers and look at only the microsatellite. Look at the color code here and here as well as here. So these are all different. So one can rule out this. Now look at this. Except one mutation, they're all identical. You remember that I was mentioning that males, all the males of a family will have identical profile. You can see that all of them are having similar profile, and it is matching with Easton's profile, and they confirm that yes, Jefferson is the father of Easton. Now you can imagine after several generations, several hundred years, it could be possible. Same thing was done when Saddam Hussein was caught and his phenotype was totally different, and they used his sons, Uday and Azan's profile to match with and identified, yes, is the Saddam Hussein. I told you about autosomes, I told you about Y chromosome, now I'll tell you about the mitochondria. As I already mentioned, mitochondrial DNA, we all inherit from the mother, and it has many more applications. I'm not going to in detail because of the limitation of time. And this I already mentioned that this is a pedigree where you see that mitochondrial DNA goes from the mother to all her offspring, son and daughters. The son cannot transmit that mitochondrial DNA, whereas the daughter can transmit. So it's maternally transmitted. Again, one example where the mitochondrial DNA can be effectively used when the autosome or nuclear genome fails. For example, here, This is the case where somebody has been killed and has been thrown into a well. The body was recovered after several weeks, and it was totally mutilated. And the local forensic science department tried to establish the relationship. They could not establish because the DNA was very badly degraded. And you can see this is the DNA isolated from the mutilated body. Generated the profile only up to here, not up to here, very faint band. That means the DNA is very badly degraded. There are 5 families claiming that the deceased person belongs to that family because government gives the compensation. Now, how to use it? Because using only this profile, it's difficult to establish a relationship. And all the 5 families has been done separately. And one of the families where, as an example, I'm putting 2 brothers, mother was not there, the fathers has been put, but there's no information from the nuclear DNA profile. So we used the mitochondrial DNA profile. Here we used the mitochondrial DNA of the deceased person and 2 brothers. Also mentioned that all the offspring will have the same mitochondrial DNA, irrespective of male or female. So in this case, mitochondrial DNA, since it's a circular small DNA, it remains intact. You can also amplify smaller fragment and assemble them together. Here we could successfully analyze this. The first one and the third one are the 2 brothers. The middle one is the deceased person's profile. Now there are 3 dots here, and if you look at that, the nucleotides of that, there's a T here, and you have again T in the real brothers, whereas C in the deceased person. Similarly, there's A and A in the real brother, whereas you have C here. Similarly, you have A and A here, there's a G here. So in a very small stretch of DNA sequence, there are 3 different mutations, clearly showing that the deceased person is not belongs to this particular family. So it's straightforward. Similarly, we also use this for case of organ transplantation. Some of the states ask the donor and recipient to prove that they are the blood relatives. So where we do both Y chromosome and mitochondrial DNA, wherever it is necessary, to tell whether they are either maternally related or paternally related. So in this case, both recipient and donor have identical sequence, whereas here there are several mutations between those 2, although they say that they are maternally related, but it definitely says that they are not related. Similarly, here there's one case where somebody has been killed and thrown into one of the IIT campus. The body was recovered after several months. They could get only the bones, which was there in a big bag. And there's a family which was claiming that the deceased person belongs to their family. And the profile was generated. You can see there's a profile of the skeletal remains, the mother, father, of course, there's no father's contribution of mitochondrial DNA. This could be different, but whereas you see mother and the deceased person profile showed same mutation, confirming that yes, this person belongs to this family. In a long term, identifying the origin of the people, we use a similar technique. Who is related? Somebody is related for several generations back, hundreds of or thousands of years back. The mitochondrial DNA is the best marker to use. We wanted to trace the origin of the people. This is one of our earlier study. We used the complete mitochondrial DNA and reconstructed the origin of the Andaman Islanders. In fact, we concluded based on the mutations exist in the Andaman tribes, there are very unique mutations. And we confirmed that they are the first modern human who migrated out of Africa about 65,000 years back. So this is one of the conclusions. Similarly, this you might have read recently that Prince William's ancestry, maternal ancestry, mother has been traced. Prince Diana's maternal ancestry has been traced to India during the British time. So I'm not going to detail. Quickly, how much time I have?
Half an hour.
Half an hour more? Or? Hello, can you confirm how much time I have?
Sir, you have time.
Sorry? Can I have another 10 minutes? Yes, I'll go into the next way of looking at. So we discussed about STR, we discussed about autosomal marker, we discussed about Y chromosome markers, we also discussed about mitochondrial markers. Now we'll look at the SNP, single nucleotide polymorphic markers, which distributed throughout the genome, otherwise called as SNP, single nucleotide polymorphic markers. So here we used very effectively, there are about 1 million SNP markers to reconstruct the Indian population history. Okay. So what we did was that we used large number of population groups, and this is called as PCA plot, principal component analysis. And this tells that the Indians are in different clusters compared to Europeans, Africans, and Chinese. And while the Europeans, African, and Chinese all with the tight clusters, Indians are the gradient of clusters. Okay. So there are a lot more can be said about this, but I want to restrict because we are talking about the DNA fingerprinting, that when we don't have any clue about an individual, as for example, you will find something, some biological material, and you isolate DNA. Let's think that there's some incident took place in the border, Indian territory. You want to identify who is the criminal who came across the border and did this. You can in fact use the Y chromosome mitochondrial markers and also the autosomal markers. That will give you, for example, if it is a Indians, the sample will cluster here. If he is a European, it will cluster here. If he is a African, it will cluster here. If Chinese, it will cluster here. So this is one of the important way of looking at the tracing the origin of the individual who involves in the crime. Now I'll talk about the ancient DNA. Ancient DNA is very difficult to handle because the materials are thousands of years old. It requires a specific laboratory. So I'll tell you that how this can be used. Again, a couple of examples. There's a place called Rupkund, probably some of you are aware. It's in the Himalaya. And this is a lake, the Rupkund Lake, where there are several hundreds of bone samples. But nobody knows who are these individuals. There are different stories or hypotheses. People say that they are traders, they were walking across this, and something happened, and therefore they are all perished there. There are other hypotheses is that there is a Nandi Devi temple, people walked and there's a natural calamity, so they have gone and perished there. They also say that there's a king who went along with his troop. Again, something happened, they all perished. Then there was an endemic disease at the time, and those who have died have been taken and thrown into that. But that's very difficult to believe. That why? Because going there itself is difficult. So all of these are hypotheses, but how we use the ancient DNA technique, try to establish this. And we analyzed few bone samples, and it appears that there are 3 different source samples. One group is clustering with India, that means they are the actual Indians. Another group with the Europeans, okay, French and so on. One is closer to and Chinese. Okay, so this may be Southeast Asian group. They are Indian, and here we further investigated and found there are Mediterranean people. Okay, using the whole genome sequence. Earlier I told about genome-wide sequence. Is a whole genome sequence. We found that. Then one more example I wanted to tell you is that that Harappan civilization. There are large number of groups. Archaeologists are trying to excavate that place. And one group headed by Dr. Rashan Sinde from Deccan College. So they identified a bone sample. There's an intact bone here. Again, we did the whole genome sequence and to try to understand the source of that individual genome. And we found that that source consisting of the early Indian, that's the Andaman hunter-gatherers, and also the Iranian hunter-gatherers, not the Iranian farmers or hunter-gatherer farmers. They are much older, but the sample itself is not very old, around 3000 BC. But there are so many inferences can be drawn from there. One is that the agriculture must have started independently in India, okay, because they are not part of the Iranian farmer migration. Of course, they could have come much later. So that's all I want to tell you. I hope I kept my time. And if you have any question, I'll be happy to answer. And thank you very much.
Thank you, Dr. Thangaraj. Now I would like to ask Dr. Gayatri Rath and Dr. Shakil Siddiqui sir to ask any questions if they have.
Yeah, I think no one has a question. Looks like.
One question I like to ask.
Yeah, please.
Yes, you have described it in a very simple way, though it is a very difficult thing to carry on, because DNA analysis is not a very simple thing. one, and you have explained the history, you have explained the different markers and how the Y chromosome is responsible for the finding out the paternity, even several generations. It is a very, very interesting thing, and it is eye opener. Just I want to know that suppose in case of the postmortem cases, generally in the forensic department, you get the postmortem samples. And in postmortem samples, generally we have seen what I have noticed, that most of the samples are degraded and most of the samples are autolyzed. And how authenticity you can give in giving the DNA report? How much authenticity will be there?
I think please others could mute the mic, it will be more clear. I think there's an echo coming. Your question is that if the sample is degraded, and when you generate the profile, how authenticity is. Yeah. If the DNA is not good, then you will not be able to generate the profile.
Yeah, that is the thing.
Right. Then there is also a way to do that. For example, I was telling about in the degraded DNA, there's a possibility that if you do the routine thing, any forensic lab, they may not be able to do that. Being a research institute, we can devise a method. For example, if the mitochondrial DNA is a 16.5 kb, if you are using 1 kb region of the DNA, you may not find any intact DNA for that 1 kb to amplify. Whereas you can design a primer to amplify only 150 base pair, very, very small fragment. Then you can also design a primer overlapping that 150 base pair, small fragment. Like for example, in the tiny the flower garland, you add, no? Similarly, you can generate overlapping fragment, sequence them, and you can assemble them together, but still you can able to generate. Okay, so that's the way we do it.
But it is, or you can go for different sampling from the bone, from the hair, from the different.
But if the bone is there, then it's easy because muscle DNA will degrade very fast. Bone and the petrous bone is a very good source for DNA. Even the ancient sample we get from the petrous bone, a tooth pulp, right? So some biological material is sufficient.
Yeah, because sometimes in crime cases, the samples you get very degraded. After months you get the sample. So I was thinking how you go on for it. Okay, thank you so much. It's a very interesting topic, and we come to know so many things. It is an really eye opener to me, because previously you used to think the fingerprinting means 3 types: arch and hole and loop types. And now the science has advanced so much, and really it is an eye opener to me and to everybody also. And from if you go deep into the subjects, and you can use different primers to find out different alleles, and you can find out the race, you can find out the origin, you can find out the paternity, etc. It's very good. Thank you so much.
Thank you very much.
Thanks, ma'am.
Thank you, Dr. Jyoti. Now I would like to summarize the talk. I must say that the talk was very enlightening, and I came to know many of the things which I was not aware of.
Okay, thank you, Farah ma'am. I guess there's a network issue or some technical error from their end. So before that, there is one question from the audience side.
what is the success rate of DNA sample retrieval with prolonged formalin preserved specimens? Sir, Thangraj, sir.
If samples are preserved in formalin, it's very difficult to isolate DNA, right? So depending upon what is the thickness of the tissue and how much it has penetrated inside the tissue, all these are matters. So there's no defined rule. Then we'll have to use different technique to get rid of all this, right? We keep trying until we succeed. Okay.
Okay, thank you, sir. As a thank you, really, in spite of your busy schedule, we came to know that you're having your foundation day at your institute, but you made your precious time for all of us. We really appreciate it.
Yeah. Thank you so much, and wish you all the very best. Thank you. Bye.
Over to Farah, ma'am.
Thank you. So the summary of the talk is DNA actually is a genetic material that we inherit from our parents. Similar to the human fingerprints, DNA sequence already varies significantly from person to person, and hence it is a technology that is being widely employed for individual identification. This technique has gained much importance due to its effective role in medical legal applications, and it's a powerful tool for understanding the basic facts and the aspects of the DNA fingerprinting. Dr. Thangaraj has very well elaborated the autosomal characteristics, Y chromosome, mitochondrial DNA, ancient DNA, and various other markers to identify the different unknown aspects of DNA fingerprinting. For once, I would like to tell you that DNA is very stable. It does not change with age, can be isolated from any material, and shows variation between different individuals. This genome is equally inherited from the father as well as the mother. And William Herschel was the first who used fingerprints 160 years ago on native contracts in India. These markers are very important to establish paternity. That was all about the summary of the talk. I would like to thank all of the delegates present in this session, along with the chairpersons, Dr. Shakil Siddiqui sir, Dr. Gayatri Rath ma'am, and Dr. Jyoti and Dr. Kaveri for helping us out in this conduct of the session. Thank you so much.
Thank you very much.
Welcome, ma'am.
Thank you, everyone. We appreciate your presence. And now, with pride and joy, on behalf of Team NATCON 68, I present the Certificate of Appreciation to Dr. K. Thangraj, Dr. M.S. Siddiqui, Dr. Gayatri Rath,
Thank you.
and Dr. Farah Ghaus. We appreciate your support.
Thank you so much.
Thank you.
Thank you.
Thank you for being with us. Thank you.
Welcome.
Continuing with our program, let's begin our next talk on the topic Forensic Podiatry, Foot and Footprint Science. Dr. Keval Krishnan will deliver his talk, and the moderator for this session is Dr. Nidhi Sharma, Professor, Department of Anatomy, Tirthankar Mahavir Medical College and Research Center. She is the founder member of Society for the Healthcare and Research Development. Lifetime Achievement Award. She received the Lifetime Achievement Award for the World Association of Medical Editor. It's our privilege to have you, ma'am, and I request you to kindly invite and introduce the chairpersons of this session. Over to you, ma'am. Nidhi, ma'am.
A very good morning to all my respected stalwarts of anatomy.
First of all, I would like to thank Team NATCON 2022 for giving me the opportunity to become the part of this wonderful academic fest. I'm sure that this conference would certainly induce new ideas among the participants, paving way for innovations in the field of medical anatomy. I would like to take the opportunity to congratulate the whole organizing team, especially Professor Preeta Manik ma'am, Professor Jyoti Chopra ma'am, and Professor Anita Rani ma'am, and the whole team for organizing such a wonderful event. Now, I would like to start the sessions with the warm welcome to the honorable chairpersons. Kindly share the screen, please.
नहीं गड़बड़ हो जाती है, मैं बाद में कर लूंगी.
I request Dr. Kaveri to kindly share the screen of the honorable chairpersons, the PPT of the honorable chairpersons.
Ma'am, it's already shared.
Okay,
Can you see that?
just give me a minute, please.
Okay, ma'am. Yes, ma'am.
Share. No, it's not visible, Dr. Kaveri.
Ma'am, just give me a second.
Sure,
Just sorting
sure. Thank you so much. Thank you, Dr. Kaveri. Now, there are no words to introduce our first chairperson. He is a remarkable teacher with experience of more than 50 years, a great researcher with publications at national and international level. I welcome warm-heartedly respected Dr. S.D. Joshi, sir, Dean at Shri Aurobindo Institute of Medical Science in Indore, Madhya Pradesh. Sir was elected as Vice President of ASI 5 times. He is the member of the editorial board of JSI. He was given KG Saxena and Memorial Award for publishing best papers in JSI. He was also awarded Dr. Shamer Singh Memorial Oration in 2012. Sir, I welcome you warm-heartedly in this session. Our next chairperson. Our next chairperson is the lady who is of international fame and an ideal person for the young anatomist. That is Dr. Shashi Raheja ma'am. Profession head, Dr. Baba Saheb Ambedkar Medical College, New Delhi. Former director, professor and head at Lady Hardinge Medical College, Delhi. Ma'am has co-authored 2 books of anatomy. She has chaired the scientific session at 18th Congress of the International Federation of Association of Anatomists, that is IFA 14, which was held at Beijing, China in 2014. She is a member of expert group of medical assessment and rating board of National Medical Commission. She was honored for giving best oral presentation in the 6th Asia Pacific International Congress of Anatomy and 13th National Congress of Indonesian Anatomists Association at Surabaya in 2011. She is the recipient of Tridecennial Award for Teachers on 5th September 2012 by National Board of Examination. Ma'am, I welcome you in this session. Now, I request respected Professor Shashi Raheja ma'am to kindly introduce our guest speaker. Over to you, ma'am.
Thank you, Dr. Nidhi, for your kind words. And it is my very proud privilege to introduce a great personality and a person of repute in the field of forensic podiatry, Dr. Keval Krishan, who is Professor, Department of Anthropology, Punjab University, Chandigarh. Next, please. He has done BSc Honors and MSc Honor gold medalist and a PhD from Punjab University, India. He is a former chair at the Department of Anthropology, Punjab University, Chandigarh. Recently has been included among the world ranking of top 2% scientists. And happy to announce that he has been positioned at 17th rank in legal and forensic medicine category in the career long and 5th rank in the single year impact in the world as reported by team of scientists at Stanford University, USA and Elsevier BV. Next, please. And he has devised many methods of criminal investigations which are being used in the criminal investigating process. Like we all know, I'm happy to announce that fingerprinting was first done in India, and very less importance is given to the forensic podiatry study. And Dr. Keval Krishan has worked very hard to devise many methods at the criminal site where foot can be used as a tool for identification, specifically in the interest of the global interest, where there are terrorist attacks and you have no identification features available at that time. And you can, trace the person from their footprints and from their gait at the site of crime scenes. And Dr. Krishan has worked very hard on this, and he has published more than 273 articles and served as a reviewer and referee panel for more than 113 international and national journal of repute. His research has been cited more than 23,000 studies, and his contributions in the Encyclopedia of Forensic Science and Encyclopedia of Forensic and the Legal Medicine. He has been awarded Fellowship of Royal Anthropological Institute of Great Britain and Ireland. It's a very honor for us to have you in the 68th NATCON of ASI, and he'll be speaking on his forensic podiatry, and over to Dr. Keval Krishan.
Thank you very much, Professor, respected Professor Shashi Raheja and Dr. Nidhi Sharma and the chairpersons of the session. Thank you very much. I hope I am audible to all.
Yes, you are.
Yes, sir. You are.
Can I share my screen, Dr. Nidhi?
Yes. So we are making it. Yes, sir. Now you're able to share.
I hope everybody can see my screen.
Yes.
Yes, sir. Now is it visible?
Thank you very much for all these kind words and a great, introduction to my work and me. Thank you very much. I am quite grateful to you and the NATCON people, particularly the organizers, who has allowed me to speak on this forensic podiatry.
Sir, kindly make your slide in a slideshow mode.
Okay, ma'am. Is it okay, ma'am?
Yes.
Yes, sir. Now it is. Thank you.
So at the outset, let me congratulate the NATCON organizers for organizing this great conference. I have never seen more than 500 people in the webinar or in any web conference. I am so surprised to see that more than 500 people have joined over there in your conference. And this is a great success. And I am grateful to the organizers that you allowed me to speak on my topic of forensic podiatry. And this is another, forensic podiatry is another aspect of anatomy, anthropology, and medical science. Because of the combination of these three subjects, this has become a unique specialty in forensic science field. let me tell you that forensic podiatry is considered as a rudimentary science. Footprint and footprint science, this is considered as a rudimentary science in India since long. And let me also tell you that now people, the scientists from India have done wonders on this science, and they are recognized all over the world. Many scientists now, they are working on forensic podiatry, not only in India, not only in the European world, but in USA also, and in Australia and New Zealand also. And many people have started working on this depending upon its value in the country, particularly in the USA as well as in the UK. I hope you can see my screen moving.
Yes, sir.
Okay. Okay. So let me define forensic podiatry. It is the application of sound and research podiatric knowledge and experience in forensic investigations to show the association of an individual with the scene of crime, or to answer any other legal question concerned with the foot or footwear that requires knowledge of the functioning foot. So it all revolves around the anatomy of foot. So this is particularly considered with the concerned with the anatomy and morphology of the foot. Every kind of evidence which we receive for forensic purposes related to the foot is called pedal evidence, And the main focus of this evidence, of this research or this investigation on foot, footprint and footprint, foot and footprint, footwear science. This subdiscipline has emerged from the experience and practice of podiatric medicine, anatomy, morphology, and anthropology. So now this has become a specialized branch of medicine as well as forensic science for the study of diagnosis, medical and surgical treatment of disorders of the foot, ankle, and lower extremity, and using this knowledge in the forensic sense. Although there are a few forensic podiatrists, but we have established one American Society of Forensic Podiatry in America, in USA, where more than 50 forensic podiatrists are now working on this cause. And they have been helping the FBIs and the other investigating agencies in solving the cases relating to the footprint and foot signs. Basically, they are giving their evidence in the court of law, and maybe for the conviction and for other things, and the civil as well as for both the criminal cases. And they have made their name in the forensic podiatry field. My presentation will focus on 5 aspects of forensic podiatry. First, I will be talking about individualistic characteristics of the foot and footprints. That what are the individualistic characteristics the foot can create, the morphology of the foot and anatomy of the foot can create over there as footprints, and how can those be helpful in the crime scene investigation. And then prediction of body size, stature. Let us see if we can estimate stature and sex from the footprint also. Then sex determination from the footprint ridge density. This is a new concept which we have introduced called footprint ridge density. Let us see that how accurate we will be if we can determine the sex from the footprint ridge density from the footprint. And then we'll talk about stride length, step length, and gait analysis. Then we'll be talking about the association of foot with footwear found at the crime scene. Sometimes in a hurry, the criminal or the perpetrator, they leave the footprints, they leave the footwear at the crime scene, and then can we do something with those footwears or something like that? So let us begin with individualistic characteristics of the footprints. As everybody knows that you are a gathering of anatomists as well as morphologists, So you know better than me that foot impression is unique to an individual. Every foot impression is unique as the fingerprints are. Morphological and anatomical variations in the feet and footprints form the basis of this uniqueness. I hope everybody can understand that how the arrangement of the bones in the foot as well as the morphology of the foot can create a unique footprint at the crime scene. And this is all about the shape, size, alignment, and relative lengths of the toes, humps, creases, margins, cuts, cracks, pits, flat-footedness, as well as deformity in the foot. All these things are helping in the crime scene investigation. And whenever we receive any footprint for examination, then we can see all these features and we can relate those features to the criminal. These are the various regions of the toe of the foot, which can be quite variable. And you can see if anything like this is impressed over there at the crime scene. So can they be unique? Can you please see the structure of every toe? Can you please see this line? The hump line is quite different. And can you please see the distance between these toes? This is also variable, and this remains a unique feature. And can you please see this part also? This is also unique. And can you please see this line over there, which makes a unique line over there? And can you please see the impression of each toe? So all these are different in different individuals. So this seems to be a small thing, but this can help a lot in crime scene investigation. When even a single toe can be impressed at the crime scene, then we can reach up to the criminal. That depends upon the investigation process and other things. Can you please see this typical toe line curvature? This is also unique to an individual. Some of the individuals, they are having this typical toe line curvature. And can you please see this space? This space is also unique. And can you please see the instep region? Instep region is not at all impressed in this case. So this is also uniqueness as well as the individualistic characteristic of the footprint. And can you please see the humps? There may be 2 humps, there may be 3 humps. There are 3 humps over there in this toe line. And can you please see this? And these all are unique to a person. And these phalange marks, sometimes these phalange marks are impressed over there in the footprint and sometimes not. So these can really help in the crime scene investigation and in defining the footprint of a criminal or a perpetrator. Can you please see this line, this ridge? Can you please see this one? So this is quite different. In every foot, this is different. So can you please see the crease marks? There are many crease marks on these footprints. Although these footprints are flat-footedness condition is there over in these footprints, and the many crease marks, different crease marks and numerous crease marks are related to the flat-footedness also. So this is one characteristics as well as the combination also of these 2 characteristics. Can you please see these crease marks? And the shape of this region is quite different in different individuals. So this is, although seems to be, you might have all seen the your footprint also on sometimes on the floor or sometimes on the floor where the water is there, a little bit water is there, and you can see your footprint. And you might have not observed these kinds of things in your footprint. So these kinds of things may also be unique in your footprint. Just have a close look when you happen to see your footprint. And these crease marks, the vertical crease marks, so these are also helping. Can you please see the second toe is the larger, the length of the second toe is greater than the first toe. So this also varies. Can you please see this distance? This distance is also similar in both the footprints of the same person. Okay, so there may be bilateral variation or there may not be any bilateral variation in these, but in many of the footprints, there is bilateral variation. Can you please see the various pits? Although these pits and cracks may not be permanent, but there may be, these may be due to some kind of maybe barefootedness or some kind of, when we go to the fields or when we go bare feet to the fields, and these are developed as a result of maybe walking bare feet. But these may not be permanent, but immediately can help us in crime scene investigation if something like this can be found. And can you please see the fifth toe is not visible in the in this person. These are the person. These are the footprints of the same person, and the fifth toe may be the riding toe, or that may help in a certain kind of uniqueness or certain kind of association with the crime scene. Or in this, you can see the instep region is not at all impressed. So this kind of footprint is usually found, and we can see this may have about 20%, 20 or 22% in the whole population. Like if the instep region of the footprint is not impressed at all. And now you can see these footprints. These footprints are not having the fourth toe. And fourth toe may be riding on the third toe or on the fifth toe. So it means the fourth toe is not visible over there. And sometimes this happens, and this may lead to certain uniqueness and certain individualistic characteristics, which can tell us, which can give us lead in crime scene investigation. And this is how we compare. Suppose this is the footprint found at the crime scene, and you can see this in this case, particularly basically in all the cases, we can find this kind of blurred kind of footprints at the crime scene. So this may be all blurred, but we have to make this maybe visible with certain chemicals or by using certain things over there at the crime scene. And then we can compare each and every part of the footprint available at the crime scene with the known footprints. And suppose this is the gap over there, and this gap we can see if this gap is available over there or available over there, and then we can match it. And we have to match at least 20, 25 or maybe 30 features to give the evidence in the court of law. Like in fingerprints, we have to match the 8 characteristics, 8 ridge characteristics, but in footprints, we have to match more than 20 characteristics so that we can give our evidence in the court of law. And then another aspect is the estimation of sex and estimation of stature from the footprint. Can we accurately define the sex of the person, or can we accurately estimate the stature of the person on the basis of the footprint, or on the basis of maybe the foot available at the crime scene or at the mass disaster scene? As you know, the size of the foot is directly related to the body size of the person. Everybody, I hope, knows about the allometry of the human body, that each and every part of the human body is related to the other part of the human body. And similarly, the foot is related to the human body. Importance, we know that what is the importance of estimation of stature in personal identification. If we can estimate the height of the person from the footprint, then we can, assume that we have reached up to some kind of conclusion that we can exclude some criminals as well as the perpetrators. Then stature is the linear dimension of the skull, vertebral column, pelvis, and bones of the lower limb. So everything in the body, every skeletal part in the body contributes to the height of the person, contributes to the stature of the person. So stature depends on all these things, not only on the foot, not only on one part of the body, one bone of the body, but it depends upon the whole body. So there is a definite relationship between the stature of a person and various body parts, such as head, trunk, length of the upper and lower limbs and their segments. And so on the basis of these, we can correlate stature with all these segments of the body. So when we calculate the height of the person, so we have to see the correlation coefficient. We have to see whether that how much that portion is correlated with the height of the person. And can we calculate the regression equations or regression lines out of that? And how far those regression lines and regression equations are reliable? So can we do that? So estimation of height helps in narrowing down the pool of possible victim matches. So this is one of the aspects. And many papers in the literature are now being published over there on the basis of estimation of height from the footprints. And I encourage particularly the young anatomists and anthropologists to work on this aspect so that they can devise some new aspects of this kind of relationship of the human foot with the whole body. Then sex determination. Although sex determination is very difficult to determine from the footprint dimensions, but we can attempt, we can estimate. Okay. We tried to estimate sex estimation from the this heel ball index. Can you please see this? This is the breadth of the ball, and this is the breadth at heel of the footprint, and of the footprint also. So this has certain relationship, and this index has been calculated. The heel ball index has been calculated, and this paper was published in Journal of Forensic Sciences from Blackwell. And this paper has received many citations, and many people are using this paper in the crime scene investigation also, particularly in the European countries as well as in the America. And because they usually give this evidence as a weightage in the court of law. I don't know about our system of identification and our system over here, but we can corroborate this evidence on the basis of other evidences found at the crime scene. So in this, the index is larger in females in both the feet. And this may be utilized in sex determination when part of the foot is brought for medical legal investigation or in forensic investigations. These are the basic measurements of the footprint or the foot when we try to estimate the height of the person and when we try to estimate the sex of the person. Maybe some people have tried to estimate body weight also. So these are the length measurements and these are the two breadth measurements, which are quite helpful. And we have borrowed these measurements from Robbins et al. Robbins, she worked on the footprints long back in 1985 to 1990 in USA. And she developed certain these methods to estimate the height as well as the sex from the footprints. Recently, can you please see the BMI, relationship of BMI with the shape and size of the footprints? And here we can relate the body weight of the person with the footprints. Can I give you one example? That suppose we have found a footprint at the crime scene and we want to know that the body size, can the body size be estimated from the footprints and the vice versa? Or the body size of the person or the footprint can be estimated from the body size. So recently I guided my PhD student and the degree is awarded now, and she worked on this aspect. And this has come out to be a very good study, and many papers are published now in Science and Justice, an Elsevier journal, as well as in Forensic Science International on the basis of this aspect. And we are now expecting a book on this so that we can write on various aspects of the body weight as well as the estimation of body size from the footprints found at the crime scene may be on the basis of the normal range or pre-obese persons and obese persons. I hope this is a very good topic for all the anatomists as well as for anthropologists to work upon. And if they want to know more about all these things, they can contact me, and they can devise certain instruments and equipments also, just to know the size of the foot, and just to know the plantar pressure distribution. The plantar pressure, can you please see the plantar pressure distribution of this obese person? And this is not the flat-footedness character. This is not the flat-footedness condition, but this condition, flat-footedness is just due to the fact that the person is obese. Okay, I hope you can understand all these things, and we can work on these things. Then sex determination from the footprint ridge density. Although many papers are published on the sex determination from fingerprint ridge density, and since last 20 years, many people are working on this aspect in Europe as well as in America. And now we have also worked on this aspect. We have been working on this aspect since last 5-6 years. And then we thought that can we work on the footprint ridge density also? Sometimes we can find a very fine kind of footprint from the crime scene like this, where all the ridges are visible and available. And sometimes the footprint may be, found in a blood or something like that, where these ridges may not be available. But sometimes when we suppose when we are illuminating some of the fingerprints at the crime scene, and when we are putting some of the chemicals over there, and in the similar way we can define certain footprints, we can illuminate certain footprints also, and we can immediately take the photograph of the footprint, and then we can preserve them, and we can have an idea about the footprint ridge density also. What is this? This is a new biometric parameter of the footprints. And similar friction ridges or epidermal ridges, as in case of fingerprints, are present on the palms, toes, and on soles of the humans, and thus the palmar and plantar surfaces also leave impressions like footprints like this. The finger, palm, and footprints are commonly recovered from the crime scene, as you all know. And ridge density has been noted, density of the ridges, how we measure it. And ridge density is defined as the ridge counts in a defined area on a fingerprint, palm print, or a footprint. So this is a ridge count in a defined area. Suppose we take a defined area and we take the ridge count of that area, and in a particular manner, in controlled conditions, and then we can see whether those can be different in females or males. Why this difference is there? Females tend to have finer ridge tails than males. This is the genetics of the female. Therefore, females have more ridges than males in a defined area of a fingerprint, palm print, or a footprint. Scientists have made use of this principle in distinguishing sex of a print. Although we cannot be very much sure up to the sex of the person, but we can reach up to some kind of conclusion, like up to 70% of the accuracy, that we have defined the sex of this print found at the crime scene up to 70 or 75% accuracy. If we can do that, then the focus of the investigation may be restricted to a particular sex only. And then we have reduced the work of the forensic officer or a police officer. This is just an example how we are doing this in the fingerprint, and we can do that in footprints also. In case of male, you can see the ridges are quite wide. In case of female, the ridges are quite narrow. In a defined space, these ridges may be more in females, and these ridges may be less in the males. This is our paper published in one of the journals, great journals in forensic science, the Journal of Forensic and Legal Medicine. And We have published this paper in the journal Homo, the Journal of Comparative Human Biology. This is the paper, and you can see at different points, we have taken these points at different species. And you can see, you can also take some other aspect. You can also take these, ridge density at some other part of the footprint. We have initiated it, and you can take the other things also. And in these, we have found that in a female, the ridge density is always more than the males. So we have found an accuracy of about 70%, but not more than that. But that is also sufficient in clueless, murders as well as in crime scene investigation. Then we can talk about the stride length and step length and gait analysis. Sometimes we can find a series of footprints at the crime scene, and from that series, can we define the gait of the person, or can we define what the person was doing at the crime scene, how he moved at the crime scene, and what he was doing at the crime scene, and from where he entered and what he picked up, and is there any difference between the footprints when he came out of maybe from a house after doing a burglary, when he came out with a gunny bag on his shoulder, and his foot impression was changed or not? Definitely there will be a change in the foot impression. If he is wearing shoes, that is okay, but sometimes they don't wear shoes because they can see that the shoes can create problems, and so they walk barefooted. And when they walk barefooted with some gunny bag of 20 kg or something like that, so we have published this paper, and we have taken various situations, various conditions over there, and this paper was long back published in Forensic Science International in 2008. And of course, that paper received many citations. More than 100 citations are there of that paper, that how the body weight and the other additional weight can affect the footprints. So what is the difference in the gait pattern? Can we define that also? So gait analysis is a systematic study of the human locomotion, and everybody knows that the bipedalism has been acquired in the process of human evolution. And for the very first time in the human evolution, Australopithecus, for the very first time, they walked bipedally. And since then, the gait of the humans is unique. You also observe, I hope, when you go for a walk, that if somebody is walking, if you are walking maybe at night or maybe in the early morning, and you can see some persons, they are walking, but you recognize them from their walking, that is the person. Same person was there yesterday and same person was there day before yesterday, because of his unique gait, because of his individualistic or her individualistic gait, you can recognize that person. So the gait is not only depending upon the footsteps and footprints, but the whole body dynamics. So that is the anatomy and morphology of the body, which makes a gait of the person unique to a person. So the humans have a unique gait pattern. So nowadays it has drawn extensive attention because of the demand of video-based intelligent security surveillance systems called CCTV. Sometimes we can have the CCTV footage over there, and we can find out that the criminal has left the CCTV footage over there. And can we recognize that criminal on the basis of the gait? If the face of the criminal is not available at the CCTV footage, then can this help? Yes, there are many people who are working in USA as well as in UK, but I don't know whether in India somebody is working on gait analysis or not. Maybe some computer persons from the IITs they started working on the gait analysis, and I have also guided one student who is now writing a book on gait analysis, and I hope so that that book will be ready within 6 months or so, and we can also say that we have worked in India also on the gait pattern as well as on the footprint science. The identification of a person or person by their gait or features of their gait, usually from the CCTV footage and comparison of footage of a known individual. So we can, if the known footage is there, and we can compare that footage with the crime scene footage, and then we can see whether these persons, they can, whether the person was same or the not. Then abnormalities, then we can guess, we can have any clue about the abnormalities in the gait, then what kind of person he was, and whether he was having some abnormality in the gait, or he was having some abnormality in the, maybe in the body by the gait pattern, and what may be the age of the person. If the person is old, maybe more than 50 years, or maybe a young person, or an middle-aged person can be there. So we can have an idea. This is just an idea, and we cannot be quite accurate on this, and we cannot give the percentage that how much we can be accurate on this. Though, so gait patterns may be studied from the series of footprints. Movement of the criminals can be tracked at the crime scene, and then we can measure the step length as well as the stride length of the persons, and whether they are uniquely walking, or they are running, or they have some kind of abnormality in the gait or something like that. So all these features can be helpful. This is another aspect, association of foot with footwear found at the crime scene. We have worked on this aspect also. So I have completed one research project from the University Grants Commission, and I have been now publishing this in international journals of repute. At many international conferences, I have spoken about this aspect, that how can we associate the foot of the person with the footwear found at the crime scene, or can we do that? Is there any impression on the footwear found at the crime scene? Is there any impression like the footprint, like this? You can see the insole of the footprint over there. This is the insole. We have cut the that shoes, and then we have found some insoles, just like the footprint. Okay, then we can see whether these footprints are the same person's footprints who was having this shoe or something like that. So footwear can be left in a hurry by the perpetrator after committing the crime, such as sexual assault or murders. Okay, the forensic specialist can compare impressions found inside the footwear. So insole of the footwear. So I'm talking about insole of the footwear. I'm not talking about outside the footwear, which we are usually recovering those kinds of footwear impressions at the crime scene. Although those can also help, those are also part of the forensic podiatry. But this is also a part of forensic podiatry, which we have, found that with the footprints of the suspect and criminal can be associated with the crime. I have worked on this aspect, although I have not yet worked on the shoes, but one of my associated scientists over there in collaborator is working in USA on shoes. And now we are comparing these results with whatever I could find on the chappal. I could find on the slippers. This is my own slippers, and I have worn these slippers for about 4 months in controlled conditions. I did not allow anybody from my house to wear these slippers. Now you can see this is my own footprint, and how can this be associated? How these prints, how these weight-bearing marks can be associated with the footprints of the person. Suppose if we can find this chappal at the crime scene, And can we associate? Yes, we can associate up to 90 or up to maybe 100% accuracy with certain points. Okay. And I have marked these points over there. Can you please see this? And how I try to associate all these marks with the footprints of the person, my own footprints. And these are the, so we have worked on then chappals, about more than 500 chappals or so. And then we have seen how these are creating a unique impressions on the footwear, on these chappals, on these slippers. And what are the weight bearing marks? Is the kind of, sweating creating these weight bearing marks, or can we associate the weight of the person with the footprints, with the chappals also? So these are the questions which we are trying to answer, but we have not yet concluded on these aspects. So we are working on this aspect. And you can see all these weight bearing marks. And not only this, but the space, this unique space, which we usually find over there in the footprints, that is also there on the insoles of the chappals. And you can see all these. So these spaces, so this space is there. And can we create this? Can we see this? All these things. I hope you can see this very clearly. And all these impressions. So can you please see this space over there? And can you please see the weight bearing marks? Okay, this space over there. So this is the space. So all these things are helpful in the crime scene investigation. So individualistic and peculiar characteristics on the insoles of the footwear can be compared for the matching of the prints, and the criminal can be identified who left the footwear at the crime scene. Okay, that is all right, but there are certain limitations. Every science has limitations. So this science is also having many limitations. So we try to work on one aspect, but another aspect is left. That is okay. So suppose I can tell you on these chappals. So the prints which we are receiving in the shoes, we are not receiving those kinds of prints in the chappals, because these are open-ended and shoes are closed. And sometimes the shoes can be tight to a person, and shoes can be not very much tight to that person. So all these things, they can have some effect on these aspects. So thank you very much for all the attention you have paid to my presentation, and thank you for inviting me to deliver this lecture. And you can ask me any questions, any queries.
Thank you so much, sir. Your talk was very interesting, very informative, sir. Now I request Dr. S.D. Joshi, sir, to kindly enlighten us with his expert comments. Dr. S.D. Joshi, sir, kindly. Sir, please unmute yourself.
Thank you so much. It was a real pleasure to listen to Professor Keval Krishna. Not only that, it was very informative, but extremely challenging one. And a lot of topics have been given so that the younger generation can get into this and start with the work that he's already talked about, like the regional variations that can be there, or the various landmarks and measurements, whether footwear or any other one. It had been very, very impressive, very informative, and I'm sure it would be something which would definitely help the younger generation to learn from this, because earlier podiatrics, we always thought of only medicine or the surgical component, never knowing how very much positively important it is in forensic medicine, in identifying the individuals and separating them from the criminals that are there. We are really very happy that we are fortunate. We have heard this man who actually is very eminent anthropologist of international repute. We thank him, and there would be naturally some questions coming forth. And we also thank the organizers for having given us this opportunity to chair this particular session of this great and very great forensic portrait person. Thank you so much.
Thank you, Joshi sir, for your very expert comments, and thank you very much for these kind, comments and kind compliments. Thank you very much, sir. Thank you, sir.
We are grateful to you
Thank
for
you.
your. Thank you so much. Thank you.
Thank you, sir. Now I request Professor Shashi Raheja ma'am to kindly share her views and enlighten us. Please unmute Dr. Shashi Raheja ma'am.
Thank you very much, Professor Keval Krishnan, for your very enlightening talk. And it has generated so much of interest in everybody to do research on the, study of the footprints and the gait cycle. And soon you will find in India also, maybe it is rudimentary at the time being that we are only concerned about the fingerprints or the personal identification in the retina, iris, or, face reading. And soon to, deal with the terrorism. This footprint, which is available in some states like UK, Australia, you have seen that they are already doing in New Zealand, but India is not far behind. They will try to include the personal identification will be the footprints also, and to catch people, and they can catch at the crime scene. The same person, we heard that there's a bomb scare in one area and the bomb scare in another area. So the same criminal can be caught with the footprints by his gait or the, footprint, shoe print, or the gait cycle by the way of his walking, their foot, their leg, or even the upper body can be, taken care of, spine curvatures. All these features can help in catching up the criminals at the border side when they leave the area, or in the various, the sensitive areas of nanotechnology or the chip making, or in the pharmacy, wherever we find that these things are getting, very leaked out or things like that. And in the defense, they can be very useful. Thank you very much for your very informative talk, which has stimulated everybody to do something better.
Thank you. Thank you very much, respected Shashi ma'am, and for these encouraging words, so that the anatomists and anthropologists, they start working on various aspects of footprints as well as the foot signs, and we can also create wonders in India, ma'am. Thank you, ma'am. Thank you very much, ma'am.
Thank you, ma'am. Professor Keval sir, there are many questions in the chat box I would like to discuss with you. The first question is raised from Vice Principal S.K. Jain sir from Tirthankar Mahavir Medical College, Muradabad. He's asking how much it is safe to tell the general public the secrets of forensic science because it can be misused by the culprits.
Yes,
so your
right.
views, sir.
Yes, Dr. Jain sir, you are very much right. Now the criminals have become very much smart, very smart, because they know all these features. They know all these things as the forensic scientist is knowing. That's why the crime is going on. And the criminals, they don't leave anything, at the crime scene. So this is the fact that that you are saying that is it safe or not. I think we should tell our public also that these are the things which should, be taken into consideration. Suppose, if the police personnel doesn't visit the crime scene, and suppose the forensic scientist is not there, and he has not visited the crime scene, and then this is the public who is the first one to visit the crime scene, and who can help the police also. So I think the awareness is must to everyone. But we have to stop somewhere that these criminals, they also are aware of all these things. And
Okay,
I
sir.
hope, sir, Jan sir. Ji ma'am, Nidhi ma'am.
Sir, the next question is from Dr. Kalpana Chetri.
Ji.
She's asking, several studies document that changes in the fingerprint pattern is observed in various diseases. So can we rely on finger and footprinting for identification?
Yes, we know there are many diseases which can affect the fingerprints as well as the footprints. But, those diseases are quite rare. There are some skin diseases which can affect. This is, I hope there was a case few months back at the immigration center of the Delhi, that somebody wanted to go to USA, but his fingerprints were not matched to the fingerprints found in the system, because he was suffering from some kind of disease called dermatoglyphia. But this is quite rare. We can exclude all these things, that is okay, because there are certain limitations in every case. The fingerprints are also having limitations. We cannot be 100% true in fingerprints, but somewhere we have to rely upon the evidence. Is it clear, ma'am? Is it okay? Nidhi ma'am.
So I guess there is a technical error from the Nidhi ma'am's end.
I hope I am audible to all.
Yes, sir, you're audible.
Ma'am, other questions?
So the audience, they're asking, any specific difference between the footprints of criminal versus normal individuals?
I don't know, ma'am. Criminals are also individuals, ma'am. Criminals are also persons like us. And there cannot be anything else, but something can be related to the psychology. Something may be related to the psychology, but this is a purely biological thing, and this is purely genetically controlled. And I don't think that these may be related to certain kinds of psychology or something like that of the criminals or certain kind of things. I don't know, ma'am, about this, that this can be related to the topic or not.
Thank you, sir.
So there is another question in the chat box saying
Right,
that have some studies being done to correlate the footprints of neonates with the adult footprint, since in OBG we routinely used to ask the footprint of neonate. Though I don't know if the data is being maintained or not digitally.
Ma'am, can you please repeat it? I couldn't hear properly. I could hear just the footprints of the neonates.
So the question is, yes, sir. So the question is, have some studies being done to correlate the footprints of neonates with the adult footprint, since in obs-gynae we routinely used to take the footprint of neonate, though I don't know
The data is being
Yes,
if the data is being maintained digitally.
Ma'am, no data is being maintained in India, but we are trying to maintain data in such kind of data in USA. In USA, they have now started taking, there is a project going on over there in American Society of Forensic Podiatry. They are doing some kind of project that they take the footprints of the neonates, and then after 5 years, after 10 years, after 15 years, it's a longitudinal study, and up to 20 years. They are now doing up to 20 years, that whether any change in the footprint characters or the footprint characteristics is observed within these years or something like that. But we are not doing this. And long back, 5-6 years back, somebody from USA and one person from UK also, they asked me about this kind of data, but I searched everywhere. This data is not available. Nowadays also, we are taking footprints of the neonates in the hospitals just to distinguish them, that because we cannot take their fingerprints, but we can take their footprints, because footprints are also unique. It is considered that the footprints are unique like fingerprints or like handprints. So that's why we are taking footprints, ma'am. I hope the answer is clear. If anything, maybe you are absolutely right that digitally we have to create this kind of data. We have to make this database that if we can do this longitudinal study, then that will be very good. We can find out whether that is the same person who was there at about 5 years of age or something like that. And this longitudinal study can do a lot. And on the basis of this, we can work a lot on this aspect of footprints. So this is a very good question, ma'am.
Okay. Sir, sorry for the interruption. I think Professor S.K. Jain sir is raising hand. He want to ask them.
Yes, Jain sir.
Yes, sir.
Yes, Jain sir.
Sir, thank you so much for this wonderful lecture. And sir, I am also working on anthropometry.
Okay, sir.
And your lecture has opened many paths to work more on it, and especially on the foot. So in future, I would like to take your guidance to go further in this field. And thanks for answering my question also.
Thank you very much, sir. Thank you very much for these encouraging words. Thank you, sir.
Sir, there is one more question from Dr. Ravi Shankar Periswamy. He just wants to know, how can we correlate the footprints of syndactyly person with genes involved?
How can we correlate the footprints with, ma'am?
How can we correlate the footprint of syndactyly person with genes involved?
Ma'am, what is that? I couldn't understand.
Syndactyly.
What is that, ma'am?
Sir, the condition of having some in which all the fingers and toes are partly united. But the finger, there's some congenital anomaly with the fingers.
Okay, ma'am. We have come across, I don't know about this kind of congenital abnormality, ma'am.
No problem.
But we have come across many congenital abnormality while I was working on the footprints in my PhD.
Yes,
as well as my students are also working on that. And whenever we find that, and we take that footprint, and that footprint is considered as a unique one due to the congenital abnormality. Maybe the shape will totally be different, and that is very good to recognize the person on the basis of that congenital abnormality.
Okay, sir.
Isn't it, ma'am?
I
don't know about the abnormality, actually, that abnormality which she is pointing out. But it's okay. We can create a database of the congenital abnormalities of the footprint also, whenever we come across those.
Sure. So there is one very interesting question.
Right,
how to differentiate between the flat-footed and obese person footprint?
Okay. So I also did not know earlier that how to differentiate between these. When I came to know that the obesity can create flat-footedness, and I have just given this topic to my student, MSc student, that the normal, what is the percentage of the flat-footedness in the normal individuals, as well as how many of the obese individuals can create that kind of footprint. Like we found in that study, which I just mentioned, that one of my PhD students worked on that, about 2% of the persons who were obese, considered as obese according to WHO, but they did not create the same kind of persons the other obese persons are creating the footprint. Maybe, I don't know, maybe the structure of their footprint is like that, that their weight is absorbed up to the instep region only, but instep region is not fully impressed over there. There can be certain variation. There can be certain variation, but there is, I just tell you, in my study, I could find that there is just 2.5% of the flat-footedness in the normal individuals. This condition is 2.5% is there, the percentage is there in the normal individuals. But in the obese individuals, 98% of the population of the obese persons can create this flat-footedness. There is no difference.
So, thank you so
no
much.
Difference at all, because in all the cases, almost in all the cases, all parts of the foot are impressed. But one thing is there. Let me tell you who has raised this question, the person who has raised this question, that flat-footedness in the normal individuals is also associated with the numerous crease marks. Numerous crease marks in the footprint. I have shown just in my presentation that how these numerous crease marks are there in the flat-footed individuals. But that kind of flat-footedness, that kind of numerous crease marks are not there in the obese persons, because later on they have become obese, but genetically they are not obese basically, and genetically their footprint is not flat-footedness. I hope you can understand.
Yeah. So very convincing answer. I think the question was a very interesting answer of a very interesting question.
Very interesting
Yes.
question. Because I thought just 6 months back about this, and I gave the topic to my MSc student that we can work on that, and now she is working on that, and hopefully she'll be able to complete this within 3 months or so. Then we can discuss the findings with that person.
So thank you so much, sir, our esteemed guest speakers and chairpersons, for sharing your knowledge and making this session so informative and seriously very interesting. And special thanks to Professor Keval Kishan sir for such a fascinating and mesmerizing talk. Topic was very innovative, sir. Now, it's my pleasure to summarize this topic. Forensic podiatry is a new scientific discipline dealing with the foot and footprint inspections, which is generally encountered at the crime scenes. This science is applicable in forming an association between the podiatric knowledge and experience in forensic investigations with the legal questions concerned, and the legal questions concerned with the foot and footwears. Forensic podiatry contribute to the personal identification in crime scene investigation, and thus this kind of physical evidence can positively link a suspect to a crime, or it can prove one's innocence also. Besides this, by using different anthropometric methods like the stature, sex, body weight, we can also estimate all this from the footprints which we have recovered at the crime scenes. The second important thing which we conclude is that foot, since they are enclosed in the shoes, so they are the most common mutilated part which can be recovered in the mass disasters. This science will prove to be very useful in establishing the identity of disarticulated remains in the case of mass fatality incidents like terrorist attacks, accidents, floods, earthquakes, etc. So hence this topic carries both anatomical and forensic importance. So for this, Professor Keval Kishan sir, thank you so much again for sharing such a nice and informative topic. Now I request Dr. Kaveri to please continue.
Thank you, ma'am. Thank you, ma'am. Thank you very much for these kind words as well as for these encouraging words. And I want that many people now can take this topic of research. There are many, the research is open in this topic. And any topic can be taken and a good research can be done on this. And I am quite sure that a paper of an international standard can be published out of this. Thank you, ma'am.
Sure, sir. Thank you so much.
Thank you, Chairperson, Sir Joshi, Sir Reheja, ma'am. Thank you very much, ma'am. Thank you very much, sir, for chairing this session. And thanks for these encouraging words. Thank you, ma'am.
Thank you. Thank you, everyone. Dear audience, it is really impossible for us to take all the questions, but the questions which have been asked from your end, we are going to forward these questions to our guest speakers.
Yes,
and the
ma'am.
answers of the same will be shared to you on the WhatsApp groups. Thank you.
Right, ma'am.
Now,
You
I
can send
thank
me emails,
everyone.
ma'am. You can send me
Sure, sir.
emails, ma'am.
Sure, sir. Sure. Thank you.
Ji, ma'am.
Thank you, Cable, sir.
Ji, ma'am. Thank you, ma'am.
Now,
Ma'am,
on
commence
behalf
with
of
the session now.
yeah, sir, just kindly be patient. Just give us
Okay.
1 minute.
Okay, ma'am. Okay, no problem, ma'am.
On behalf of NATCON 68, it brings me an immense pleasure to present the certificate of appreciation to Dr. Keval Krishnan, Dr. S.D. Joshi, Dr. Shashi Raheja,
Thank you.
and Dr. Nidhi Sharma. We are really thankful to all of you. And now,
Thank you,
before we proceed, thank you. Before we proceed to our next session, I request all our admirable guests to kindly switch on their video so that we can create a memory here by taking these screenshots.
Thank you, ma'am. Ma'am, I can't hear you.
Thank you. Thank you, sir.
Thank you, ma'am. Thank you very
Yes, sir.
much.
Proceeding ahead to our next session, entitled An Introduction to Forensic Facial Reconstruction and the Drive to Advance the Field. Dr. Tobias Halton will be speaking about it, and Dr. Anand Mishra is the moderator of this session, a proficient and advisory member in various committees. It is our great honor to have you here, sir, and I request you to kindly invite and introduce the chairpersons of the session. Over to you, sir. Dr. Anand, kindly unmute yourself.
Hello, am I audible?
Yes, sir, now you're audible.
So it is my great pleasure to introduce Professor A. Sharif, who is a professor and head of Department of Anatomy, and he is also the professor in charge computer facility and telemedicine facility at All India Institute of Medical Sciences, New Delhi. He has been the former professor and head of anatomy in BP Torala Institute of Health Sciences, Nepal, and also in Oman Dental College, Muscat. He has introduced various facilities at AIMS, like the e-learning facility, the enteric nervous system research lab, the digital histology lab, and the clinical ecotoxicology facility. We welcome you, sir, as one of the chairpersons for this session.
Sir, your voice is quite not audible. Anand, sir, you're not audible properly.
Am I audible now? Am I audible now? The second speaker, the second chairperson for this session is Dr. Bala Subramaniam. He is the professor, Department of Anatomy at Johnson Medical College, Bangalore.
So you're not audible.
Anatomica Karnataka. He's also an avid blogger and a YouTuber also, and has thousands of subscribers. IT consultant in the domain of healthcare.
Anand sir, you're requested to kindly put the mic. So kindly bring the mic closer to your.
Am I audible?
No, sir, you're not audible.
They have received the
So the mic is too far. So kindly keep your. So kindly pull up the mic and.
Hello.
Sir, I'm going to introduce Bala Subramaniam. Sir, I'm introducing Bala Subramaniam, sir.
Okay.
Dr. Bala Subramaniam, Professor, Department of Anatomy at St. John's Medical College, Bangalore, an avid researcher and a founder editor of Anatomica Karnataka, a web blogger. His YouTube channel has thousands of subscribers. He is an IT consultant in the domain of healthcare e-learning and is certified, which is a certified digital content writer. Sir is a recipient of Founder Fellowship of the Anatomical Society of India. He receives Arjuna Award at Karnataka Chapter of Anatomist, Shushrit Gold Medal Award, which is a lifetime achievement. And sir have been also recipient of Professor Sameer Singh Memorial Oration in 2018 by Anatomical Society of India. It is our honor, sir, to have you here with us. And now I request you to kindly introduce our guest speaker.
Thank you, organizers. It's my proud privilege. Am I audible, please?
Yes, sir, you're audible.
Thank you. It's my proud privilege to introduce the speaker for this session, Dr. Tobias Holton. Before I introduce this esteemed speaker, during our postgraduate exams, I'm talking of early 80s, we used to have this question as craniofacial morphoanalysis as a short note question in our postgraduate examinations. Subsequently, I've seen it quite often in other postgraduate exams across the country and elsewhere also. The point is, beyond academic knowledge, we have realized that this is an area of a tremendous clinico-forensic importance, because using multimodal approach, you can actually predict the face of the individual whose details are not very clearly available. So in this context, it will be best to hear a stalwart in this field, Dr. Tobias Holton. Sir, it's my proud privilege to make an introduction of you to our esteemed audiences. He is a facial anthropologist, lecturer in the Forensic Art and Forensic Facial Imaging Center of Anatomy and Human Identification from the University of Dundee. The title of his presentation is An Introduction to the Forensic Facial Reconstruction and the Drive to Advance in this field. Now, he holds an MSc in Forensic Art and a PhD in Facial Anthropology from the Center of Anatomy and Human Identification, called CAHID in short form, from the same university, namely the University of Dundee, UK. He has been a postdoctoral research fellow for 5 years now at the University of Witwatersrand in Johannesburg, South Africa. During this time, he formed part of the Human Variation and Identification Research Unit and delivered specialist craniofacial identification services. And now, presently, he has been appointed the course lead for the MSc in Forensic Art and Facial Imaging program at the University of Dundee. He continues in the same institution. He is a researcher and his research interests in the fields of craniofacial identification, forensic anthropology, history of physical anthropology, and cultural heritage is noteworthy. To his credit, he has lots of publications in established international peer-reviewed academic journals such as the Forensic Science International and the Journal of Forensic Sciences. He is the third speaker in our current today's feast on a very, very interesting topic on how to identify the unidentified. You see, that is the theme of the session, and he will add his scholarly knowledge. He will share it to us. We are grateful to have you with us, Dr. Sir, and we look forward to learn from you. Thank you.
Thank you very much.
Dr. Tobias, we are shortly going to play your video.
From you. Thank you.
Thank you very much. Hello, and thank you very much for the generous invitation to come and present at this online conference for the Anatomical Society
Dr.
of
Tobias,
India.
we are shortly going
I am
to play
Dr.
your video.
Tobias Holton, and I am presenting an introduction to forensic facial reconstruction and the drive to advance the field. Thank you very much. Forensic facial reconstruction refers to the process of building up the face of an individual from their skeletal remains. It currently relies on practitioner artistry to generate the facial image, while drawing on scientific knowledge of the face taken from existing research within the fields of anthropology, osteology, and anatomy. It is used in police intelligence to prompt recognition of unidentified cold cases, and it is a last resort that is performed when no other, more reliable means of identification, such as dental records, DNA, or fingerprints, is available. You will also see it being applied within archaeology to promote public interest in historical or ancient figures. In this context, there tends to be a more flexible artistic license given. Now, this slide demonstrates how facial reconstructions tend to vary between forensic and archaeological cases. In forensics, reconstructions are limited to presenting information that is evident with the biological material available. They therefore tend to avoid suggesting skin tone and eye color. Sometimes hairstyle and color might be presented if sufficient evidence of hair is retained on the deceased individual. Personal effects that they were found with might also be depicted in the presented reconstruction. For example, the blue jersey in the top right forensic image. In archaeological reconstructions, there is otherwise more freedom for interpretation, hence how they tend to be more colorful and suggestive of clothing and hairstyles that are not necessarily directly evidenced with that individual, but might instead show a general trend in fashion for people of that time and culture in which the reconstruction comes from. This slide essentially emphasizes why we take care with the superficial details we apply to a facial reconstruction within forensic contexts. The skull alone cannot infer details such as color of the skin, eyes or hair, nor hairstyle. It offers no reference to ear shape, age-related skin changes, nor common facial expressions someone might have had. There obviously would be no indication of personality from the skull. We won't know if they had any superficial body modifications, and we won't be able to tell their body weight. The two photo portraits on screen show how much a face can be altered in appearance just by the superficial details applied. Both those photographs are taken of the exact same person, but the change in hairstyle, fashion, and application of glasses can make him appear like two completely different people. This would be very problematic in a human identification scenario, so we take a lot of care in the superficial details we apply to a reconstruction. Now, the science utilized in forensic facial reconstruction can also be applied to other practices. It is relevant in craniofacial superimposition, where we compare the morphology of an unidentified skull with antemortem missing person facial photographs to determine if there is a possible match for further investigation. This helps a great deal in mass grave scenarios, where there is a lot of unidentified and missing person cases to process. A similar understanding of the face is furthermore important in medical practice as well. An application of morphological patterns in facial features and their relationship with the skull is relevant for surgeons involved in cosmetic or reconstructive surgery. It can also be relevant to the field of maxillofacial prosthetics. Though I will be essentially discussing the practice of forensic facial reconstruction, it is good to be aware of the broader applications the science relevant to this field has. In terms of the modes and methods of reconstruction, facial reconstructions can be performed and presented using both 2D and 3D modalities. 2D methods utilize either traditional sketch approaches using tracing film and paper, or image editing software like Adobe Photoshop, using a photograph taken of the skull for reconstruction. In this instance, it's essential that the photograph taken is in focus and free from perspective distortion. Typically, a subject to camera distance of 2 meters and above would help avoid this. The benefit with 2D reconstruction is that it is a quick and affordable means, but it does not offer the same level of shape and dimension information as with 3D approaches. 3D reconstructions can otherwise be traditionally modeled by hand over a skull cast or 3D print of a skull using clay or modeling wax. More recently, preference has otherwise been assigned to 3D digital modeling approaches using software like Geomagic Freeform. 3D reconstructions tend to be more reliable than 2D because we have a better spatial awareness of depth and form when undertaking the reconstruction. Computerized 3D reconstructions also have an added advantage against traditional sculpted methods that use clay or modeling wax, because we can readily adjust the opacity of the skin and muscle layers to continually check how well the features are aligning with the skull. With the traditional manual approach, you need to cut into the model to expose the bone, which then requires remodeling of the modeling material to restore the face. The only downside to performing a reconstruction in 3D in general, whether it be manual or computerized, is that it is much more time-consuming and costly than 2D. When commencing a forensic facial reconstruction, the first thing we try to establish from the skeletal remains is the basic biological profile of the individual, essentially their sex, ancestry, and age at death details. This will inform some of the scientific standards we use in order to generate the face. Now, even though we can use the skull to determine sex with some degree of accuracy, it is often best determined from the pelvis. The pelvis is obviously one of the most sexually dimorphic regions of the skeleton. The overall shape of the male pelvis is narrow and steep, and the female pelvis is generally broader, with a larger pelvic outlet to facilitate pregnancy and childbirth. An approximate age range can only be offered for age at death, especially with adult cases. In juveniles, a more exacting age determination could, however, be offered due to a more frequent and standardized pattern in musculoskeletal changes occurring during early growth and development. More characteristic details we would otherwise refer to for facial reconstruction include any signs of facial pathology or trauma that is evident on the bone, which might impact how the soft tissue presents. We also refer to various bony details across the skull, which indicate how the soft tissue facial details would appear. With regard to how the face is estimated from the skull, there have been a number of morphoscopic qualitative observations and morphometric quantitative approaches indicated by scientists over the years. Modes of data collection to develop these guidelines have comprised of anatomical dissection, which has offered a useful resource for understanding anatomical structures to gain an idea of how muscles and ligaments attach and establish facial form across the craniofacial complex. This includes understanding the structural foundation of facial features and how they relate to the bone. There are, however, evident limitations when working with cadaver material. Anatomical research is essentially performed on embalmed cadavers, where the soft tissues are invariably altered from their natural state during life. Common embalming solutions such as formaldehyde and ethanol dehydrates and hardens the soft tissues, whereas the likes of teal overtly softens them. Even if accessing fresh, non-embalmed cadavers, taphonomic changes are instantaneous following death. Therefore, signs of muscle slackening, eyelid tautening, and other effects of early decomposition will be evident. The uses of palpation or the taking of direct measurements have been applied. This can be a useful approach for relating the soft tissue surfaces to bony features, but you may not be reliably locating the actual position of a palpated feature. This issue has given rise to the fact that you are unable to simultaneously visualize the bone and soft tissues using this method. Then the introduction of clinical imaging has otherwise enabled much more flexibility and versatility, enabling scientists to visualize deep and superficial tissue structures while accessing living data subjects. Clinical imaging modalities include X-rays, MRI, CT, cone beam CT, and ultrasound, and they all come with their various advantages and disadvantages. In terms of how the general face surrounding the facial features is built, there are three methods frequently cited in existing facial reconstruction literature. The Russian method is one of the earliest established techniques founded by Soviet archaeologist and anthropologist Mikhail Gerasimov. In this approach, anatomical insight is used to sculpt the larger muscles of the superficial face, which are specifically the temporalis and masseter muscles. Average tissue depth data is otherwise used to inform you of the thickness of tissues across the craniofacial complex. Then there is the American method, which is considered to solely rely on average tissue depth data. Facial muscles are not modeled in this technique, but an understanding in facial anatomy is still considered crucial. This is because the bone typology can change dramatically between tissue depth landmarks. Thus, some knowledge of face anatomy must be used if tissue depth markers are to be connected in ways that represent realistic face contours. Then lastly, there is the Manchester method, which alternatively models all the superficial muscles involved in facial expression and mastication, and also utilizes facial soft tissue thickness reference markers to guide skin application. Now, all three methods have proven to be effective in forensic practices, but the Manchester method is believed to be the most comprehensive approach. This is because it proactively takes into account how muscle structures attach or fit over an individual's skull, rather than heavily depend on average tissue depth data. When it comes to considering facial soft tissue thickness, this refers to the markers that project from cranial landmarks to facial landmarks. The length of these lines corresponds to the average measured thickness of the soft tissue at that particular location. We typically rely on average data banks, which are often matched to the demographic of the individual being reconstructed. Most published data banks are organized according to population, sex, age, and sometimes even body mass index. These collections are also differentiated according to the data collection modalities used, which can range from needle puncture studies using cadavers to the application of medical imaging systems such as CT or ultrasound on living persons. There have been research developments emphasizing the use of tallied total weighted means that pool all existing facial soft tissue thickness data ever published together, regardless of population, sex, or even method of measurement used. This came about following the argument that when all published facial soft tissue thickness data is collected together and analyzed, the results identify no clear secular trends at frequently investigated landmarks. A wide variation in soft tissue depth measures between different measurement techniques, irrespective of whether living persons or cadavers were considered. Also, no clear clustering of non-Caucasoid data far from the Caucasoid means, and minor differences between males and females. Therefore, by substantially increasing the sample sizes using weighted means and standard deviations, it was seen to help cancel out random and opposing study-specific errors. Regardless of these promises, most facial reconstruction practitioners refrain from using this data with more demand for its validation needed. As an alternative, there have been a couple of regression studies looking into subject-specific approaches, but they have not proven to perform significantly better than the available mean datasets. Therefore, it is quicker and easier to maintain the application of mean data. With regard to how facial feature details are estimated, particular care needs to be taken with regard to which reconstruction guidelines are implemented. A lot of the early science dating back to the 20th century lacked the scientific rigor expected of modern research today. Some have been based on informal observations, which have not undergone any empirical tests that have been published, so their accuracy has for a long time been unknown. This arguably gave rise to numerous conflicting guidelines, such as that witnessed for mouth width estimation, where all of the proposed approaches on the slide, dating back to 1957, 1962, and 1986, were all published without robust testing to substantiate their use. Much later testing by Stefan and Wilkinson et al. Has, however, suggested that given these three options, mouth width is most reliably estimated as being between the medial borders of the iris. One could, however, argue that this guide remains a problem when dealing with skeletonized remains, where mouth width is now highly dependent on the accuracy of eyeball placement and prediction of the iris parameters. Many facial reconstruction practitioners thus prefer to use Krogman and Ishkan's method because it eliminates that risk in error and has been considered as the next best available option. So, as implied by the previous slide, developments in testing the validity of existing facial reconstruction guidelines have been started, predominantly since the noughties. And existing validation tests have highlighted significant errors in some of the original guidelines, and consequently prompted the development of new ones. Original nose prediction guidelines experienced this, and more successful regression equations have since been introduced, a development that was likely made possible with the introduction of enhanced clinical imaging modalities and a better appreciation for rigorous statistical analyses. This is a scientific trend that will help to continually enhance the facial reconstruction practice as time goes on. Evident limitations in past guidelines relate to issues with sample size, mode of data collection, and the deficient practical application the proposed guide might have in facial reconstruction, has also prompted more comprehensive standards. A good example would be how the method for estimating eyeball position in the bony orbit of the skull has evolved. For a long time, early cadaveric studies by Whitnell was referred to, which very specifically determined that the outer circumference of the globe of the eye is positioned approximately 4 millimeters from the orbital roof, 6.8 millimeters from the orbital floor, 4.5 millimeters from the lateral wall, and 6.5 millimeters from the medial wall. A later cadaveric study by Stefan et al. Approximately agreed with Whitnell's findings, but identified that there was more of a lateral bias in the positioning of the eyeballs. Stefan et al. Furthermore identified a problem with the proposed approach in approximating eyeball position, where fixed measurements are indicated between the outer circumference of the eyeball and the orbit, in that not all orbits are obviously the same size. To overcome this, they identified that the eyeball had a 1.4 millimeter superior and 2.3 millimeter lateral median divergence from the orbital center. Though these standards helped in the development of understanding eyeball position, the studies by Whitnell and Stefan et al. Depended on cadaveric and embalmed data samples, which could present some distortion from what would be witnessed accessing a living sample. The represented sample sizes were also very limited. Guillaume et al. Has since performed the most recent and comprehensive investigation into eyeball placement, accessing a large database of 375 CT scans of living persons. This by far delivers the best study for predicting eyeball placement to date. They identified regression formulae and presented simplified proportional predictive models using maximum orbital height and maximum orbital breadth measurements taken from the skull to estimate the distance of the center of the eye from the superior and medial orbital margins of the skull. They report the R-squared, or coefficient of determination value, which indicates the predictive accuracy of the statistical models, which are 0.49 or 0.42. This is unfortunately considered statistically weak, and is not an uncommon problem across many reconstruction guidelines, which is probably why the author suggested a simplified proportional solution for estimating eyeball placement. The reported standard error of the estimate, which is abbreviated to SEE on screen, however, demonstrates a standard error of just over 1 millimeter, which at least is not too bad. Another concern with many of the existing standards developed to support facial reconstruction is that they often only offer a linear guide. Knowledge of how hard and soft tissue features spatially relate is therefore limited. With a team in South Africa, we have been looking to establish a better sense of how the morphology of the mouth relates to the underlying dentition of the skull, applying geometric morphometric techniques. Geometric morphometrics is grounded on a statistical theory of shape that captures considerable spatial information encoded in landmark coordinates, and consequently offered a good solution for investigating dentition to lip mouth morphology. We were also interested in developing more standards that better represented the South African population, because there was no existing data available for especially Black Africans for approximating mouth morphology, which was presenting considerable concern for reconstruction specialists in the South African Police Service. Accessing cone beam CT scans, where we benefit from viewing faces that are orientated in an upright rather than supine position, a sample of 109 Black South Africans and only a small white South African sample of 39 individuals were accessed. These samples comprised of people aged between 20 and 75 years. The mean age of the total sample was approximately 36 years, with a standard deviation of 11 years. The sample otherwise comprised 86 males and 61 females. Viewing the scans using clinical imaging software as IRIX, the scans were fixed in the Frankfurt position, and anterior profile stills were captured of the face and underlying skull. ImageJ was subsequently used to digitize 12 dental landmarks and 8 soft tissue landmarks. The patterns in hard and soft tissue landmark relationships were subsequently assessed using MorphoJ. The results identified were as follows. A pooled within-group regression analysis, which investigated the sample according to independent population and sex variables, was used to assess age. This identified that age presented a significant impact on mouth shape, accounting for about 14% of the within-group variance observed. An increase in age was affiliated with a decrease in lip and teeth height, while mouth width was seen to increase. The oral fissure and Chilean placement was otherwise seen to enter a more inferior placement with relation to the teeth. When sex and population groups were compared, the influence of age was not seen to significantly differ between them. But it is noted that the sample would have benefited from a greater number of white South Africans and a greater number of older-aged individuals, in order to gain a more substantial understanding of the influence of age. After accounting for age, principal component analysis was performed. Common patterns in morphological variation are expressed in principal components 1 to 4, which accounted for 84% of the total variance. Statistically significant population differences were visibly presented in principal components 1. This was confirmed in two-way ANOVA tests, which was furthermore identified along with significant sex differences. General trends in dentition to lip morphology is illustrated in the given wireframe diagrams. Consistent with existing literature, the white South African sample demonstrated a soft tissue mouth that sits within the margins of the teeth, closely aligning with the cemento-enamel junctions. The oral fissure being located approximately 1/4 the maxillary incisor crown height superior to the dental occlusal line. In contrast, Black South Africans presented with thicker lips in relation to the teeth, with the oral fissure closely bisecting the dental occlusal line. A downturned oral fissure was common in white individuals, whereas a more horizontal alignment was prevalent in Black individuals. Males generally presented larger dimensions than females, but females had a greater lip to teeth height ratio than their male counterparts. And in general, the points of the Cupid's bow typically sat within the margins of the maxillary central lateral incisor junctions. Currently, this study is the first of its kind to graphically demonstrate how hard and soft tissue features of the mouth relate, offering a visual guide that is designed to support existing approximation methods that utilize linear data, which we have already recently published for South Africans. This study continues to be a work in progress. We hope to build on the study with an increased sample size to better represent sex, age, and population variables. We also wish to explore 3D geometric morphometric techniques, because we believe this will offer a more reliable method for landmark allocation and will yield more comprehensive results. In terms of other visual approaches being established to assist with facial reconstruction, I work with another South African team for a master's thesis developed by Smidlen, looking into patterns in facial aging. Previous studies mostly investigated facial aging in European faces, so this research aimed to provide data on patterns of facial aging in an African sample. Data collection comprised anterior and lateral photographs of 189 Black South African males aged 20 years and older. 30 individuals per decade were included, though only 9 individuals could be accessed for those aged 80 years and older. A facial aging scoring system, based on previous research, was constructed for non-metric age changes, such as appearance of wrinkles and sagging around the orbital area. Metric age changes, such as ear lengthening and lip thinning, were investigated separately using 3D photogrammetric surface scans taken of participant faces. The results indicated that Black South Africans tended to show delayed crease formation compared to Europeans. Also, features such as nasal elongation, nasal tip drooping, ear width increase and ear length increase did not show a clear change with increasing age. To visually aid forensic facial reconstruction practices, where the suggestion of age is often required, an average face per a decade model was generated by morphing all faces within that decade to produce a visual mean, using Abrasoft Phantomorph software. This has proven to offer a very useful and more intuitive visual guide to assist reconstruction specialists within facial reconstruction. Besides research that deliver improved standards to support the manual and more artistic process of reconstructing faces, there have been some very interesting alternative approaches explored to generate facial images for the recognition of unidentified persons. There have been several attempts at automating reconstruction. A recent example is research by Geitzen et al. They attempted to generate automated reconstructions using an amalgamation of mean facial tissue depth data and principal component analysis, using reference model data derived from CT collections. This offers an interesting alternative to the manual method of facial reconstruction, but has experienced considerable problems when it comes to building a substantially sized CT reference sample to offer more robust results. Another very interesting approach worth mentioning is the development of DNA composites using DNA phenotyping. This has advanced rapidly with the application of machine learning approaches, but the extent of its current capabilities is hotly debated. Akin to voice concerns with automated facial reconstruction, scientists and artists alike have argued that the face can appear very generic and are not always good at characterizing the individual's face. There will also be a number of population groups who are not yet adequately represented, if at all, in existing DNA research. This might, however, change given time and continued investigation. In terms of the reported accuracy of facial reconstruction in general, not referring to the automated or DNA composite methods, but the original methods of reconstruction requiring a forensic artist. The first accuracy assessments ever made were initially based on forensic case success reports. Facial reconstructions that had been successfully identified were compared against existing ante-mortem photos of that individual. This approach was, of course, heavily biased towards successful cases and would not necessarily help in identifying where a practitioner might be going wrong for reconstructions that were not being successfully recognized. Blind studies, where a forensic artist approximates the face of a skull that has an existing ante-mortem facial image available for comparison following reconstruction, has since been used to better assess how reconstructions perform. As with the example on screen, some studies test accuracy by presenting the reconstruction along with an array of ante-mortem facial images known as a face pool, to examiners that then attempt to match the reconstruction to the actual target face from the pool. This method does, however, pose one problem. It is heavily dependent on unfamiliar face recognition, which is notably much weaker than when people are expected to match familiar faces. A more recent method of blind study testing that is perceived to be more favorable is one that matches the 3D facial reconstruction directly with a scan of the actual face, either from clinical CT data or a 3D surface scan. Reverse engineering software can subsequently calculate how well the reconstruction and target face match and generate a surface map illustrating the range and error witnessed. Using this method, tested reconstructions have ranged from a 52% to 90% surface correlation with the actual target face, allowing a minimal 2.5 plus minus millimeter difference. It's not too bad, but could ideally be better. With developments in the scientific standards used to generate the reconstructed face, accuracy results will hopefully improve as time moves on. There has also been a growing emphasis in performing validation tests that quantify the accuracy of independent facial reconstruction standards that are used to reconstruct several aspects of the face by comparing estimated soft tissue dimensions used for reconstruction with actual soft tissue dimensions. This is typically conducted using clinical images where both hard tissue and soft tissue features can be visualized independently and together. This approach does not require an actual reconstruction to be performed, which saves a considerable amount of time. Now, generally speaking, all tests performed have demonstrated that facial reconstruction does hold a feasible degree of reliability. But of course, there is room for further developments in the science. Another important factor that we also need to consider are the secondary influences that could also impact recognition with facial reconstructions in a forensic environment. This includes considering what level of exposure or media coverage they are being given. Do family and friends of missing people know how to go about running a missing persons inquiry? And do they feel safe or comfortable about doing this? Is the individual from another country? Have they been missing for a long time? Are family and friends aware that this person is even missing or dead, depending on what their social habits during life were like? Evidence of personal effects, such as items of clothing and jewelry, might also trigger a recognition more so than the actual facial reconstruction. All these factors can affect the rate at which reconstructions are recognized and identified. In terms of general research developments needed across the field, we need to build on sample sizes represented in studies in order to ensure inclusive reconstruction standards are being established that are not being influenced by statistical noise due to limited data. This is often easier said than done because sourcing appropriate data can be a time-consuming and challenging process. But there needs to be a better representation of different ages and populations. More consideration is needed regarding the influence of body mass and also pathology. All the guides we use in facial reconstruction have been developed using healthy data samples, but we have no direct reference for those affected by different medical conditions, nor even an idea of how to reconstruct faces of edentulous persons, referring to those who have lost all their teeth. With the development in clinical imaging, we can improve and develop on the existing data we have using more reliable modes of data collection. It would be ideal for all reconstruction standards to be based on living persons. Due to gravitational influences that act on the face, it would be preferable to utilize medical imaging modalities that has the participant orientated in an upright rather than supine position, because depending on body weight and age, this can present a considerable dorsal to ventral displacement of tissues. Statistical analysis needs to continue developing in its rigor. Modern science now demands considerable attention to repeatability and validation testing. Publications of new standards need to explicitly state how reliable the technique is, reporting key values such as the correlation coefficient and coefficients of determination, so we know how strong statistical models are when delivered. Many of the existing guidelines are linear in their approach. With developments in the likes of geometric morphometrics, we need to take the science more into the third dimension to comprehend in more detail how soft tissue details of the face relate to the skull. This includes establishing more reliable references to how we determine shape and projection details of the face. Currently, details such as lip form and ear projection are highly presumptive. The field of facial reconstruction is one that will benefit greatly from growing interest with anthropologists and anatomists alike. This screen now delivers all the various references utilized within my presentation, of which you are welcome to receive a copy of. Thank you very much for your attention today, and I look forward to hearing any questions that you might have.
Thank you, Dr. Tobias Holton, for giving such a beautiful insight on facial reconstruction.
Pleasure.
I invite all the listeners to ask if they have any query regarding this topic.
Yeah, I think before we go to the questions from the audience, I would like to take this opportunity to thank the, I'm Dr. Sharif, who has been chairing this session. I would like to thank the organizers of this 68th NATCON and the team, and you are doing an excellent job, and thank you very much for giving me this opportunity to chair this excellent session by Dr. Hulton. Now coming to the presentation, I think Dr. Hulton congratulations. It has been a great presentation, and I can easily say that probably this may be the highlight of this conference. So congratulations, excellent job, very well done, very exhaustive, very informative, and very stimulating for all the anatomists that have been listening to this talk. Thank you so much. I was wondering, and in fact, after reading some of the literature about the facial recognition, it was more in the realm of a combination of science and art. And there was more of art and there was less of science. And that was the reason why it was not as reliable as we would like it to be. Now, I think your talk has given the direction to make it a bit more scientific and less of an art. And I think this is a very good opportunity for all the anatomists, particularly in India, because we have all the variation possible probably across the world in India itself. If you travel from north of India to the south of India, you might find all kinds of cases. You know, from east to west and north to south, probably India is the place where you find all the faces of humanity here. And this is a very good region, and the anatomists who are spread across the length and breadth of India can contribute to the data sets that Dr. Hulton was referring to. The data sets from the living faces using the modern imaging methodologies, particularly in upright position, I think. Yeah. Okay. I think I am very excited about about this whole discussion and the talk, and I'm sure it stimulates a lot of research in this area among the anatomists and the budding anatomists, and I'm sure a lot of thesis, MD thesis are going to come up on these lines. Thanks, Dr. Hulton, for stimulating this. With this, again, initially I had a question in mind which has been adequately answered. However, I just want to say that question. I just wanted to ask Dr. Hulton about his thoughts of how to bring it more scientific and less artistic. I think his talk has adequately answered that, and I don't expect anything more from this. Thank you so much. Again, I thank all the organizers and Dr. Hulton. It has been a pleasure listening to your talk. Thank you.
Pleasure. Thank you very much. It's been an honor. Thank you.
Can I make a point, please? Hello. Can I add a point to what Dr. Sharif mentioned? Am I audible?
Yeah.
Yes, you're audible.
Yeah. See, there is one more thing. See, this is a fantastic introduction to a new battery of research or a new line of research. Interesting because looks like almost all the equipments and the devices he mentions are reasonably, easily available in our country.
Yes.
He brought in the importance of the local prevalence, the racial, the ethnic diversities. All these are relevant to us. It probably opens up a Pandora's box of curiosity and knowledge that we could build huge data sets, as Dr. Sharif said. Second, most of the equipments he is talking about, which adds excellent weightage, value add to the understanding of the anthropometry, not only in terms of bone, but also soft tissue. This level of virtualization that is coming up in the field of anthropometry to add value to the actual process of identification of the individual. This raises tremendous scope for younger generation to take this up on a more rigorous and a more energetic basis. So it was a great learning experience, and at least as the sessions are progressing, we seem to have got the impression, Dr. Tobias, that here is one methodology which we can develop on the lines that is suited for our local ethnic and local population requirements. Thank you for this enlightenment. Over to the audience.
Okay, I invite anybody to ask any query from Dr. Tobias.
Any questions, please?
Well, sir, I have a question. Dr. Tobias, sir, that how much 3D printer is applicable in facial reconstruction? Nowadays, we are heavily relying on 3D printer in cases of maxillary injury or facial fractures or maxillary fractures to reconstruct the face. How much of 3D printer is helpful in facial reconstruction?
In terms of 3D printing, it depends very much on the mode in which facial reconstructions are being performed. So certainly if people are using the traditional modeled approach where they're using clay or modeling wax, then that's when 3D printing would be applied. Mostly in terms of the work I do, we keep strictly with digital reconstruction on the computer itself. So we tend to be working directly with superficial surface scan data of skulls that are being collected with various scanning modalities. And certainly also CT scanning as well has been performed to capture a good, high detailed, high definition capture of the skull in order to perform the reconstruction itself. So certainly in terms of 3D printing, if that is to be performed, as long as you've got a good reference to the actual skull available, that is very practical to aid with facial reconstruction. So you can refer directly to the details of the bone always, because of course with 3D printing, there will always be a degree of detail that would be lost depending on the quality of the 3D printer. But of course, the main thing is that you should never perform the reconstruction directly on the skull. So that's where the relevance of 3D printing skeletal information in order to conduct a reconstruction, if doing it the manual way with clay, is best. I hope that helps answer your question.
Any other question, please?
There is an interesting question. I think Dr. Kaveri, are going to read that question which has just appeared.
So
CT scan images or 3D photography? Which one is more reliable to collect data on facial parameters? I think that's an interesting question. Dr. Holton.
You don't mind repeating that question again? Oh wait, I think I see it. I see it now.
Yeah.
So 3D photography, I'm guessing photogrammetry, CT scan images. So there has been studies looking into the sort of error rates in which skeletal data is captured using CT, and also studies looking into the success of 3D scanning, whether it be laser scans or photogrammetry. Now, I need to actually look at the data together to just see how they compare. But certainly, I think the accuracy of CT scanning directly with bone, where you haven't got soft tissue, isn't as reliable as when you're scanning a full body, where you have the soft tissue there. Saying that, though, the accuracy of the CT scan, when you're just working directly with bone, I wouldn't say is negligible. It will be, you still get a reliable result. And certainly, with photogrammetry, which is what we tend to use, certainly I've tested it myself, performing various craniometric measurements to see how they tally up. And generally, I see nothing to concern about. So certainly using the likes of photogrammetry or laser scanning to capture skeletal information is much more accessible than trying to apply CT scans or any other kind of mode of medical imaging scanning, of course, CT being the best for capturing hard tissue bone. Yes, so that's actually a very interesting question. And I do feel that either methods are appropriate given the circumstances.
There was another question from the audience end. How to take environmental and lifestyle changes in consideration in scientific way? Dr. Tobias.
Thank you. This is very difficult because obviously this is now something that's on a scale. So of course, if someone has been abusing alcohol or drugs, it's variable and how much will impact that individual. There's also genetic influences that will take a part in this. So I do believe there has been studies conducted looking into obesity, for instance, or malnutrition. And they've sort of implemented a program where you can input a face and then generate a scale to demonstrate what the individual looks like if they were underweight or overweight. And I believe that they gathered the data using medical information that was available on participants, and they generated this program basically referring to body mass index and where individuals sort of fit into given body mass index values. So that's how they generated a scale. But again, it's extremely variable, and how to determine, what lifestyle habits an individual had directly from bone can be tricky and not always determinable, unless, of course, there's been, say, extensive drug abuse or something very dramatic and detrimental occurring on the bone. So this is always something that's going to be very difficult, and maybe with the developments of computer learning and engineering, we can certainly sort of input a reconstruction that's kind of demonstrating an average of how this person would typically look given ideal circumstances, and then introduce a scale to show sort of the variabilities that can present in the soft tissue, the sort of the extremes between underweight, overweight, impact of disease. But of course, this would take a considerable amount of time and data collection in order to generate valuable results.
Thank you. Thank you, sir. And so it was a wonderful presentation, very informative. And now I would like to thank everyone for being with us. And now on the behalf of Team NATCON, we would like to present the certificate of appreciation to Dr. Tobias, Dr. A. Sharif, Dr. Bala Subramaniam V, and Dr. Anand Mishra. Thank you, everyone. Once again, thanks for your support.
Thank you.
Thank you.
Dear audience, there is a lunch break of half an hour, and we'll be joining you all very soon at 2 p.m. Thank you. Thank you for your patience.
Thank you.
Thank you. Thank you.
Thank you.
Thank you for being with us.
You're most welcome.
Thank you very much.
अनम्यूट आ गया। अनम्यूट तो हो गया, अनम्यूट तो हो गया ये, लेकिन वीडियो नहीं। हाँ हेलो, तो क्या कहते हैं, अनम्यूट तो हो गया, लेकिन हाँ स्टार्ट माय वीडियो, वीडियो नहीं स्टार्ट हो रहा है सम रीजन। इससे क्या दिक्कत आएगी? आई कैन सी विजेंद्र, आई कैन सी प्रोफेसर अनिल अग्रवाल।
Welcome back. Welcome back, everyone. Very good afternoon. So moving further with this journey, which is full of great knowledge and tremendous experiences, let's begin our next session. The title for the same is Identification from Prints, a Forensic Approach, and will be delivered by Professor Anil Agrawal. The moderator for the session is Dr. Lakshmi Rajgopal, General Surgeon and Anatomist, retired Professor of Anatomy from GS Medical College. Thank you, ma'am, for being the part of this event. And I request you to kindly invite and introduce the chairpersons of this session. Over to you, ma'am.
Okay. Yeah, very good afternoon to all of you. It's my, it's a proud privilege to introduce the chairpersons of today's session. Dr. Ashok Sahai, sir, is a senior most anatomist, one of the senior most anatomists of India. Dr. Kaveri, the screen is not showing the biodata of Dr. Sahai. Can you please project the biodata of Dr. Please share the screen.
Yes, ma'am. We are sharing it. Now, is it visible to you, ma'am?
Yeah, yes. Okay. As I said, Dr. Ashok Sahai is one of the senior most anatomists of India, former professor and head of Department of Anatomy of King George's Medical University, Lucknow. He is currently honorary professor of anatomy at Dayalbagh Educational Institute, Faculty of Integrated Medicine at Dayalbagh, Agra. He has edited and reviewed 10 chapters of 40th edition of Gray's Anatomy, and he's also published many prospectus for various medical colleges. Next slide, please. Sir is former advisor of Federative International. Hello.
Yes, ma'am, you're audible.
Yeah. He's a former advisor of Federative International Program of Anatomical Terminology of IFAA, and is also a member of South Asian Advisory Board of Walters Clovers as well as Lippincott Williams Wilkins. And he has been a life member of Anatomical Society of India and served the society as treasurer and vice president. He has won many honors, of which the foremost is he has been the first anatomist in India to be portrayed by the Anatomy Constellation as discovery of special anatomists from India. He has also won awards like Legend of Anatomy, Dr. APJ Abdul Kalam Award, Rashtriya Gaurav Award, and Saraswati Samman. So we welcome you to chair this session. I request you to show. Yeah, thank you, sir. The next chairperson is Dr. Brijendra Singh, who is a professor and head of anatomy at AIMS Rishikesh, India, and is also the Dean Social Outreach at the same institute. And he is the current president of Anatomical Society of India and is vice president of Anatomical Society of India of UP chapter, and also vice president of NMO Uttarakhand state. He has served Anatomical Society of India actively in various capacities since 2002. He has served as chief editor, executive editor, and joint editor in several national and international journals. Next slide, please. Yeah, Dr. Brijendra Singh has also published 11 books and contributed a chapter in a book. He has won many awards like Best Teacher Awards in the year 2017 and 21 from AIMS Rishikesh. He's been awarded with Seva Ratna, Him Ratan Award, Delhi Ratan Award, and Gem of India Award, and also Atal Sikitsak Shikar Samman, and many more. We welcome you, sir, for this session. Now, I request Dr. Brijendra Singh to introduce the speaker of the session, Dr. Anil Agarwal.
Thank you, ma'am. It's my privilege to introduce Professor Anil Agarwal, sir, who is a very well-known, reputed figure from medical sciences from India. As such, he has been head of the department from forensic medicine, Maulana Azad Medical College, New Delhi. Professor Anil Agarwal, sir, is a renowned figure, and he served UCMS Delhi, and to his credit are more than 70 research articles from national as well as international journals. His research are so important that these researches has been cited in more than 500 studies. His proposed new classification about necrophilia, and one of the leading authority of, he is one of the leading authority of necrophilia in his subject of the forensic medicine and anatomy. Dr. Anil Agarwal's Internet Journal of Forensic Medicine and Toxicology is most widely read and popular peer-reviewed forensic medicine journal. It includes forensic sciences and allied subjects like criminology, police science, deviant behavior. He is also having a prestigious academic advisor to University of Edinburgh position. This is our kind privilege, as well as we are having a great opportunity to listen and to witness knowledge and his views on this subject. We welcome you, sir, on the behalf of Anatomical Society of India for all the learners. Thank you very much, sir. Welcome.
Thank you, Dr. Brijendra. And Ritesh, have you started the slides? Ritesh, are you there?
Yes, sir. Yes, sir.
Very shortly,
No, because Ritesh said he will start my slides. So I was just.
Yes, sir, he's going to start. He's going to begin your presentations.
All right, great. Thank you. Now, thank you, Dr. Brajendra, for such a nice introduction. I just don't know from where you took all this material because I didn't give anything to you. But anyway, it's a huge, huge honor to give this talk on prints, and I was quite overawed by the biography of our respected chairpersons. So I hope nothing goes wrong in my presentation. Now I welcome you all to my presentation. Now today we are going to talk something about identification from prints. Now we chose this subject because I thought it would go well with your theme of anatomy, because there are certain areas in anatomy where we use, these areas we use for identification and for several other purposes. May we have the first slide, please? Ritesh? Okay, let me start myself.
Sir, we are at a bit of time. Please allow me a minute. I'm just starting.
Just 1 minute. Are we okay now?
Please continue, sir.
All right. So this is our topic, identification from prints, and I tried to make it very worthwhile for anatomists, because I know anatomists are there and not forensic people. So basically, my idea was to all right. So the identification, I'll just very briefly give you some background of identification, because otherwise you might not be able to appreciate these prints that I'm going to talk about. Now, identification, as you probably know, is a problem with us. We, several places, we have to put our thumb to gain entry inside a particular place, or sometimes we have to do some other things, passwords are there. And it is a issue both in, living as well as in the dead. I'll just tell you some historical example. This, actually, I keep some news cuttings. This is a very interesting cutting from Times of India, which came, I think, 2 or 3 years back. I was very intrigued about it and just saved it. I thought I should show it to you. You see, this is a very interesting case where a man spent 10 years, simply because he was misidentified. And he kept on saying that, I have not done this crime, and yet nobody believed him. And for almost 10 years, he spent in prison only because of misidentification. And you see here, you see that cops take 9 years to verify his fingerprints. This is a real story, and it would give you a very interesting, insight into how important this identification can be. Another, quickly I'll go through these newspaper stories. They will tell you that they are very important in our daily life. This is from Hindu of 2015, where there is a woman who is actually alive, but she's dead on paper. So she has stopped receiving all pensions. She's no more having a health insurance. And she's going around everywhere telling that I'm alive, but nobody's able to believe him. She's right in front of them, but nobody's believing her. And so she's dead on papers, although she's alive. So this is another classical case of, wrong identification. And then again, a very interesting thing, most cases which we do are where, a person fakes his own death by killing somebody else who is very similar in appearance to him, or similar in height, similar in physique. He finds a person, then kills him, distorts his face completely so that police cannot know who this person is. And then his wife, who is an accomplice, she goes to the life insurance and says, look here, my husband has died and you give me the money. But here this man has faked death because he doesn't want to pay alimony to his wife with whom he has got divorced. So he actually killed somebody so that he could, people would think that he is actually died and he doesn't have to pay alimony. So these are the very strange cases where people try to snatch identity of somebody else. And these misidentification cases can actually be solved by several of our anatomical, techniques. Now, I'll just tell you one or two historical. This is a very, very famous historical case, although I'm just telling you one. But history is replete with such cases where misidentification occurred. This is, Raja Ramendra Roy, who was the king of Dhawal state, which is in current Bangladesh. And he died in 1909 at a very young age, around 25 or 26. And then supposedly he was cremated. Now, people who attended the cremation, half of the people were saying that he was cremated, but half said that as the funeral pyre got started, there was a sudden rain and everybody ran away, leaving the corpse as such. So nobody knows what actually happened. But after around 15 years in 1921, this sannyasi, which is known as a Dhawal sannyasi, he comes up and he claims that he's Ramendra Roy. Now, at that time, there was no good method to differentiate and to confirm whether he was really Raja Ramendra Roy or not. So they compared things like his tooth structure or some moles, some scars, some broken tooth. 21, 22 characters like this were matched, and they all matched. So it actually appeared that he's the same person. But, and actually this case went into court also. But by the time the court gave the decision in 1946, he was already dead. Now cases like this are very, very much there. This is I have taken one from mythology, where even just to say that even lords have problem in identification. There is Lord Rama who is seeing the, Lord Rama who is seeing the fight of Bali and Sugriva, and he, in the first fight, he couldn't differentiate between the two. So he requested Sugriva to have another fight with him, and this time he actually has a garland around him so that he can identify it. So, just to show that this problem is very much there from the very beginning. Now, there are some more instances from the Bible, but I'll not go much into it. But this blind person, who is the Isaac, he had two sons, one with the goat skin and another without the goat skin. So this person actually wears goat skin, and he, poses as if he's the other brother whom his father loved more. So, and then there are more. And see what are the wrong things that can happen when there's a misidentification. One very common, which probably is common to all of us, is that you can have an operation on a wrong patient. Now, I have had several cases. I'm personally aware of several cases where two persons of the same name were admitted in a hospital for some kind of operation, and doctors, by mistake, they conducted an operation on the wrong person. That's again actually a misidentification case. And then identification of the wrong culprit. We have already seen that, you see this, if you would misidentify a culprit, you can actually imprison a wrong man and post-mortem on a wrong body. Now, we as forensic people, or those of you who are in forensics right now, must be aware of at least one or two cases, maybe in their hospitals or maybe in their colleagues' hospitals, where operation on a wrong body has occurred. I myself am involved in some cases where In one case, we had 3 bodies, 3 female bodies, completely burnt, and they all were of the same age and the same name. And misidentification occurred, and it was a very horrible mistake. I will not go into the details of this case because one of them, you see, girls was of a different religion, so her body was buried, and it was not her body. Then it was exhumed, given over to somebody else. A very, very confusing case. Now, to avoid, you see, wrong operations, we all know that we put a band. Identification parades are there where they identify. Here I had a small movie, but I'll skip it up just to save some time. Just this is an identification parade from a movie. I had a and where actually a witness comes and identifies the right person. Now here the problem is that our identification has become very much witness based. This is a witness based identification. This is the way she identifies the man from the lineup, and this is witness based. But we don't want a witness based system, because witnesses may speak a lie, or they may genuinely be misconceived. So we want to have a very good forensic system. Now, some actual cases I will show you where I've been involved. This is, you can see me conducting postmortem here. This is another senior doctor. And this is a body which we received, which is completely burnt. And it was found inside a car, actually. The car was also burnt. The story was that this belongs to a medical student who was in love with a girl, whose that girl's family was not in favor of this marriage. So the allegation was that they had burnt him. So we had a very great difficulty in identifying him because he was completely burnt. But by applying some techniques, we could identify this man, as I will tell you later. And this just another case, completely burnt, in which some techniques, which I'm going to tell you, can help. Then there are some historical methods of identification. This is a very old Barthillon method, where they used to take, you see, measurements of all people. So they would take the height, they would take the reach, they will take all kinds of you see measurements. Around 11 measurements were taken, and then they thought that these 11 measurements are never the same in 2 people. But you see, a good identification method is something which you can find at the scene of crime. For example, fingerprints. Fingerprints you are going to find at the scene of crime so that you can actually apprehend him. There's no way a person is going to leave his 11 measurements at the scene of crime. So that's why this method actually never caught up. And it was replaced completely by fingerprints. Now we are, with this background, let us come to the prints that we are going to discuss. First, we'll talk about ear prints. Ear prints are not very well known, but they are quite similar to, fingerprints. When we touch a surface with our fingers, sometimes you can have a visible print. For instance, I have my hands drenched in blood. Let us say somebody has committed a murder and his hands are drenched in blood. And then he puts his hand on maybe some cloth or some wall or something. Then you can see that print very clearly. This is called a negative print, a positive print, because you have left something. But if let us say it's a painted door, and on that you put your hand, and then you take it away, then it's a negative print because you take away something. But these are visible prints. 99% of the times, the prints are latent. For example, let us say this is the tabletop, and I put my hand here, and I remove it. So you don't see anything. This is a latent print. But what actually happens is that some oils from my ridges have actually got deposited there. So if I just put some talcum powder there and blow it away with some air, then this talcum powder will keep sticking to those ridges where the oil was there, and then it can become prominent. This is known as developing a latent print. So in a similar way, actually ear prints can be developed, as I can show you. I'll show you. Then lip prints, again, very less known thing, but we'll talk about it. Lip prints will come into play when somebody, for example, drinks from a glass or brings his lips in contact with some other thing in whatever way, maybe in some attack or something. Then nose prints, again, something which is not very well known. Palate prints and tongue prints, and finally the tooth prints. These prints are not very well known, and I thought I must talk about this. Now, about the ear prints. It's also known as earology or automorphology, but if we take earology as such, then it includes more than ear prints. Now, when ear prints will be seen, actually, a common situation is when a burglar puts his ear to the door or window to listen. Now, this is not as uncommon as we might believe, but this sometimes happens. And then during a struggle, when a person is pinned down to the floor, and suppose one side of the head is touching the floor, then the ear print will come on the ground. Then when we have seen when the body is being positioned or moved, somebody has been killed already, and you are positioning it, moving it. Then also, if the ear comes in touch with the floor, then the ear prints will come. Remember, in none of these positions, fingerprints will be seen on the crime scene. As a matter of fact, a burglar may be smart enough to put gloves on his hand, but probably he will not put something over his ears. So this ear prints can come. This is how a developed fingerprint will be appearing. You know, we have developed it by putting a powder over it now, and then we can see it. Now, it might surprise you a little bit as to how from this ear print we can identify a person. For this, I will show you some dimensions. Now, here comes our anatomy, some dimensions of the ear. We do not talk much of ear anatomy normally, but here it becomes very important. Now, this is the ear from an actual subject I have taken, and then I drew some lines so that we can see all the things. There are 8 points here from A to G, and A is where the ear, attaches, the top point where it attaches to the head, and B is the bottom point from where it attaches. Now, this line AB forms the base of the ear. Then C is the Darwin's tubercle. Darwin's name has been associated with this tubercle because Darwin was a very great believer in earology or ear anatomy or identification from ears. So that's why Darwin's name has come into this ear terminology also. So C is this, and D is where the tragus, actually the top portion of the tragus ends. So the line CD, they call it the anatomical length of this. Then E is the topmost part of the ear, and F is the bottommost part of the ear, which is known as the true length or the actual length of the ear. And G is the innermost part, and H is the outermost part. So this is known as the width of the ear. Now these 8 points and these 5 lines, they are supposed to be unique for every person. And the main idea is to look for these points and these lines in those ear prints. Now, you know, with different professions, ears can take different shapes. This is just an additional dimension to it. So once I went to Sports Authority of India and I saw some wrestlers there, and I had a sudden impulse to look at their ears because I know that wrestlers have very peculiar ears. So now if you start touching somebody's ears, I mean, and you don't want to annoy them. So first I just had a friendship with them and told them that, look here, I want to have a look at your ears. And they said, why? Because I said, because this has got an anatomical interest. So, I mean, they were quite happy. So I was sure that they are not going to harm me in any way, because otherwise, if I annoy these 5 people, then I'm in trouble. Now, you see, this is a cauliflower type ear. This is, and I saw virtually on all of them. Now, you might be surprised as to why it happens. I asked them, how do you get it? Although I knew it. But they said, we actually hold the other person by their ears. And then we try to, you see, overpower him. And that's why this ear is actually going, repeatedly, it's going trauma. And I believe you anatomists probably may be knowing more, that this cartilage gets broken, and then there's a regeneration, then gets broken, then regeneration. That's why these kinds of cauliflower ears. Now, these ears are very, very typical, and they can give away the profession at once. You have sometimes pierced ears, and you have earrings, you have other kind of ornaments over it, or also, which also give you away. This is from another person. And now, a very important thing is not that you can now take DNA from all prints. So you can take DNA from fingerprints, you can take DNA from ear prints, you can take DNA from limb prints. Now, if this DNA is recoverable, then it adds an amazing amount of dimension to it. So that just adds to the prints. That doesn't lessen the value of the prints, but that adds to the prints. And let us talk something about lip prints. Now, I told you, lip prints can be there in sexual crimes. They may be there like saliva, tooth marks, lip prints. And most often they are seen on glasses or cups or plates when this burglar has a desire to drink something from there. This has happened. This doesn't happen very commonly. So these ear prints or lip prints or these things are not going to come into use in everyday practice, but we must be aware that they are valuable, maybe in an offbeat case. Now, they have, you see, classified these prints into several these things. I wouldn't go into the details, but just the type 1, I mean, they call it type 1 and things like that, but what we have to see is sometimes these fissures or these cracks will run vertical and they will reach the other end. Then we call it type 1 or the vertical one, but sometimes they will not reach the other end. This is known as type 1 dash. Sometimes they are branched, then we call them actually type 2. Sometimes they are intersecting, we call them type 3. And then there are grooves that form a network. And then there's some miscellaneous types. Now, if you look at your lips carefully in front of a mirror, whenever you have some leisure time, you can actually identify which type you have, and you are not going to have the same type all over. Actually, you can actually divide the prints into 4 quadrants, and each quadrant might actually have different kinds. Now, this is a project which we gave to our students. I gave to my students, where we just applied some lipstick on the lips of all people, whether they're male or female, and then there's a paper, A4 paper. We just fold it, and then he just brings the folded portion here, and then he does it like this for some time, and then we can unfold it, and then we can get this kind of a print, which we try to study. Now, please remember, this is such a new branch that not much has been done on it. All the 4 or 5 prints that I'm going to talk about, nothing much has been done, and there's lots and lots of, scope for developing it. These might be given as thesis. I personally gave many students of my college ICMR projects on these. And some of these won good prizes also, surprisingly, only because this is such a nascent branch, such a new branch, that virtually whatever you do is going to be new to everyone. Now here, we just try to see whether these things really happen in real life or not. So this is what my classification was. I could see some vertical lines from here to the other side. So we call them type 1. Then I thought that these are the branched ones. Whatever you say, people are going to take it because nothing much has been done on it. So this is branched, so we called it type 2. And then here I found you are having some intersecting lines here, here, here. So we called it type 3 or intersecting. These types are not mine, but we just try to classify our subjects according to that. And then this appeared very much like reticulating to me. So we called it reticulating, and our project, it went on well. Now, this is how we actually divide the lips into 4 quadrants. And you have left upper, right upper, left upper, and like this. And as you can see, these prints are not same in all the thing. So according to each quadrant, you classify it, and then the overall profile becomes your real lip print. This is the real lip prints of one subject before applying the lipstick. Now you can actually see, if you stand before a mirror, you can actually see these kinds of things. See, this is a vertical line, but it is reaching the end. So I would take it as type 1 in the lower lip. However, the top lip is very much reticulated, type 4. So we can take it as type 4 in the top lip. Then something about nose prints. Now this is again very, very new branch, and you may not believe it, but nose prints actually started in animals. This is a very old photograph from 1930s, which was taken during a dog show. Now in dog show, you have thousands of, dogs, and there's every chance of these dogs getting mixed up. So at that time, there was a counter where they said, you bring your animal here and we will register their nose prints, so that in case of any mix-up, a wrong dog being claimed by a wrong owner, you can always go back to the prints and you can assign the right, dog to the person. So nose prints used to identify, these dogs. And in some cases, I personally know that in order to prevent people from stealing their cattle, they actually take nose prints of their cattle so that they can always go to the thief and say, look here, this is my cattle, and this is the nose print I have already taken. This has actually been done. This is a picture from the same competition where another dog is being nose printed. And this is from a cow. Actually, this is actually a particular clone. So they were just trying to see whether if we clone the same cow from several, whether these prints will be different. And surprisingly, these prints came different. You see these lip prints, these ear prints, and the tongue prints, which I'm going to talk about. They are not same even in identical twins. That's a very, very important thing. DNA, which is supposed to be such a good technique, that is identical in two identical twins. So sometimes your DNA profiling may fail when you are dealing with identical twins, but these kinds of prints may not fail. So if this corresponds to one nostril and the other thing corresponds to the other nostril, then this entire print actually belongs to this area. Now you might think as to how this can apply to humans. Now, to the best of my knowledge, this has never been applied in humans. But if one were to duplicate it, then the area to print would be this. But I've never done it. But again, it's a very new thing. Area to do will be this. But what we can do right now, and what is well known, is the various shapes of noses. So many shapes. And I will show you this is a picture from not a forensic journal, not an anatomical journal, but from a journal of plastic surgery. I was amazed to see that they have done a lot of work on these facial structures, especially the nose. They have, for example, measured the protrusion of the nose, the width of the nose, and they have found them quite unique. Another, picture from the same journal talks of a nasolabial angle. So this, if we take things like this, and if we make a comprehensive, comprehensive thing out of it, it's my belief that we can probably come to something big. So this is again a very, very nascent branch, and a lot of work needs to be done in this case. Palette prints is not so much unknown. We probably know it most about it. Anatomists know because of this peculiar palette, these grooves, these grooves which are there. Now, the interesting thing in this palette prints is that palette, being a structure which is inside the mouth, a murderer may mutilate your face, burn your face, but he would not think of mutilating the palate, because he doesn't know that you can actually be identified from palate also. I have seen in mass disasters, like even in air crashes, the body may be completely destroyed, but the palate is remarkably preserved. That's what I've seen. So you can actually retrieve the palate prints. However, the big problem is, suppose I go to a mutilated body, retrieve his palate prints. How am I sure that this is the same person whom we are looking for? So remember, one drawback of all these prints, I'm talking about drawback, not the advantage. The drawback of all these prints is that you need to have a print of the person whom you are trying to trace. Because if you don't have that print, you cannot compare. Now, fingerprints, you can always go back to the person's house. You can look for his articles, his comb, his toothbrush, his glasses, and probably you can take some fingerprints. Ear prints also you can probably take. But how you're going to get palate prints, that's a very big dilemma. Now, palate prints can be taken in one position that comes to my knowledge. People who have had dentures, when you make dentures, the dentists actually take a print of your entire palate actually, because the denture has to be fitted in a particular place. Now, if this person has a denture, now we are asking for a very big if. This is not, we got to admit that this is not going to be the normal case. But then you can definitely identify this person. So this is some cases which I tried to take pictures from, but unfortunately, our camera couldn't go inside the mouth. Otherwise, you would have got pictures like this. But we just took pictures from outside. So you can see these palette prints there, and they're very peculiar. This is from another subject, very peculiar prints. And this is a cast which I borrowed from a dentist friend of mine. So these are the prints which you can have, and I believe that the people who have had dentures will have prints like this. This is another cast. And then we'll talk about tongue prints. Now, tongue prints are not something which you are going to leave at the scene of crime, or, I mean, even if you touch your tongue with something, I'm not sure if any print will come. I think it will not come. Then how in the world is this tongue prints going to help you? Tongue prints are best used as a biometric aid. Just like I put my finger. Now these days there's a lot of scare that I will not come into contact with anything. I don't want to touch my thumb with something because of this corona or whatever. So what about sticking your tongue out? Because just like our friend Mr. Einstein, this picture was taken during a very famous conference when he stuck his tongue out. So you don't have to do anything, but you just stick your tongue out, and there will be a tongue scanner which will take your, tongue geometry from front. Now, tongue geometry is not the only thing that's going to help you. This is a profile geometry. Then there are some tongue surface cracks. So they are also very peculiar. And then the tongue surface textures. So if you actually combine all these into one particular thing, then we can probably have a good tongue print for biometric identification. So I think that's all with the presentation. And if you have any questions, we can take the questions now. Thank you very much. Any questions, I would be happy to take.
Can you please unmute me, Dr. Kaveri? This is Dr. Lakshmi Rajagopal.
You are unmuted, madam. I can listen to you.
Audible, madam.
So I would like to thank Dr. Anil Agarwal for a wonderful lecture on the role of...
You're muted now. Dr. Lakshmi, you're muted.
No, I'm trying. Okay, yeah, now I'm unmuted. Okay.
Yeah.
I would like to thank Dr. Anil Agarwal for a wonderful presentation on the role of prints in forensic identification. I would like to invite the chairpersons, Dr. Ashok Sahai sir and Dr. Brijendra Singh sir, to ask any questions or any comments that they would like to make. Dr. Ashok Sahai sir.
Thank you for calling me. It has been wonderful to listen to Dr. Anil Agarwal and to be with Dr. Lakshmi Rajagopal and Dr. Brijendra Singh. Dr. Anil Agarwal's talk has given us and the audience and all those who are connected in this session wonderful ideas and unknown, unthinkable aspects of identifying a person with the help of, say, palate, lips, lip prints, and nose, ear. I mean, could never think of all this. We have only known DNA fingerprinting and the dactylography, that is fingerprinting. But I have some queries. Well, I would like to understand, because I don't know, that is there any effect of pressure on the prints which are taken from the fingers or from the lips or from anywhere?
Thank you, Dr. Ashok. Yes, please.
Yeah.
You were asking something else. I'm sorry I interrupted.
No, I was just trying to understand if this will interfere with the accuracy of the printing.
That's actually a very good thought. And initially, I wanted to include this aspect of pressure, and I am very happy that you brought it out. Actually, I wanted to cut down my talk to just 1 hour, so I actually removed that. Pressures do have, you see, effect on all of these prints. Not only pressure, the sideway movement. If I touch my fingers on something and I unintentionally or probably intentionally slightly move it sideways,
Exactly.
even then, yes, you get a smudged fingerprint from where it becomes very difficult. And coming to your specific point of pressures, actually ear prints have been done. The studies that have been done are with light pressure, with moderate pressure, and with heavy pressure.
Very
Now they have done it, and even with fingerprints. Now with fingerprints, we are, because ear prints are almost 100 or 150 years old, so there's nothing much to discuss on this. Lots and lots of things have done on this. But ear prints and lip prints, I am sure, and I do know, because we know it from personal experience, having conducted experiments, that this does has effect. The effect that I am aware of, when you have a light pressure, less features tend to get, transferred onto paper. With moderate, there'll be more, but with great pressure, actually, paradoxically, the features will tend to get obliterated because everything tends to get flattened. So, so the best is probably moderate, but a very good question.
Thank you very much. Another thing I had in mind, that is digital fingerprinting better than the manual fingerprinting?
That's now it has come digital, and we all see that wherever we go, even in passport offices or wherever, they just ask our finger, and then sometimes they ask us to put a little bit more pressure. Now digital is always better because of the automatic comparison which computer can do. Now when we do it manually, we have to actually compare 10 or 12 or 16 points depending on what you have got. But computer does it everything very much out. You see, just how do we unlock our phone? We just put our thumb, and this phone automatically does it. So digital, I would believe, is far better, simply because of the automaticity that it provides.
Yeah. Thank you, Dr. Anil Agarwal. Any other questions?
Thank you, Dr. Sahai sir. Dr. Brijendra Singh, would you like to ask any questions or?
Yes, just I want to say that it was really a very wonderful and very evocative talk. We are using fingerprints commonly. We know that in all legal documents, our fingerprints are being taken. But this is a new vistas which should be involved in the medical teaching also. I think that is important. Thank you very much, sir.
Yeah, thank you, Dr. Brijendra Singh. Sir, Dr. Anil Agarwal, sir, as a moderator, I would like to say that the chat box is filled with compliments for your excellent talk, presentation, and thanks, sir. And in fact, Dr. Ramesh Babu has written that he used to write stories for children on forensic applications in Science Reporter.
I'm surprised. It is 1990s. He remembers from that point.
Anyway, sir, thank you very much for a wonderful lecture, sir. To summarize your lecture, in fact, you started with a beautiful setting of telling about stories about how to use various prints in forensic identification of the living as well as the dead. And also you mentioned about the various prints like ear prints or automorphology, the lip prints, the various classification of them, nose prints, palate prints, tongue prints, and tooth prints. You mentioned about so many of them. And in fact, it has opened a plethora of possibility for anatomists to work with the forensic scientists
Yes.
to bring out more research papers. In fact, the entire symposium today, CME today, is opened up a lot of research topics. So
It has been wonderful.
I'm sure it will benefit all the young anatomical researchers. And thank you very much, sir, for a wonderful presentation. And I thank the chairpersons also for Dr. Ashok Sahai sir and Dr. Vijendra Singh sir for chairing this session. And at the last, I would like to thank, I would like to thank the organizers for giving me an opportunity to be the moderator. Thank you very much. Over to you, Dr. Kaveri.
Thank you, ma'am.
Yes.
Thank you, everyone, for being with us. Now I take the opportunity on behalf of NATCON 68. I would like to present the certificate of appreciation to Dr. Ashok Sahai, Dr. Vijendra Singh, Professor Anil Agrawal, and Dr. Lakshmi Rajgopal now. Thank you. Thank you for being with us.
Thank you.
Thank you. Thank you very much.
Thank you, sir. Thank you, everybody, sir. Thank you.
Hi, Vijayan. How are you?
Blessing, sir. आपका आशीर्वाद.
बहुत अच्छा लग रहा है. Nice to see you.
Thank you, sir. Thank you. सभी गुरुओं का आशीर्वाद.
Sunil Dutt.
Thank you, Paul, sir. Thank you.
Thank you. Thank you. Thank you very much.
Now moving ahead to our next talk entitled Cortical Bone Microanatomy for Forensic Identification Purposes, Estimating Age at Death in Unknown Individual. And Dr. Julieta Gomez Garcia Donis will be presenting this topic to us. Dr. Prashant will be moderating the session. Dr. Prashant is a member of editorial board of various national and international journals and has the experience of chairing several scientific sessions. We are pleased to have you, sir, with us, and I request you to kindly invite and introduce the chairpersons of this session. Over to you, sir.
Good afternoon, all of you. Honorable Speaker, Dr. Julieta Gomez Garcia Donas, lecturer in forensic anthropology, Center of Anatomy for Human Identification, University of Dundee, UK. Both are Dr. Julieta, respected chairpersons, Dr. D.R. Singh, Emeritus Professor, King George Medical University, Lucknow. We also have Dr. G.P. Pal, Emeritus Professor, Modern Institute of Medical Sciences, Indore. Organizing Secretary, Dr. Punita Manik, and her team, friends, colleagues, ladies and gentlemen. Today, I look around and I am in awe with the galaxy of eminent luminaries gathered here, and I am deeply honored to introduce our today's chairpersons for this session, Dr. D.R. Singh, Emeritus Professor, King George Medical University, Lucknow. Sir, the former professor and head of Department of Anatomy, King George Medical University, Lucknow, UP. Former professor and head, Department of Anatomy, BP Koirala Institute of Health Sciences, Dharan, Nepal. Former professor and head, Nepalganj Medical College, Chispani, Nepal. Next slide, Kaveri. Kaveri, next slide, please.
Yes, sir.
Sir has authored 7 books. His recent book is Essentials of Anatomy for Dentistry Students. 2 editions have been translated into Turkish language. His achievements are Founder Fellow, Anatomical Society of India. He was awarded Hariom Ashram Alembic Research Award by MCI. He was awarded Dr. Dharam Narayan Memorial and Dr. Mehdi Hassan Gold Medal. Obtained fellowship from Indian Academy of Neurosciences in 2003. Awarded Dr. Sukhbir Prasad Jain and Dr. Shamim Singh Memorial Oration by Anatomical Society of India. An author, an outstanding researcher, a meticulous teacher of this country. Now I would like to introduce Dr. G.P. Pai, Emeritus Professor, Modern Institute of Medical Sciences, Indore. He's famous for his researches on biomechanics and load transmission of human spinal column. Renowned scientist across the globe. Kaveri, slide please. He has authored textbook of genetics, general anatomy, osteology, neuroanatomy, histology, dissector, and atlas. He has been honored Shanti Swarup Bhatnagar Prize for Science and Technology, one of the highest Indian science awards for his contributions to medical sciences in 1993. A very refined human being, very good friend of mine, a mentor and down-to-earth personality, Dr. G.P. Pai. May I kindly request Dr. D.R. Singh, Emeritus Professor, King George Medical University, Lucknow, to kindly introduce our today's guest speaker, Dr. Julieta Gomes Garcia Donas. Dr. D.R. Singh, please.
Good afternoon to everyone. It is my proud privilege to chair this session by our guest speaker, Dr. Julieta Gomez, who is a forensic anthropologist from the University of Edinburgh, Scotland. She is a lecturer and researcher at the Center of Anatomy and Human Identification. She has performed researches on sex, identity, ancestry, and age estimation, developing population specific standards, and she has specialized on age estimation using histomorphometry, that is the bone microstructure for forensic identification purposes. Now, most of us are aware about the forensic part of anatomy, in which we were told to identify the individuals. Am I audible?
Yes, sir. Yes.
So we were taught in our college days that the identification of a person is mainly done by the bones, and gross bones, and the other parameters, for example, the teeth, the pelvis. But sometimes, as we are all aware, that all these bones may not be available for identification, and therefore some more sophisticated methods are available now. And Dr. Julieta, who is expert in the forensic anthropology, will give us talk on the detail and of the bone histomorphometry and its role in identification of the individual. So I request now Dr. Julieta to deliver her talk.
Thank you very much.
Julita ma'am, kindly share your screen.
Yes. Can everyone see my screen now?
Yes.
Yes.
Good. Thank you so much for the kind introduction. I am so pleased to be here with you today. I am in Dundee, in Scotland. So it's 9 in the morning. It's very different than 3 in the afternoon for you. I'm still with the coffee. So today I am going to speak about the role of histomorphometry, as Dr. Singh said, and how we use histomorphometry for identification of humans, mostly in relation to age estimation. I would like to say also that histomorphometry is not only used for age estimation, but also for identification between animal and human remains, which is actually the first step that we do in the identification process, because if the remains turn to be animal, there is no medical legal investigation to be conducted. As it was said, I work in Dundee in the Center for Anatomy and Human Identification, and this is going to be just an introduction for you to know the studies, the principles, etc. So we start, the presentation will be organized and we will speak a little bit about bone tissue. I was not sure if the audience was, I am sure that they know what bone is, but I would like to remind or refresh the knowledge about what type of tissue we find, then the principles and the fundamentals of bone remodeling, which is the age estimation methods that we use are based on these principles. Then the main parameters that we use for age estimation, and we will review some methods that has been developed in forensic anthropology for estimating age at death. And finally, some advantages and limitations of histomorphometry for forensic identification. First of all, because I am not purely an anatomist, although we use anatomy for identification, I would like to introduce you what is our field. And what we do is based on biological anthropology. If we come back to the roots of the words, they all come from Greek, and is bios, that means life, and logia, that means the study of. And then if we said anthropos, it comes from anthropo, that means human in Greek, and logia, the study of. So biological anthropology is the study of humans, anthropos, based on their biological, physical, and behavioral characteristics. And then we have forensic anthropology. And what we are doing is to use the principles of biological anthropology applied for the medical legal context. For forensic anthropology, we don't tend to be called for remains that have soft tissue, like for example, individuals that are in the early stage of the composition, because that will be the work of the forensic pathologist, but we analyze skeletonized or semi-skeletonized remains, and we try to identify the individual. So biological forensic anthropology, will be biological anthropology applied to modern human populations for forensic identification. And when we said human, it depends a little bit, because depending on I am from Spain, for example, and in Spain, they consider that if the individual died 100 years ago, then it's a forensic case. But there are some cases, some countries, in which this will vary between 70 years and 100. If it is more than 100, for example, the remains will be considered archaeological and there will not be a medical legal investigation. Okay. Now that we know what forensic anthropology is, that is based in biological anthropology, what the forensic anthropologist does. And what we do is to construct the biological profile based on the skeletal remains, on the bones that we find. For the biological profile, we need to gather several pieces of information, and these are sex estimation, age estimation, ancestry, which is the biogeographical origin of the individual, and stature, the height. And because this presentation is about histomorphometry, we are going to focus on age estimation, in this case, on adults. And for this, we have macroscopic methods that are the first thing that we do, because these methods are easy to apply. You just need the bone and observe the surfaces. For example, for age estimation, we observe, as you have in this image, the auricular surface of the pelvis, and we see the degenerative changes, and that will give us an indication of the age. We also use, for example, a method developed by ISCAN, one very famous forensic anthropologist, the external end of the rib, as you can see on the image, and the calcification of the cartilage. And then what we do is to estimate age. Now, this is the first approach. Everyone knows how to do it, but then what happens when the remains are highly fragmented or taphonomically affected? By taphonomy, I mean that due to the deposition of the remains in the environment, the surfaces have been damaged, so we cannot observe anything. And the only option that we have DNA, other molecular methods, but the only option one of the only options, therefore, that we have is the microscopic approach. And this microscopic approach is based on the fact that what you do is you take the bone, cut a section, produce a thin section, and then you observe the microanatomy and microstructure of the bone under the microscope. Just a reminder for you that we have the gross anatomy, what we can see with our eyes in bones, and is the cortical bone and the trabecula. The trabecula localized mostly on the epiphysis, in this case in the femur, and the cortical bone varying in thickness. We have it mostly on the mid-sub and then around the whole bone because it's the outer hard layer. What we need to see, or what we would like to see, is within the microstructure, what happened with age in the bone. And there has been some recent studies that has used the trabecula for age estimation for medico-legal purposes, or for forensic science, let's say. But this is a more undeveloped area or field. And what we are going to talk about today, and is one of my specialties, is the cortical bone. Again, just as a brief refreshment, or because of how bone ages and how bone is produced, we need to remind the key functions of the three bone cells: osteoclasts, osteoblasts, and osteocytes. And this is important because at some point, when you see on the cortical bone, the ones responsible of this are these type of cells, and any imbalance of their activity will produce changes in the microanatomy and microstructures that we see. So osteoclasts are the ones that create a reabsorptive bay. They remove the mineralized matrix, and they are what we call them the bone resorbing cells. We have the osteoblasts that are the ones responsible for synthesis of organic and inorganic components. So these ones are the ones that will be the bone forming cells. They are depositing the bone. And then the osteoblasts can either die or become osteocytes. And the osteocytes are amazing because they are the ones that are regulating everything that happens in the bone once the bone has been formed. And these ones, therefore, are the responsible for the maintenance of the bone matrix. And I am telling you all this story because these cells are the responsible for the mechanism that we observe and we use to apply age estimation methods, which is the terms modeling and remodeling. And modeling is something that is mostly used, is mostly happening during growth, childhood, and in special circumstances in adulthood. And the modeling is these cells that are working together in order to change the size and the shape of the whole bone. So it's adapted, it's related to growth and adaptation. But what we are interested in forensic anthropology for age estimation is bone remodeling. And the remodeling is the coordinated and coupled activity of osteoblasts and osteoclasts. And these are, what they are doing is trying to maintain and repair the bone. You need to think that when the bone is already formed, then we live through our lives. And what happened is that the material and mechanical properties of bone start deteriorating. So osteoblasts and osteoclasts are trying to replace the existing bone by new bone that is better, has a better quality. If you see this table, we see that the remodeling is going to produce new bone will happen on all the bone envelopes, periosteal, endocortical, and intracortical, and also trabecular. Is a cyclical period, a cyclical phenomenon. And then it happens throughout the lifespan. And that is why we focus on histomorphometry for age estimation on bone remodeling. So I hope that this introduction about the type of skeletal tissues, bone cells, and modeling and remodeling will help us to understand the methods that we are going to review in the next slides. Age estimation methods are based on the physiological basis of cortical bone remodeling related to aging. And we know now that the bone is being replaced in a cyclical phenomenon. And the key structure that you probably know, all have heard about it, is the osteum. And that is a microstructure that we will see in cortical bone. Obviously, age estimation for forensic anthropology has its limitations, and we will discuss them later, but I would like you to have them in mind while I am reviewing the methods. And obviously, it's destructive. We need to cut the bone, and sometimes that is not possible. We have seen that it's population specific. That means that depending on the biogeographical origin of the individual, the remodeling, the bone turnover will be different. We know that there must be some sex-specific differences in bone microanatomy and microstructure, and that there are other factors such as pathology, diet, environment that will affect the microanatomy that we see. So have these limitations in mind so you can maybe later ask me some questions when I finish the presentation. And let's have a look at the osteum. And you see in this image, this is a cross-section of a bone. Is let's say a femur. You have the trabecula in the middle and then the cortical area around. And the osteum is this tubular structure that are like columns that form the cortical bone. And they have in the center a canal that we call the Haversian canal, because the osteum is also known as the Haversian system. And this Haversian canal houses blood vessels and nerves, and it's important because this will give or distribute the blood and nutrients that are necessary in this highly mineralized environment that is the cortical bone. As you can see, there are several, we will see it in the next slide, but there are several rings around the central canal, and this is lamellar. This is a beautiful image of one of our samples, and this is what we see actually in real bone is a rib of an individual, 45 years old. I just did this close-up of the osteum for you to know how it looks in real life, let's say. So you have the central canal here, then you can see the concentric lamella that is going around. These black dots are the osteocytes that are there, as we said, to maintain and repair the bone. And then, as you can see, there is a line that you can even draw. I don't know if you can see my mouse. Can you see it?
Yes.
Okay. So you see this line that goes along around the osteon, and this line is we call it the semen line. And it's very important for age estimation because this line will tell us if the osteon is a primary osteon or a secondary. A primary bone means that the bone has been deposited where there has been not resorption or remodeling. And secondary bone, that is the one that we use for age estimation of secondary osteons, means that they have followed the sequence of remodeling, which means activation, resorption, and formation. So this line, as you will see when we speak about the parameters that we use for age estimation, will be very important because it's also the delimitation between the osteonal structure and the remaining bone tissue. And this case is parallel lamellar. So what happened in the life cycle of human bone? What we see is that at the beginning we have primary bone. Well, we can start from the beginning, but we will say that first is a primary lamellar bone. Then some resorption happen. The osteons start appearing. As time passes, more resorption happens, and then we have a higher number of osteon appearing. And you can see in the diagram, in the lower diagram, what you see is from birth, we have the fetus birth 1, 2, 3, 6, and so on until 70 years. And what you can see is at the beginning, obviously the bone is still developing, but then after 6 years, for example, we have already, let's say, a thick cortical area, the osteons start appearing, and then finally the cortical area gets crowded with the osteonal structures. And we will use what we know that this is the life cycle of human bone in order to understand when we find an unknown individual, what was the age at death. So if we want to see it actually how we do it in histomorphometry, we will see that this is an individual. I am working with ribs. So this is a cross section of a rib. This is a 19-year-old individual. You see how thick and healthy the cortical area is. And then what we have in a close-up, I think that this image is beautiful. And what you can see is that all the, parallel lamellae are located, forming the cortical thickness. And then what we have is just few osteons, as you can see here, lying on this area. Now, as the individual ages, what will happen is that the cortical thickness at the end of 30 is still looking healthy, as you can see if you compare the cross section of the 19 years old with the cross section of the 30 years old. But if you compare actually the close-ups, what you can see is that the lamellae, parallel lamellae, circumferential lamella that we see on the younger individual is not anymore on the 30 years old. We have some remaining parts here on the endosteal envelope, on the periosteal envelope, but what we see mostly is osteons. You can see them that have been creating due to this remodeling process that we were discussing before. And now, as you start aging, if we, for example, are 56 years old, you can see that there is a huge difference in the cortical thickness between the 19 and the 30, and then the 56 years old is very, very thin. And what happened is that the trabecula start expanding, so it's eating up actually the bone. And what happened is that the osteons, as you can see, are more in number, and also they reduce their size because there is no more space as the cortical thickness has been reduced. So this is the life cycle of the bone as we see it in histomorphometry when we apply age estimation methods. Now, this is a young individual, a semi-polarized light. You can see the lamella, again, beautiful. This blue color is because it's polarized. And then you can see the osteons lying with some space in the middle. Actually, if I will ask you, to count the number of osteons, you will be able to do it, I am sure. No, because counting is something that we do for age estimations. 1, 2, 3, 4, 5, easy. So if I see this image, I will say that the individual is not old because I can see still space and the lamella and the osteons are big. Now, what happens when the individual gets older? That remodeling is happening cyclically. So this cycle will create osteons over osteons over osteons. And what we will have is this crazy image in which the osteons are all on the top of each other. And probably if I ask you to count the number of osteons here, it will take a little bit of time, I'm pretty sure. So what will happen is that we don't have any more the parallel lamellae that we were seeing before. The remodeling events or the traces of the remodeling are overlapping to each other. And what we have is what we call the interstitial lamellar bone. And this bone means that, imagine that we focus on this osteon over here. Okay, this is clear an osteon. This is another one. This is another one, but I don't know what is happening in this area, for example. It might be an osteon, it might be the lamella, etc. So this will be a problem in age estimation methods because with all individuals, there are some remodeling events that are underneath the latest event that we cannot see. And that will create a lower correlation between age and cortical bone microstructure. And just to give you as a glance, we have this individual that is 45 years old. These images are taken in the same magnification, and you see how thick, how the lamella is present, how there are osteons. There are many osteons, but there is still space between them. And then the 75 individual that has thin cortical area and the osteons are overlapping. So I have already told you some clues about the parameters that we will use, but for histomorphometry, and you will see it in the next slides, in the methods that we use, osteon number, the osteon is the key parameter, and osteon number is correlated to age. And it's because as age increases, the number of osteon increases. So one thing that we do, as I was commenting before, is to count the osteons. Normally in the methods, they will give you a field of view depending on the bone, and they will tell you how many osteons are there in relation to the area that you are observing. And this is what we call OPD, osteon population density, which is one of our key parameters to express or to reflect the remodeling events on the cortical bone. Other parameters that we use in forensic anthropology are cortical area, and we do is to measure the cortical area that we see. This is the rib, and also relative cortical area, which is another parameter that is independent of size. And then we take the osteon, the beautiful microstructure, and we take parameters on the osteon. We measure, for example, the osteon area, perimeter, and circularity. And we do this surrounding the semen line that we were discussing previously. And what happened is that the area and the perimeter it has been shown that reduces their size as the age of the individual increases. And some say that it's a matter of the cortical thickness getting very small, and therefore there is no space, and therefore the osteons are smaller. Some said that it's a matter of biomechanics. And circularity is very interesting because apparently what happens is that the more circular, the more resistant. So circularity actually increases as age increases. And then we do the same with the Haversian canal, area, perimeter, circularity. This is just for you to have an understanding of the parameters that we use and why we use them for age estimation. Why we use them is because the studies that have been performed have shown that there is a correlation with age. So you see here osteon OPD, the population density increases with age, cortical area decreases with age, as we have seen from 19 to 30 to 75, and then suddenly, as we see, osteon area decreases. Depending on the sample, on the statistics that we apply, on the characteristics, demographic characteristics of the individuals, we will have a higher or lower correlation, but this is what you expect from the parameters in relation to age. And what happened is that one of the first that understood about this relationship and was brave enough to create a method was Kerley from the United States in the 60s, excuse me. This first paper that we see here. And we will review him because it was the pillar of all the following methods. But you see that in this table, I have summarized that people have used, researchers have used different populations with different ancestry, Caucasians, mixed ethnicity, Asian populations. Then we have tried different bones, the femur, the tibia, the humerus, etc. And the parameters, as you can see in this column, go from 19 variables that Thomson used to just a few of them, like for example, OPD, Haversian canal, etc. We are trying to play with the parameters, with the sample and with the information that we have in order to improve our standard error of the estimate. That means that the lower the error, the higher the accuracy of the method to estimate age on an unknown individual. Let's review some methods. Here, for example, we have Kerley, that was the first one. He used 3 bones, the femur, the tibia, and the fibula. And as you can see, he decided to use anterior, posterior, medial, and lateral. And this is because I don't know how many osteons can fit in a femur, but in the ribs, there is in individuals over around 50, 60 years of age, the mean is around 300 osteons. So to measure, to count, imagine all the osteons in the femur is impossible. So what authors do is to pick sampling areas, representative sampling areas, to get the remodeling event from the whole cross section. Is the first method. It includes individuals, as you can see in the graph, from 0 to 100, and it gives us, there is a male overrepresentation with few females included in the sample, and therefore this will mean that if in the future you want to identify a female individual, you might have problems due to this imbalance in the original sample. And then the standard error of the estimate, remember that this tells us how accurate the age of the individual will be, is between 5 and 13 years, depending on the bone and depending on the parameter. This was the first method that produced a formula that we can apply when we have a case. And that means that I should be able to collect the data on my case, input my values in the formula, and that will give me the age estimate of the individual, of the skeleton. This is another method that is for you to show how the authors are playing with different sampling areas. This is still the same bone, but what you have is that in this case, the author is using just the anterior part of the femur and 5 sampling areas. They get creative with how we represent the parameters that we are using, for example, in this diagram that you see here. And then this author was able to show some sex differences between the cortical bone in her sample. The errors are from 9 to 12 years. And then this is a method on a Japanese sample. And I brought this up because instead of doing the plain microphotographs that we do, normally they were using microradiographs. And as you can see, you can also see the structures, the osteocytes, the osteons, etc. This method is only for males, and the standard error of the estimate is low, which is up to 8 years, lower than the previous studies. What happened is that all these studies use the humerus, the femur, and then at some point, some authors realized that if we use other bones that they don't have any loading, we will avoid the biomechanical influences, the remodeling due to biomechanical influences, like, for example, activity that is related to the femur. And they thought that the ribs are a good bone to test because the remodeling, it has biomechanical influences, but the remodeling is mostly constant due to respiration, and it should be constant across populations. There is some clinical data available that we can use to compare males and females, to compare pathological individuals and healthy individuals. And remember that some of our samples come from autopsies, from forensic cases. So collecting the rib is easier than collecting the femur when you are conducting an autopsy. And finally, it's easy to do the data collection, as I was saying, because in the femur you are forced almost to take sampling areas because the cortical thickness is very big. However, in the rib, you can just do all the collect area over all the cortical area. Collect the data, sorry, over all the cortical area. More examples about other methods, just on the ribs. The clavicle has been also used based on the same principles that it is not a weight-bearing bone, and it has an easy extraction. And you see that different populations, again, Polish, Mayan, Central Europeans, and different also accuracy for age estimation. And let's review a couple of papers. This is the first one that used the rib and the clavicle. As you can see, what we have is, this is the first method using these two bones and provided a formula for us to use. There is a sample of mixed ethnicity. Again, there is a male overrepresentation, and this is due to the nature of the sample because they were forensic cases. And then the age estimate is very similar to the femur. So this is telling us that the rib is also can be reliably used or can be a substitute to the femur if we want to do age estimation with histomorphometry. This is very interesting because it's one of the first studies comparing two different populations. And as you can see in the graph, we have the African Americans, which are the dots, and then the European Americans, which are the squares. And as you can see, the OPD, which is this variable that reflects the remodeling rates, differs between the two populations. And this tells us that probably we will need two different formulae for the two different populations in order to ensure accuracy for the age estimation. And this is a pilot study that we conducted. In this case, this is on the clavicle, on the Albanian sample. And it's also to show you that in this case, we compare Americans, white Americans versus Albanians. Albania is a Balkan country in Europe. And then what we have is that the dots are the Americans, and you can see that the remodeling rate in the Americans is much higher than the one that we observe in the Albanians, indicating that, again, there are some genetics, physical activity, diet that is making bone turnover different between the populations. So I hope you had an overview or an introduction on what we do with bone histomorphometry for forensic identification, and the methods that are available and the parameters that we use. And this is mostly using microanatomy and microstructure for identification purposes. Now, the advantages of histomorphometry, and I am super fan of histomorphometry, so I'm going to be very positive here, is that it is a suitable method and reliable method for identification, and is the only one that is left. It remains are fragmented or highly damaged. Then we have observed that it is giving us microscopic methods are giving us the same error as macroscopic methods, so we can use them reliably. You need to take into account that there has been studies, as we have seen, that have used different skeletal elements. Some use the clavicle, the rib, the humerus. So if you find the skeleton, you will have different possibilities. You are not just restricted to using one single bone. And then we know that there are samples, different samples that has been tested, and different methods based on the ancestry of the individual that has been developed. This means that if you have any assumption about the origin of the individual, you should go to this specific population-specific method and apply to your individual case in order to estimate age. So there are different options. There is something that we can observe in microscopy that macroscopically speaking, it might not be so clear, or yes, it depends on the case. But I would like to show you some examples of these cases. On the left side, you have two cross sections of rib. The cause of death was gastric cancer and brain cancer. And you can see, but I didn't know at the moment. Then I looked for the cause of death to know. But I was going to use them in the sample, and what happened is that these individuals were clearly microscopically pathological, but in the externally, we couldn't see anything. So what you see is that the bone, remember the thin sections, that the cross sections that we were looking at before, they were healthy, they were thick. These individuals are old. So you are expecting to have thin cortical area, but not that the cortical bone is eaten up. As you can see that there are gaps everywhere, both in this rib and in the brain cancer. The other thing that we can see macroscopically, for example, is the muscle attachments, and we use them to infer if the individual was highly active or not. We can also see that microscopically, and for example, this is a study that was testing the differences between the cortical area and remodeling in tennis players. And they realized that the playing arm, as you can see here, had a thicker cortical area and a higher remodeling than the non-playing arm, and this is due to activity. We can use histomorphometry, for example. It can be used to differentiate between populations or samples that were highly active and populations that were more sedentary, and we have seen differences in the remodeling rates. The challenges of bone histology are that, as we said, when the individual gets old, what happens is that at some point the cortical area is so packed with osteons that you can just see the last remodeling event. What is underneath is impossible to be collected. And this will give errors or a lower correlation between age and the microstructure for individuals mostly after 60 years of age. And we are working on how to improve that. Then there are differences between the sexes that we need to take into account. And sometimes the histomorphometric parameters that we use do not reveal that, statistically speaking. For example, we will assume that females will have higher intracortical porosity due to osteoporosis, due to menopause. And we will assume that that will happen after 56 years of age. And this is something that we will need also to control when creating the methods. It's a method that I just like it. I think that is very suitable, reliable, but you need a specific training, and you also need specific equipment. And although we have been trying to revise the methods or the technique for thin section preparation and make it cheaper and easier, it's still time consuming, and you still need a laboratory, a histology lab, in order to produce the sections. And finally, we have seen that there are methods developed in Koreans, in Japanese, in white Americans, Black Americans, etc. But still there are many populations that we need to test, because we know that pathology, diet, environment, physical activity, and genetics play a role in the microstructure and microanatomy on cortical bone. So we need to still explore what are the main differences and the main influences, and how we can use them in order to inform about the demographic characteristics of the individual. I think that that is me. I would like to thank you for listening, and I hope you enjoyed and understood me. And if you have any questions, I will be happy to answer. Thank you very much.
Thank you very much, madam.
Thank you.
May I request Dr. D.R. Singh for his comments on today's presentation, sir? Hello? Can you hear me, sir? Professor Dr. D.R. Singh, please.
Hello.
Yes, sir.
Hello, am I audible?
Yes, sir. For your expert comments, sir.
So let me thank Dr. Julieta Gomez for her beautiful talk on the bone histomorphometry, which is a new method and more precise for the identification of the age of the individual. This is a new method for identification of age, and in regard to the earlier methods, this appears more sophisticated and more fruitful. Thank you very much.
Thank you. My pleasure.
Dr. G.P. Pal, sir, any contribution?
Thank you, Dr. Natekar. And thanks, Dr. Julieta, for a very scientific talk, very much scientific talk it was. I enjoyed each and everything. I was expecting rather more of the technical thing, but you have covered a general aspect of the micrometry of, histomorphometry of this age determination from the cortical bones. You have almost answered what was there in my mind, including the population, variations, including the sex variation, including those of the external factors which are subjected in the microarchitecture of a cortical bone. I had many questions and you have just answered many of the things. But I will like to still ask something from you, ma'am. Now,
of course,
you have said so wonderfully about this, and one of the regret which I will carry with your talk is that I could not go in depth of the parameters which you have chosen to identify and how to exactly use it. Okay, you have just very broadly said that you will just count the osteons, the cortical thickness of the bone, and by that regression equation or regression formula, you will try to determine that. That's very good. Okay, very good. But I was expecting much more from your talk. That is there. But anyway, it was a wonderful talk. But I will like to ask you some of the question that, madam, in earlier, one of the earlier slide, you have said, or you have shown rather a black and white drawing, it was there, and you have shown that there were very few osteons along with the lamellar pattern. Okay, lamellar pattern was much more extensive as compared to that of the osteons which were present. Probably it was from the earlier age of the cortical bone. And then the question came to my mind that the osteons and the Haversian canals, they just carry the blood vessel, as you have said it. And when you said that in another of your slide, that the cortical area of the bone is full of the osteons, full of the osteons. There were very few lamellar bone or interstitial lamellas were still shown. You have very well shown that cement line. Okay. Then these blood vessels, if they are there in the osteons, they are running parallel to the long axis of the bone, okay, of a long bone. But then those lamellar patterns were not running parallel to the long axis. What may be the reason why there are so many lamellar bones were seen in one of your slides? Because we know that the long bones, as far as the human we are talking of human, they are vertically oriented and they transmit the load, as it is the main, region of the, main function of the long bones to transmit vertical load. But this lamellar bones, they were right angle to those of the osteon. That I could not understand, because then no blood vessels were seen which were parallelly running along with those lamellar bone, because osteons are meant just to carry the blood vessels in particular directions. Okay, more osteon, more blood vessel. less osteon, less blood. That way I see the histology of the bone. Okay, if you can tell something about that. That second question which I wanted to ask, which you have already very well explained, that why you choose the rib as compared to that of the other bones. Say for example, femur, you have also chosen the fibula at one place for this, and the clavicle you have chosen, but your, I think the best bone which you will prefer to use it was the rib. But as you yourself have said, the rib is not subjected to mechanical forces as the other bones are subjected to mechanical forces. But this fact itself is not, I mean to say, influencing the architecture of a bone. Less influence of the stress, there will be less architecture, I mean to say less osteon develop into the bone. Okay, I think yes, this is an important fact, ma'am. Just these are only two questions. I had so many questions, but let the others should also ask.
You can email me, feel free to email me and we can discuss it, or we can have a meeting, you and me. For the lamellar bone, and that there is, it might be not in, you are thinking about the long axis of the bone, and most of the images that I have there are from ribs, the costal element. So what you are seeing is the cross section of the rib, and it might not, because the rib goes like this, no, and we are cutting it like this. Maybe the lamellar bone is, this parallel lamellar bone is located at different angles because it's not the long axis of the femur. Yes. So most of the images that you see there are from my main research, that is ribs from the Mediterranean individuals. So that might be the difference in the microanatomy. For the other question on the rib are chosen, and as I said, they are doing still the femur, and we are trying almost every bone. There are methods on the occipital bone, on the mandible, and the rib is easy because there are 12 ribs. We have seen that if you use a standard rib, the remodeling rates are not going to vary much. Okay. So if you find an individual and ribs are difficult to identify and they are generally fragmented, yes, so you could potentially use any standard rib without introducing any error or bias for the remodeling. Then they are the easy extraction. They are not used for routine osteological osteometric, for example, that they are not measured like the femur it is normally for height or stature. And there are biomechanical influences, but what we are trying to look at for age estimation, I understand your point totally. For age estimation is trying to get away or avoid most of the other influences that we have, like for example, biomechanics, in order to have that remodeling that only happened in relation to age. Yes, because our main question is age. In the femur, for example, the femur has many advantages, but one of the disadvantages is that is a load-bearing bone, and what happens is that remodeling will be related to age, obviously, but also related to the physical activity. And for example, they have shown that the cortical area differs from anterior to posterior because in the posterior part of the femur is the linea aspera, and these muscle attachments are pulling and activating the remodeling. Do you understand? I am answering your question.
Yes, ma'am, to somewhat. The last question which I will like to ask, this you have not referred to any. This is a very specialized technique by which you can, you need a training. Okay.
Yeah.
I suppose if I want to determine the age with the histomorphometry, I will not be able to do it because it's a specialized technique. And then, but then it may vary from person to person, even who is expert. So what will be the inter and intra observer errors which you could have to see on this? Okay, ma'am.
That is a very, very nice question. I didn't have enough time to cover everything, but that is a question that has been shown, and there are some parameters that will present low inter-observer error and low intra-observer. Like, for example, I said that there are parameters that we measure the osteon area just surrounding the semen line. Those have some low inter-observer errors because you just take the image, use a software, measure the area, and click the button, no? And you have a metric. Now, there are other parameters, like for example, OPD, the identification of osteons that have been so very difficult because at some point there are so many osteons and you have the interstitial bone underneath that you don't know if it is an osteon, if it is not. So those have some high inter-observer error. And there are some authors that are trying to work on revising the definition of osteon, revising the way we take the parameters, like doing automatically. For example, I'm working on a project with the computing department now in Dundee for using artificial intelligence to identify the osteons, just to take out this high inter-observer error that we see.
Right.
But you are totally right. There will be some parameters that are easy to collect and low observer error, and others that are very challenging.
Thank
Nice
you.
question.
Thanks.
You're welcome.
Thank you very much, madam. Any questions from the audience? Let me summarize. Today's topic was histomorphometry of the bone in age estimation. Madam has discussed quantitative histological examination of undecalcified bone to obtain quantitative information on bone remodeling and structure. Osteological age is one of the key aspects in creating biological profile from human remains. Main goal is the systemic examination of cortical and trabecular age-related changes to explore age at death for individuals. Madam has described macroscopic and microscopic methods, modeling, remodeling, and describing the life cycle of the human bone. On behalf of Organizing Secretary and 68th NATCON of Anatomical Society of India, I am delighted to have with us Dr. Julieta Gomes Garcia Donas by gracing this occasion, participating and sharing her overview on microstructure of human bone versus animals and addressing key points on estimation of age-related changes, its applications, advantages, challenges for human identification. Madam Julieta has highlighted the state of art of histomorphometry, widening our horizon, keeping ourselves abreast with the recent advances at international level in the field of forensic anthropology. Muito obrigada por ter vindo, Madam Julieta.
Thank you very much.
I would like to thank our respected chairpersons, Dr. D.R. Singh and Dr. G.P. Pal, for their valuable contribution and their expertise comments. I would also like to thank the audience gathered to attend this very important topic with the valuable inputs. I would like to thank our organizing secretary, Dr. Punita Manik, Dr. Jyoti Chopra, Dr. Anita Rani, and her team for providing us an excellent academic feast. And we wish you all the best for the next 2 days to come. Thank you very much. Have a nice day. God bless you. Take care.
Thank you, Dr. Natekar.
Thank you, Natekar sir. Julieta ma'am, there was a question from the audience end for you. How do we process the bones for this?
There are many techniques used for this. There is the one that you have to have an isomer, which is a high precision machine to cut the bone. This is a bit too technical, but there are several papers. I have shown in the presentation a paper that we published, like doing it cheaper and faster. So, but what we have is we take the bone. You can do it even manually without any equipment. You take the bone. If it is a femur, for example, a femur, we will take the cross section, and you cut just a cross section which is perpendicular to the long axis. Okay, it has to be perpendicular, because if you cut it like that, the remodeling event will not be the same. Okay, so you cut it and you get the cross section, and then you use polishing paper, grinding paper, like you can do it manually wherever you are. grinding paper, the thin section that is around 2 or 3 millimeters thick, and then you start polishing in circular motion, and polishing and polishing and polishing. When you see that is getting like a white color, you put it under the microscope and you see if the light is transmitted, because the only thing that we need is the light to be transmitted through the thin section in order to see the microstructure. And then back, polish and polish it until you can see the osteons. You mount it in the microscope and you are good to go.
This is a ground sectioning, yes.
Yes, yes. This is a method that was developed. This is called the Frost Manual Rapid Method that was developed by Matt, revised by Matt et al. That they are in Netherlands. But if someone wants to contact me for further information or for data collection or whatever, I am super happy. I will be there.
Sure. Thank you.
No problem.
And now, on the behalf of Team NATCON 68, it is my great pleasure to present the certificate of appreciation to Dr. Julieta Gomez Garcia Donas, Dr. D.R. Singh, Dr. G.P. Pal,
Thank you.
and Dr. Prashant Natekar. Thank you once again.
Thank you.
Thank you, ma'am.
Thank you, sir.
Thank you.
So, coming to the last talk of today's event, entitled Role of Imaging Anatomy. And this talk will be delivered by Dr. Tanuj Kanchan. The moderator for this session is Dr. Ruchira Shetty, an experienced teacher, an enthusiastic educator, recipient of Lata N. Memorial Award by Anatomical Society of India. It is our privilege to have you here, ma'am. And I request you to kindly invite and introduce the chairpersons of this session. Over to you, ma'am.
Hello, am I audible?
Yes, ma'am, you're audible.
A very good afternoon to one and all. And thank you, Dr. Kaveri, for this nice introduction. First and foremost, I would just like to emphasize that putting the last session directly on the right time is actually commendable and appreciable for the whole team. And I introduce the chairpersons of the session, Dr. P.K. Sharma, sir, who is Professor and Head Department of Anatomy at AS University, Lucknow. Sir has been the former head of the Department of Anatomy at King George's Medical University, Lucknow, India. He has been an alumnus of KGMU itself, and he has been pioneer in developing the image anatomy lab, established unit of ultrasound, and the CT scan galleries, both 2D as well as 3D, and the MRI gallery in the Department of Anatomy at KGMU, Lucknow. Sir has also authored a book, Atlas of Imaging Anatomy. Sir has been a member of the National Academy of Medical Sciences in 1999. He has been the Executive Council of the Lucknow University, a member of it. Sir has been a member of the American Association of Anatomists, USA, and is a member of British Medical Ultrasound Society, UK. Sir has been a secretary, is a secretary of Alumni Association of University, has been elected the reviewer of Indian Journal of Experimental Biology, New Delhi, and he has been
Sir has been honored with the Seva Bhushan Award by the Governor of Uttar Pradesh for extraordinary services in the medical field. I welcome you, sir, for chairing the session here. The next chairperson is Dr. C.S. Ramesh Babu. He has been the ex-HOD Anatomy, LLRM Medical College, Meerut. He has been the ex-associate professor, MMC Medical College, Muzaffarnagar. His keen interest in radiological anatomy, clinical anatomy, endoscopic ultrasonography, and endobronchial ultrasonography. Sir has also authored a book, Essentials of Clinical Anatomy. Sir has been the member of the editorial board, Journal of Anatomical Society of India. He has been a reviewer of JASI, JASI, the Clinical Anatomy, Curious, Radiology, Case Reports, and many other journals. Sir has been awarded by Dr. Mehendi Hasan Memorial Gold Medal 2014 for the best paper presented in the field of clinical anatomy. I welcome you, sir, to chair the session.
Thank you.
I invite Dr. P.K. Sharma, sir, to introduce the guest speaker for the session for the day. Over to you, sir. Dr. P.K. Sharma, sir.
Thank you, Dr. Sethi. Good afternoon to all the listeners. It gives me immense pleasure introducing Dr. Tanuj Kanchan, Professor and Head, Department of Forensic Medicine, AIIMS, Jodhpur. Dr. Kanchan has a wide variety of experience as far as the forensic imaging anatomy is concerned. His topic is role of imaging in forensic. Dr. Kanchan has authored textbooks. Number one is Rapid Review of Toxicology. Another book is Atlas of Forensic Pathology. An overview. Dr. Kanchan has also contributed chapters in the Encyclopedia of Forensic Sciences, Encyclopedia of Forensic and Legal Medicine, and Encyclopedia of Archaeological Sciences. He has authored nearly 500 scientific articles in leading journals, out of which more than 5 PubMed. And awarded fellowship of International Association of Law and Forensic Sciences in 2019. Dr. Kanchan was elected as a member of National Academy of Medical Sciences in 2019. He is also editor-in-chief of the Journal of Anatomy, Journal of Indian Academy of Forensic Medicine, and he is Vice Dean Examination at AIIMS, Jodhpur. With these few words, I invite Dr. Kanchan for his talk.
Good morning, everybody. Good afternoon, everybody. Am I audible?
Yes.
Yes.
Is the presentation visible to all?
Yes.
Yes, sir.
And is the presentation working, moving? Is the PPT animation working?
So can you make it full screen, please?
You move the slides.
Slides are moving now.
Please put your slide in. Sir, put it in the slide mode, sir, please.
It's in a slide mode only. And
No.
Now it is shifted.
Now it is shifted.
Is it okay now?
Yes, sir. Yes, sir. Go ahead.
Okay. At the outset, I wish to thank the organizers of 68 NATCON of ASI and Professor Sharma for the very nice, humble introduction. So my topic for today is radiological imaging and identification. And I'll be covering this topic, the outline of which is mentioned here. I'll be talking about briefly about identification, identification of the dead, identification of the living, and then what are the traditional methods, a very brief comments on traditional methods used in identification. Then what are the newer alternatives? We have been using digital X-rays for long, but apart from that, we are using other techniques also now in identification. So I'll be talking about that, which includes conventional radiography in identification, ultrasonography in identification, computed tomography in identification, and MRI in identification. And I'll conclude with the antemortem and postmortem records matching in radiography for identification. So that is a brief outline of my talk, which I am going to start now. So I welcome you all to this presentation and thank you all for giving me an opportunity to present it before you from whatever little I know about the topic. So human identification is an essential component of forensic practice. It is, we all realize that unless a victim or accused is identified, the law cannot proceed in a particular direction. So we need to identify the victim, accused. We are talking about a criminal case. In civil cases, as in the earlier talks, people have pointed out that in civil cases also sometimes for claim of insurance, inheritance, people need to confirm the identity before the legal course or the processes, official processes can move on. So it is an essential component of forensic practice, which requires establishing the vital profile of.
Sir, kindly put your presentation in slideshow mode.
Yeah, should I continue?
So please put your presentation on slideshow.
Slideshow.
The slides are not moving, sir.
At lower right end, the last.
Is it coming now?
No, sir.
Yes, I tried this already. I'll exit and re-enter. Is it fine if I do that?
Sir,
Yes, sir.
you have to put your presentation in a slideshow mode.
I have put it on the slideshow mode. Set up slideshow.
So please stop sharing and then again start it. Maybe then it will come. Yes, sir.
Yes, sir. It has come now.
Okay. Yeah. So the outline of my presentation would be to talk about briefly its importance in living in death, traditional methods of identification, newer alternatives, that is the use of radiological techniques or imaging in identification, and which includes conventional radiography, ultrasonography, CT scans, and MRI. So, there is a, yeah. So what the identification is essential component of forensic practice, because unless I think there is some disturbance, somebody. There is disturbance. Yeah. So unless we can confirm the identity of the accused or the deceased, the law enforcement agencies cannot move forward in a particular direction. So it becomes very important for us to identify a particular individual. Usually the identification is based on visual identification features, but sometimes when we are brought with bundle of bones or skeletal material, dismembered remains. Can Dr. Kaveri put her on unmute thing, because I think the sound disturbances from that end. Can it be done? Thank you. Thank you. So identification, human identification involves establishing the biological profile of an individual. Biological profile of an individual means we are trying to examine the biological features to come to positively identify a particular individual based on the set of characteristics. So this is important both in living as well as the dead, and in civil as well as criminal cases. So in a couple of instances quoted by the previous speakers, especially Professor Agrawal, had quoted a case of Bhawal Sannyasi case where the person had to prove his identity to ensure that he gets his dues. Similarly, in other cases where dismembered remains are found, are recovered from sea, or they are excavated or exhumed, and then obviously we need to identify the person based on a set of characteristics to ascertain the cause of death, or to ascertain whether crime has occurred or not, or how the person died. So the circumstances of identification in the living can be civil cases or criminal cases. Civil cases includes marriage, inheritance, disputed sex, issuing of passport, license, cross-border migrations. When it comes to criminal cases, obviously, as I discussed, we need to identify the victim, we need to identify the accused. Then there may be cases of disputed paternity, impersonation, fraud, etc., where the question of identity comes in a case of living individual. Similarly, likewise, in the case of dead individual or human remains brought to us, then the circumstances can be insurance claims, inheritance, or unclaimed bodies for civil purposes. And in cases of a fresh unknown body brought to us for examination, or in cases of decomposed putrefied bodies, or badly mutilated charred bodies because of explosion or fire, or bodies found in mass disasters, which need to be identified for proper justice to be imparted to the deceased. So identification of the dead primarily involves generation of the biological profile, which comprises of identification of ancestry or race or ethnicity, which it was earlier known as. Then identification of the sex of the person, age of the person, stature of the person. Individualistic features, which includes what we have had discussion before, like some prints, or it can be acquired or congenital deformities, bony deformities, and then finally foolproof identification method of DNA profiling from the material which has been brought to us for examination. So of these essential elements of ascertaining biological profile of an individual, sex, age, and stature have been considered as big three of human identification. Sometimes big four of identification is also mentioned in books, traditional books like Krogman and all. which talk about big four of identification when they include ancestry with it. So ancestry, sex, age, and stature are taken as big four of identification. But with intermingling of populations and people migrating frequently for their jobs or settling abroad, or people moving all around the globe, the question of ancestry has become very difficult to answer in modern day samples. But that is why mostly when in a practical case we are not commenting on the ancestry. It is presumed that the remains belong to the place from where they have been procured, obtained, but that may not be true in every case. So that is the limitation. So mostly we talk about sex, age, and stature of human remains when they're brought to us for examination. And most of the research also in this regard is being conducted on these three features. DNA, we had a good talk in the beginning, in the opening session. And individualistic features also are very important, one of which is prints, which already has been talked about in one of the previous lectures. So traditional method of identification means we are going to observe the remain, its appearance, and we are going to examine the bones and then comment on the sex, age, and stature of the human skeletal remains which are brought to us. Obviously, for it, the knowledge of anatomy is very essential. And specifically because knowledge of anatomy is being used for crime solving purposes or medical legal purposes, then obviously forensic osteology and forensic anthropology play a vital role in this. With regard to application of forensic osteology, anthropology, and forensic medicine in solving these crimes or these cases or identification of the missing person in disasters, obviously collaborative practices and research is vital. That is why in most of the centers, including our center, whenever we have human remains brought to us for examination, we have an anatomist from our institution on board, and to give us expert advice on the question of identification of remains, so that justice can be done. And I feel in many institutions the same is a practice, where they take anatomists on board during examination of bundle of bones or skeletal remains, or sometimes human remains also. Likewise, even I see a lot of research from anatomy journals, which is of utility to forensic personnel. So that is what in today's talk I'll be quickly going through the various kind of research that has been conducted and the way forward with regard to that. So now, when you talk about examining visual identification or morphological morphometric examination of bones, there are a couple of things which come to mind, which can be a limitation in a way. When a bone comes to us, obviously direct examination inspection is the best way to comment on the age, sex, and stature from a bone. But when remains are brought to us, then obviously we require maceration process. We require a lot of cleaning, time-taking process has to be done, and then only we can examine the bone and comment on the elements of biological profile to establish the identification of an individual. An alternative can be if a human remain is brought to us or a headless body is brought to us, for example. So in a better, easier way would have been exposing it to radiological imaging, and then utilizing the database with us in identification. So with the advent of postmortem CT, postmortem radiography and all in many institutions, this has become quite a possibility. Otherwise, the traditional method was to macerate and examine the bones, measure them, look for the changes, look for the deformities, look for individualistic features, and then comment on the biological profile. So this is how things are evolving. And when it comes to research related to this field, so that we can move ahead in a positive way towards establishing the identity of an individual, we know that most of the research conducted to establish identity have been done on skeletal repositories, which have been there mostly in the western part of the world. Unfortunately, in India, I am not aware of any established skeletal repository where researchers can sit and work on various aspects of identification. but most of them are there. Most of them that have been researched on are there in the US and in the European Union. So one thing is, when you're talking about research, we need to have population-specific models. So suppose we are talking about crime solving or identification purposes in Indian subcontinent or in India or in various parts of India, then we need to have population-specific models for our own country. So that is not the case till now. People have been working, but there is a lack of collaboration, and because of which we do not have any population-specific models for our country, which obviously provides a limitation to the criminal investigating system. So one is lack of skeletal repository, and then the second thing is that there is a need of time and resources for maceration to acquire skeletal material on which research can be carried out. So lack of material and the process makes it even more complicated. So again, to overcome this, a very easy and very good alternative is to resort to newer alternatives of radiological techniques, which can be equally effective in developing identification process and developing database for our population, which as of now is lacking. Even if we consider the Western world, some people say that they have a repository, they are doing a lot of research, and that is how it is helpful. But even that can be questioned, because we know as the humans are evolving, so is their anatomical structure. So these repositories on which a lot of research has been carried out are mostly confined. They go back to decades. Okay, so some of the studies were done in 1920s or 30s or maybe even earlier. So there is always a question as to whether those data which were derived at that particular time using those repositories, or some people are studying on those repositories even now, can those outcomes or can those observations be applied to modern day population? That is a big question to answer. So even though we are lacking on repositories, I don't think as of today, if you want to generate a data, we are at a loss, because the modern day sample, any study done on the olden samples or age-old samples can give us an idea, but they cannot be applied in real case scenario in forensic practice of identification. So in consideration of all these things, obviously the use of radiological techniques can go a long way in identification of human remains. And then obviously conventional radiography, ultrasonography, computed tomography, and magnetic MRIs have been used. A lot of research is being carried out to help the experts in identification process. So talking about conventional radiography and identification, we see that radiographic techniques can give us a good assessment, good osteometric assessment. So it should be osteometric assessment, and also it is equally effective in assessing or in examining the morphological features. So talk about X-rays in sex determination. So these can provide us a good data about osteometric and osteological details. So some of the bones which have been commonly studied include skull, mandible, dentition, long bones, and pelvis. So talk about skull, later cephalometry has been used using, I have mentioned here some 19 landmarks, and these landmarks can be used for generating osteometric data, as well as to visualize and compare male and female differences in the features on the bone. Similarly, talk about sinuses. Posterior anterior cephalogram has been used to study frontal sinus and nasal septum pattern. Frontal sinus has been largely researched one for individualistic feature, because of the bilateral asymmetry in frontal sinuses between which differ in individuals, because of the shape of the sinuses, because of the size, because of its volume, because of its margins. So many features, and they differ from person to person. And these things have been studied between sexes also. So this is one feature which can have sexual dimorphism, help in that, and also help in more helpful in establishing the individuality of a person. Likewise, if you talk about sexing from teeth, various measurement of dental measurements have been taken on orthopentomograms, be it crown length, root length, total length of the tooth, or tooth width. They are all measured to establish, to compare the sexes. Besides total tooth length, there can be measurement of occlusal plane length, root length, total crown length. All these measurements have been taken on orthopentomogram to study the sex differences between individuals. Now, the basic logic or principle behind these sex differences, be it a bone or a teeth, is that the male and female, they differ in their size and build. So once they differ proportionately, obviously, the skeletal components and dental components also will differ in size. So that is the background of studying these measurements for making sex differences, sex differentiating models. So likewise, gonial angle, mandibular ramus, height, bigonal width, etc., have been utilized for sex differences between males and females. Come to pelvis, measurement of angles, subpubic angle, dimensions, pubic ramus, then dimension of ischium, pubis, different parts, symphysis pubis, its length and maximum minimal width. They all have been used and tried to establish sex differences in practice. Talk about other bones, long bones or smaller bones, phalanges, carpus, tarsus, metatarsus. Then again, linear and angular measurements have been used for metric comparison to establish sex differentiating models, which can help in identification. Now, coming next to use of X-rays or in age estimation. So, at this point of time, I must tell you that the role of X-rays or radiological techniques in sexing has a lot of limitations, and most important amongst the big three also, if I talk about it, is the age estimation. Because now you talk about age estimation, it can be based on degree of ossification, epiphyseal fusion, obliteration of sutures, dental maturation. So all these can help in age estimation and living as well as death. So when I talk about age estimation here, I want to divert or deviate from the topic, because whatever I talk about age estimation is going to be applicable to living as well as dead. So mostly when I talk about identification of human remains and all those things, we presume that we are going to discuss about the dead individuals. But in practice, if you talk about forensic practice or otherwise, forensic utility or in crime solving utility, we will realize that a very important feature of of identification in the living is age estimation. The problem of age estimation is to be solved in cases of civil as well as criminal cases. Civil, you talk about somebody participating in sports event, somebody wants to play under 12, under 16, under 18. And then there is a challenge, or there is a doubt as to the age of the person, because he should not get undue advantage. That means we need to know about the age of the living person. Sometimes the person is claiming to be juvenile, as was the case in even many murder cases and sexual assaults, where the person to escape punishment claims himself or herself to be juvenile. We need to comment on those individuals. Somebody is a victim of, claims to be a victim of sexual assault. And obviously age is factor, because if it is age is less than 18 years, then it will be taken as a sexual assault, even if it was with consent. So need to establish whether the person has reached the age of 18 years or not. Even in older individuals, when there are questions regarding the retirement age, pension, etc., then if you go to if you see in practice, then lot of radiographs are being done for age estimation in the living also. So whatever we are going to discuss here with regard to the research and practical utility of radiology in identification will be imperative to its applicability in both living as well as in the dead. So long bones, pelvis, clavicle, sternum, skull, and dentition have been used commonly for establishing the age of an individual. Talk about the routine practice of identification of the living. It is a practice to advise X-ray wrist, elbow, and pelvis. 3 X-rays are conducted in practice to establish age of a living individual, because these 3 X-rays together help in narrowing down the age to a minimum and help crime investigation agencies. To talk about age estimation from long bones, they undergo epiphyseal fusion as the individual grows, and these epiphyseal fusion stages can be visualized on X-rays, and subsequently age of individual can be estimated. Epiphyseal fusion of humerus, radius, ulna, femur, tibia, fibula are routinely studied using conventional radiography for identification purposes. As I told you, it is a practice to advise wrist, elbow, and pelvis X-ray to comment on the age of an individual, which is brought to us for medical legal examination. So a quick view at some of the X-rays. You can see a X-ray for child shoulder joint, and you can see head of the humerus has appeared. Other centers have not appeared, and epiphyseal fusion has not taken place. So it's a very young individual. It appears at the age of around one year. Then you look at the elbow joint. You can see many centers which are unfused in the lower end of humerus and upper end of ulna and the radius. You can see the lateral view, where you can see on the right side that even the lower end of ulna and all is not fused. So this is how we look at the centers when they are not fused. Look at the X-ray wrist joint, and we have lower end of radius and ulna. The centers have appeared, they are not fused. We can see 7 carpal bones, pisiform is missing. So it appears at the age of 10 to 12 years. That obviously means at least the person is aged less than 12 years. We can see the base of first metacarpal, then phalanges also, the centers are there which have not fused. So again, very important X-ray to comment on the age. Besides giving you an idea about this, important is that because of population variations and differences in the age of appearance and fusion of these centers in males and females, as also between the populations, there is a need to generate data on this, on our population, on our groups, so that it can be more effectively applied in forensic practice. So likewise, X-ray of knee joint, where you can see unfused, you can see the centers have appeared for the tibia, fibula, and femur, lower end of femur, upper end of tibia and fibula, but they have not fused. So these fusions, they occur at a particular time and particular age, and they have been utilized in estimating age and establishing the identity. The same thing can be done in dead individuals also by postmortem radiography or even CT imaging. Now we have evolved in terms of whether to say that fusion has occurred or not. So in olden days, still some people practice it now also. They used to classify all these centers as whether they have they are present or they have appeared or not, and they are fused or not. So the way of description of these interpretation of these X-rays used to be based on whether center has appeared or not, and if appeared, it has fused or not. But we now, with regard to fusion also, we have evolved by staging the degree of fusion. So sometimes we are the basic classification used to be 0, 1, 2. That means 0 is unfused, 1 is partially fused, and 2 is completely fused. Or then 3-stage classification, we moved on to 5-stage classification, where we talked about non-fusion to fusion, and then before fusion there is epiphyseal scar formation. So we look at that scar. So by examining all these stages and going into details of the fusion stages, we are trying to make an attempt to narrow down the age of the person, living or dead, so that we can help in investigation process. So likewise, a lot of recent research has been conducted on the medial end of clavicle for age estimation, because it gives us a good idea about the ages of 18 and 21 years, which are very medically very significant. So again, you can see from class 1 to class 5, different grades. Schematic diagram is there along with what you see on X-ray, how the centers have appeared and how they gradually fuse, and how epiphyseal scar also disappears gradually. So similarly, chest X-ray has been done for examining the various components of sternum, how they fuse from below upwards, and manubrium and mesosternum, how they fuse. Most of these fusions, they occur at a later age. That is why in common practice, these are not very commonly utilized. But in some setups, I know that in pensioners or when the age has to be confirmed for older individuals, then they go for X-ray of skull as well as X-ray of sternum, and they go for lateral view to examine that. So that is the practice in many places. About X-ray of pelvis for age estimation, you can compare the left and right views. You can see that ischiopubic rami is not obliterated or not fused in the left picture, whereas the right one it is fused. Same holds true for obliteration of triradiate cartilage on the left and right. You can see the difference. Talk about iliac crest, center has appeared for iliac crest has appeared here. It is missing on the left picture. It has appeared here. It is yet to be fused. So all these features, they are examined cumulatively to establish the age of the individual. Again, be it living or be it dead. Likewise, lesser research has been conducted on radiographic examination of thyroid complex. So we in our institution also had done MD thesis work on this, where a lot of work has been done on the hyoid apparatus, hyoid bone, how it fuses, unilateral, bilateral differences, differences in the shape and all. A lot of anatomy research has been done on that. But when it comes to talking about thyroid cricoid complex and even tracheal rings, then I need to emphasize here that it's a good research area for prospective researchers, because the calcification in the various parts of the thyrocricoid region, it is a good possible cause of identifying the age of the person as the person grows in age. So when you talk about long bones, pelvis, ossification centers, all those things, we are restricting ourselves to identifying the age of younger individuals. But when we talk about skeletal remains or human remains or dismembered remains or talk about disaster victims, then we are coming across all different ages. So now we need to develop methods of identification or age estimation in older individuals also. So obviously after age of 18, 20, 21 years, all the centers which we were talking about in the previous slides, they would have fused. So it is not going to help us much when it comes to age estimation. But then if the human remains are not completely skeletonized, if the thyroid apparatus or thyrocricoid apparatus is available to us, then calcification pattern in these can be studied to find out the age of the individuals. Likewise, from dentition also, age has been estimated using radiological technique, that is orthopentomogram. And recently, even when I'm going to talk about CT, the cone beam CT also has been utilized in estimating sex and age of individual from dental parameters. So age can be estimated based on the eruption pattern as well as degree of maturation. So talk about orthopentomogram, you can see that the second molar has just erupted, and eruption takes place at 12 to 14 years. So based on the dental eruption, the age of this OPG was identified as 12 years. And the real age also was 12 years. So the age range given to this orthopentomogram was 12 to 14 years. So again, because of its utility, a lot of research is being carried out on orthopentomograms for age estimation and sexing, as has been discussed in the previous slides. Now, likewise, besides eruption, this maturation of teeth also can help in estimation of age. A lot of methods have been derived. The most commonly researched method is demergence method, which talks about different stages of tooth development, which can be identified on radiographs, that is orthopentomograms. And then a cumulative score is given, which helps in identification of age of the individual. This is basically a domain of forensic odontology, if you want to subspecialize a specialty, and a lot of research has been conducted on in these areas. So these are the observations of demergence stages on the OPG, based on which a cumulative score can be given to identify the age of a person. A quick view at ultrasonography identification. Ultrasonography is the minimally used technique when it comes to identification. I have shown here a few research which has been done on age estimation using ultrasonography. You can see what is visualized on ultrasonography of proximal humerus ossification stages, and the same has been shown in line diagram for a better understanding. The reason for limited use of ultrasonography in age estimation process or identification is simple. You need a lot of expertise for it to be done. When it is compared to conventional radiography and CT scans and MRI, etc. So it needs expertise, and then area specific in dead bodies, or you're talking about remains, it is not always feasible to use ultrasonography machine to identify it for age estimation. However, studies have been done for its use in the living individuals. As I told you, age estimation process is important in living as well as in the dead. So it has been tried successfully for age estimation in the living. The same way, proximal ulna has been used for age estimation. Base of first metacarpal, fused and fused, they have been used by ultrasonography. So with that, we move on to the most researched technique in radiology these days, that is computed tomography and its use in identification. So before going on to the same things, because whatever we have discussed till now in terms of conventional radiography, almost similar things are going to be discussed regarding the utility of 3D CT images in identification process. Before that, I'll just quickly mention how exactly we are going to use softwares for studying the various parameters and indices derive them for identification purposes. So these are some of the softwares which can be used for reconstruction, visualization of 3D CT images, of which at our place we go for Singoware and Radiant DICOM 3D Slicer have been used in our center. Other methods are there. Most of them are paid. So you have, if you are doing a research on this, you be in touch with the radiology department, and then they will guide you as to what software they have with them. A couple of them are free also. Some of them are available for trial version for 60 days, which is enough for carrying out some research. So this is a background for those who are interested in carrying out research related to CT images. So this is how we do image processing. Before we go on to read and study the features, individual features and general features for identification purposes. So talk about image processing. We load the image from DICOM database, select bone window and crop image to obtain area of interest. And then we define threshold, which is not always essential in all the softwares. Then generate 3D volume reconstructed image, use scissor and eraser tool to remove interfering bone remnants, and then save the image with area of interest intact with us for viewing it using the camera tool and further analysis. The same system which has to be followed has been shown in here. So we have an image loaded from DICOM database. Then we are now using crop image to obtain area of interest and to generate 3D volume reconstructed image. So we are using a scissor tool here to remove the remnants or the areas which are not interesting to us. So for example, in this particular case, we wanted to study about mandible. So we are trying to remove the other areas, and this is how it goes. Finally, we have mandible before us. The biggest advantage of CT imaging is that it gives you information very similar to the bone, very similar to the information which you could have obtained on the bone. It is a 3D image. You can go for metric measurements and also morphological examination of features, be it position of mental foramen, the shape, dentition, processes, whatsoever you want to study. So again, we will define the role of CT in sex determination as well as age estimation. So assessment of sexually dimorphic morphological features and morphometric analysis of skull, mandible, long bones, sternum and pelvis has been researched a lot. So you can study or you can comment if it has come for a medical legal purpose. You can comment on the sex based on morphological features, be it prominent glabella or distinct frontonasal angulation or gonial angles or eminences. Or if you this is from the lateral view, you talk about the frontal view, shape of the orbits, again the nasal aperture, foramina, dentition, all those things, basically morphological features can be studied. And whatever we usually do for sexing based from bones directly, the same comments can be made on post-mortem CT examination based on these features. Talk about measurements. Numerous measurements have been utilized for sexing from bone as well as CT, which includes maximal length of the cranium, width of the cranium, bizygomatic diameter, upper facial width. These are some of the common measurements which have been utilized in sexing of an individual. And same measurements, many of these measurements have also been utilized for stature estimation. So bregma height, bimaxillary diameter, biorbital diameter, interorbital breadth, nasion, prostheon, nasal breadth, all these measurements, orbital height, orbital breadth, they have been utilized in sexing, along with some measurements in the base of the skull, which includes dimension of the foramen magnum, which is the most popularly or most commonly studied parameter for sexing. Even the area of foramen magnum has been studied for differences in male and females. So bimastoid diameter, biauricular breadth also have been studied for sexing of an individual. So likewise, different measurements have been taken on the mandible to look for differences in amongst males and females. Similarly, for long bones, maximum length, diameter, muscle attachment sites, architecture, and all has been. So basically, morphological and osteometric measurements have been taken in long bones too, and other bones for differentiating males and females. From sternum, how they derive a lot of indices, ratio between manubrium, mesosternum, and then individual dimensions, and then differences in the dimensions to be cause for deriving gender of an individual, sex of an individual. Similarly, measurements, different indices can be derived from pelvic bone, pelvis. One of them is AP index, where different measurements can be taken on the CT scans and indices can be derived. Sacrum, length and width of sacrum, and then again lot of indices are there in sacrum to identify the sex of a person. Now talk about use of CT in age estimation. CT is not commonly employed in the living, owing to the ionization hazards and all. But with the advent of postmortem CT imaging, or even some centers are developing autopsy centers, use of CT in age estimation is going to be increasing in the days to come. So okay. So again, the same thing which we saw in conventional radiography, skull, pelvis, clavicle, sternum, ribs, and vertebrae has been used, commonly studied for age estimation process, and results have been very encouraging. When it comes to a skull, we go for obliteration of the skull, sutures, and then it gives us an estimate of the age, though it gives a broad range. The important thing here, or the useful thing of CT over radiography, conventional radiography here, is that you can again crop this and go for we know that the fusion differs from endocranial and ectocranial obliteration is different. So endocranially it occurs earlier than ectocranial obliteration. So advantage of CT is that whatever area of interest, whatever you want to see, you can zoom it, you can crop it, or you can see even without dissecting or without destruction of the bone, or something which you cannot see on radiography, conventional radiography, you can see on CT scan, and you can comment on that, which will help in age estimation. Likewise, you can see CT images of medial end of clavicle and the schematic representation of it on the right side to show how detailed in what great detail you can see the center appearing, and then process of fusion and its utility in age estimation can be obviously can be very helpful in identification. Likewise, a CT image of sternum, you can see the segments because they are fused from below upwards. You can see the last segment has fused and the upper segments are in process of fusion. Manubrium with mesosternum has not yet obliterated, not yet fused. So same way, CT scans can be helpful in estimating age from the ribs, from the vertebrae, from visualizing the osteophytes. Besides metric measurements and what you saw regarding obliteration, fusion of sutures and centers respectively, Then there are morphological features which can be studied in great detail. One of the commonly utilized features on pelvis that is used for age estimation in elder individuals. Now, Sushibrook method is one of the common methods used on bones, and it has been studied now that if we use CT, then cropping the image and studying the same features exactly, they can be visualized in a very good way to comment on the age of the person based on Sushibrook criteria, and there are different other criteria, other methods also to look for to examine or study pelvic bone to help in age estimation. So all in all, morphological features also can be studied in great detail to comment on the age of an individual. So if you talk about acetabulum, which obviously you cannot go in detail in conventional radiography. There is a method given by Kelsey method, which talks about groove osteophyte development and apex activity. So if you look at this picture of a young individual as per this method, you can see a grooving is minimal. Here, little osteophyte development is there, and then apex activity also is minimal. Talk about it in a middle-aged individual, you can see a groove is pronounced. Okay, compare it with the young individual, groove is more pronounced, osteophyte development is more pronounced, and apex activity is also moderate. And then in older individual, there is a very pronounced groove, there's distracted rim and excessive apex activity. So all these things can be studied, databases can be generated. We have our PhDs who are working on these methods for age estimation, and these can be very useful in age estimation of the dead individual. And when remains are brought to us for examination, because it escapes the tedious maceration process, which is not always possible when bones are not brought to us. Likewise, talk about iliac crest age estimation. You can see various stages, right from apophyseal center appearance to ossification center, its partial union, and then commencement of the fusion, and almost complete fusion of the center. So lastly, we are coming to MRI, which also is not as commonly researched area as compared to CT, and even in practice it is not that commonly used. But latest research shows that MRI can be equally or even better when compared to CT because of the lesser known ionization hazards when it comes to age estimation in forensic identification. So some of the uncommon things which have been studied for sexing and all, I have marked from a couple of papers which I went through, where they have studied ulnar groove width and depth for sexing. Groove opening angle has been calculated in MRIs to comment on the sexing from ulna, which obviously we cannot think of all these things unless we have these advanced techniques. Then extensor carpi ulnaris thickness and width, ulnar solid process length and base width has been used in sex estimation. Then diploic, the thickness has been studied, which can have importance in identification as well as in the field of traumatology and in field of studying the mechanism of injuries caused to various regions of the head. So that can be researched. Then sinuses can be studied in MRI for its individualistic features. And then bones like patella and all have been used for different measurements have been utilized for sexing from individual. Likewise, you talk about age estimation, similar to CT, we get to see similar appearances of, talk about dentition, talk about appearance of ossification center, scar formation or complete fusion. We see all those things, MRI in the same way as we see on CT. Here you see how undeveloped manubrium and developed manubrium look like on MRI. You look at the epiphyseal centers which have appeared not fused in case of long bones, which can help in age estimation process. So apophysis, their fusion, all those things can be studied. So lastly, radiology also can help in individualistic features which can help in identification process to a great bit. And this is primarily based on the principle that you are going to match the feature or identify a feature in postmortem radiography that can be matched with antemortem records. So because conventional radiography is very common And if there is suspicion of somebody being died or you want some, obviously antemortem data is there. That means you want to confirm the identity of a person. Then it has to be matched with post-mortem images, and post-mortem images can help in identification by virtue of sometimes we get to know about joint or dental processes. Even if when we are conducting autopsy or when we are examining a bone, we sometimes have specific fixators or nails which have been used, which have mark of the company on them, which have been helpful in identifying the person later on based on those features. So joint dental processes and dental fillings and all, they can help in identification. Matching can help. Similarly, calluses, deformities, caries, osteomas, etc. If there is a record with us of a missing individual and we find some things on radiography, they can be matched and entity can be confirmed. I remember of a case of a foreign individual where we were brought with bundle of bones for examination, and we could see lumbosacral transitional vertebra there. And when they brought to us, because it was completely skeletonized, when they brought to us from embassy some of the x-rays and dental x-rays, as well as because he had some spine problem also. So it helped us in confirming the identity of that Spanish individual. So that is how it goes. So that is when radiology has importance in comparing the antemortem postmortem data. When it comes to our research or anatomic role, then obviously anatomical variations, as I talked about, transitional vertebra, morphological and other features, if they have a proper record has been kept, antemortem record has been kept, then the same can be matched with the postmortem radiography or postmortem CT imaging, and the same can help in identifying a person. So to conclude, neuroradiological techniques can play a significant role in human identification process in both living as well as the dead. That is the outcome. We need to develop population-specific models. And that is not a very difficult task considering the database which we can generate if we have a collaborative research. So collaborative research and practices is the key to move forward. Thank you very much for the patient hearing. And if you have any questions, I'm open for that with the little knowledge which I have on the topic. Thank you.
Thank you, sir. May I request Dr. P.K. Sharma, sir, to share his valuable comments, taking into consideration his vast expertise in this field. Over to you, sir.
Thank you very much, Dr. Kanchan, for a very elaborative and extensive talk about radiological imaging and identification. And I could see that you talked about age, sex, and stature as far as autopsy is concerned. But what particularly I'm interested in, I was more looking into that, say, for example, number one, my question is that what is the scene, what is scenario of the virtual autopsy in India? And as far as this availability of the infrastructure is concerned, I may tell you that at our at the only in the Department of Forensic Medicine, X-ray is available. These are not available. But what I have heard that as far as the AIIMS Delhi is concerned, they are coming up nowadays, not only with the X-ray, but they are going to have their own in the forensic department, their CT scan, and they are trying to develop it. What is the scene in the different as far as these modalities are concerned? Particularly, they are very useful for the cause of the death. So what is the scene? What is the available data as far as the India and across the across the globe is concerned? So this is what I'm interested in.
So thank you very much, sir, for the question. Thank you very much, Sharma sir. So you are very right. The scenario is again very encouraging as of now. AIMS New Delhi, as you are already telling, AIMS New Delhi has already developed a Varchopsy center last year, and they have already installed the required machinery as well as the technical help which they require. So they have started to work on that. At the initial stage, they are working on unknown individuals, and they are trying to standardize the various methods. So they have not taken it into practice. If I must say, as you rightly pointed out, it has a very, very vast role in identifying the cause of death and in terms of making it a more human way of conducting autopsies. So the problem is that it is the cost involved is too high. So as of now, AIMS Delhi already has established a center, and NIGRIMS Shillong, they already have started to develop the center. They have procured all the funds and all the permissions from the respective governments. So things are moving forward. I think the premium institutions will take a lead in developing this. When it comes to despite of its utility, another problem with apart from funds, why these centers are not developing in India is its role or its acceptability in the courts of law. So as long as our investigating authorities and our judicial system does not permit us to believe in that particular system, that whatever we are coming out with, whatever we are observing in those things are equally effective as what we see on gross examination in autopsy. So those things are to be worked on, but things are moving in a positive direction. But there is a time to go for that, I believe.
One more query is there that I may tell you an example of a lady who is known to me. She was living there in England, and she died, just collapsed on the bed. And after that, virtual autopsy was done, and she was diagnosed to be suffered from pulmonary embolism. And the case was solved, that the cause of death was this. And if we know the cause of death during then we will be able to longer the life. So the point is this cause of death should be known, and if we can adopt this as far as our scene or in India is concerned, and we will be enhance the number of years which we can live. So with these few words, thank you very much, Dr. Kanchan, for an elaborative talk, and many more things to learn in this future too. Thank you very much.
Thank you so much, sir. Thank you very much.
Over to
may I request Dr. Ramesh Babu sir to please give his valuable comments on this.
Thank you, Dr. Ruchira. Thank you, Dr. Tanuj Kanchan, for your excellent presentation of use of CT and MRI and other radiological imaging methods for estimation of the age. And as Dr. P.K. Sharma sir has asked you about virtopsy, and it is very good to know that in India also 2 centers have already started establishing facilities for this method of virtual autopsy. And you have given very elaborately the age estimation by CT, and CT is available nowadays in many centers, and anatomists can utilize this methodology for estimation of the age and as well as estimation of the sex, and study about the sexual dimorphism of various bones. And you have stimulated the interest of many young anatomists by your excellent talk. Thank you, Dr. Kanchan. Thank you, Dr. Ruchira.
Thank you, sir. Thank you, sir. I don't think so there is any specific question in the chat box. I would just like to summarize what we get from the topic here. Regarding the role of the imaging in the identification, I mean, human identification has always been a challenge, and it's becoming more because of the variety and the type of crime and the criminals adding on to it. The sir has beautifully explained how this identification can be utilized both for the civil as well as criminal cases. The use of the biological profile, how it can be created by the use of the imaging, especially the three starred components of it, whether it is age, sex, and stature, how they can be well defined with the conventional radiography as well as with the enhanced modalities using the CT scan and the MRIs. Though ultrasonography is also into it, but with the minimal use of it. The limitations of the lack of repository, skeletal repositories, time and resources has added on to the addition of the imaging into this area or this avenue where identification can be well balanced by the use of the imaging modalities. And how these components that we derive from the imaging thing can be compared from the antemortem and postmortem and the identity of the disease as well as the accused, both can be taken into consideration. Thank you, Dr. Tanuj Kanchan, for a very nice, elaborate and very explicit images that could be visualized in the presentation itself. Thank you, sir. Thank you very much.
Thank you.
I sincerely thank Dr. P.K. Sharma, sir, and Dr. Ramesh Babu, sir, who with their expertise into this area, they've added further points into their into this talk. Thank you, sir. Thank you very much. And I take this opportunity to thank the organizing committee, Dr. Punita Manek, Dr. Jyoti Chopra, Dr. Anita ma'am, and their coordinated and expert team who has taken up to this level. Thank you, one and all. Thanks to the audience. Over to you, Dr. Kaveri. Dr. Kaveri, can you hear me?
Thank you, everyone. We appreciate your presence and support. And with great pride, now I would like to present the certificate of appreciation on the behalf of Team NATCON 68 to Dr. Tanuj Kanchan, Dr. P.K. Sharma, Dr. C.S. Ramesh Babu, and Dr. Ruchira Sethi. I am thankful to each and everyone here for your patience and support. We appreciate it. And now I invite Dr. Punita Manek to conclude the session.
Thank you.
Thank you.
Good evening.
Good evening, ma'am.
Thank you for being with us. I would say that all the presentations were mesmerizing, excellent, and added so much to our existing knowledge. So much to explore in anatomy that can be linked to forensic. We thank the Almighty for helping us, guiding us through the day in smooth conduction of today's function. Many, many thanks to our experts, Dr. K. Thangraj, Professor Anil Agarwal, Dr. Keval Krishnan, Dr. Tobias Hulton, Dr. Julita, and Dr. Tanuj Kanchan. I have so much to say which cannot be actually expressed in words. It is our honor that you have been there throughout the day despite your busy schedule and your commitments. So gratitude from the organizing committee to all the expert faculty members for sharing their knowledge to this vast gathering. I also thank the chairpersons and moderators of the sessions for their kind support, and I hope that all the delegates have enjoyed the variety of sessions brought forth today. Please be with us for tomorrow's scientific program. The link will be shared with you shortly. And tomorrow morning at 9 o'clock, we have with us Dr. Yogesh. He is from Jipmar, Pondicherry, and will be talking on the changing art of anatomy illustration and the potential of illustration software. So concluding here by saying that you have a wonderful evening ahead. See you till we begin tomorrow again. Thank you.
Thank you, ma'am.
Thank you.